Yeah, everything's fine. Yeah.
Great. Okay. So thank you for inviting me, and also thank you for accommodating my last minute change of schedule. It's much appreciated. And I'm sorry I couldn't be there in person, but it's always nice to be able to speak to people in any case. The problem with these online talks is that it's sort of difficult to see where, when the audience is losing you. So I will ask people to maybe raise their hands during the talk, and if I see your hand, or if one of the organizers can sort of indicate that somebody's raised their hand, I'm happy to stop and take questions.
You are happy to take few questions during the middle of the talk as they come up, and then maybe larger questions at the end.
Yeah. Just so that I prefer that everybody stays with me through the talk.
Great.
Okay, good. Let's get started. So as you, so XKCD, I was asked if I'm a fan of XKCD. I've always been since I first discovered it, but I find it keeps on. So Randall Munro somehow seems to know what I'm working on, because he keeps on coming up with cartoons that sort of speak directly to problems that I've been having in my research. So this is this cartoon here. Can you see my pointer by any chance? Maybe not.
Yes, we can see the pointer.
Okay, so here we go. So this is a sort of spoof of these old puzzles that we used to do in school a long time ago, where you were supposed to cross a river with a wolf, a goat, and a cabin. Typically, it's a herbivore, a carnivore, and some vegetables. Okay, and you have a boat, and the boat, obviously, you could all go on the boat, but then the wolf would eat the goat, and the goat would eat the cabbage. So you have to somehow go back and forth across this river and make sure that you get everybody across. So here's the XKCD cartoon. I need to cross the river. I have a wolf, a goat, and a cabbage. That's the standard, right? And sort of cue ball says, okay, here's what, because he knows the answer, but then the puzzle gets more complicated. Hi, I also need to cross. I have 2 wolves and 100 cabbages. I have 50 cabbage moths, 200 boat destroying termites. I have a wolf that can operate a boat and a goat that eats wolves. Hang on, I need to make a spreadsheet, and so on. Okay, so this is actually pretty much a visual abstract of the talk I'm going to give you today. And the transport steps we're going to be talking about are not, of course, a river, but inside a cell. And before I start, I should say that this work is done along with a former PhD student of mine, Soumya, who's now working at the IBS in Korea, and with a collaboration from a math student, Keshav Krishnan, who is now at UIUC. And you can reach me at this email ID if you have any questions about the talk. Okay. So I, as was indicated in the introduction, I think about eukaryotic cells. So eukaryotes, you all know, are cells like ours, or like plants, or single cell microbes like amoeba and ciliates and yeast and so on. But there's a huge diversity of eukaryotes, and people are really not aware of, the textbooks don't give the right impression about how broad this group is. And yes, they all have some characteristics, which is they have a nucleus, but there's many other things that they share. And of course, there's many other things that are different across all these different lineages of eukaryotes. So I like to think about this entire tapestry of eukaryotic diversity, which is, falls under this rubric of natural history. What are the cells that are alive today, and how are they related to cells that existed in the past? And we use bioinformatic approaches, phylogenetics, genomics, a lot of protein sequence reconstruction, and so on, to do that. And I had initially thought to give a talk about that, since this was a bioinformatics workshop, but then I decided to do something a little more different, so that you would learn something new, perhaps. So the second thing we think about is, so I'm a physicist, and as physicists, we like to think about how interactions between small components can lead to large emergent patterns and phenomena, dynamics. So the question we ask is, how do known molecular interactions between things that happen inside a cell set up the architecture, the fairly sophisticated architecture of a standard eukaryotic cell? And I'll show you some pictures to see just how complicated that beast is. And the third is actually a very difficult question. It's the why question, in the sense, what are the selective pressures, the evolutionary forces that happened over a very long time span that led to the present day cell and present day diversity of different cell types. So these, again, the three categories are what, natural history, how do molecules conspire to generate these patterns, and why, what exactly was the selection pressure that led to the present day. And of course, we don't have answers to, we have hardly any, we've only scratched the surface about this. So here's a broad overview of what I'm talking about. This timeline that you see in the box starts from the present day and sort of goes back in time over billions of years. So you started about 3.5 billion years ago, be slightly closer to the present, but around that time is when you have some evidence that there were already cellular structures and fossils that you find. And so the idea is that some sort of prokaryotic ancestor already existed at that time. Now, on present day Earth, there are two kinds of prokaryotes. These are organisms without nuclei, but they're very different from eukaryotes, and they're very different from each other. They're as different from each other as we are from them. And they're called archaea and bacteria. They're both called prokaryotes, but you should really keep in mind that it's bacteria and archaea, and these are two major kingdoms of life on Earth that are still existing. At some point around 2.5 billion years ago, the evidence of mineralization and oxidation states in rocks indicate that there was an increase in oxygen availability, which eventually actually changes the atmosphere of the Earth. And so this is, an example of a great episode of climate change, if you want to think of it that way. And the reason it happens is because of the invention of photosynthesis. So prior to this date, life exists and extracts energy based on chemical gradients. You want to think about, for example, hot springs, deep sea vents and things like that, where there's a lot of temperature, there's a lot of chemistry that the cell can make use of to survive. But with the invention of photosynthesis, then what you do is you directly take the heat from the sun, you take the light from the sun, and you take CO2 from the air, and you make sugars, which you can then use as a fuel to run all your other cellular processes. And you can make ATP, for example. And this leads to a byproduct, oxygen, which floods the atmosphere, and then leads to a niche for a new type of organism, which is a respiratory one. So the invention of oxidative respiration, the use of oxygen to more efficiently utilize carbon sources, comes into being. And again, these are all prokaryotes that do this. It's a prokaryotic world. At some point, about 2 billion years ago, maybe 1.5 billion years ago, there's a strange niche, again, somewhere in the Earth, perhaps in the North Sea, which is where present day lineages that represent that ancestral state seem to have been discovered, where a group of archaea, which are presently called the Loki archaeota, or the what is the new word? They're all named after Norse gods. Okay, so there's this big clade of archaea, which tend to live today in symbiosis with bacteria. They're studied with bacteria on their surface, and they do gas exchange, and this is how they actually survive. There's a symbiotic relationship. One of those, they're called the Asgard superphylum. So one of those ends up entering into a very tight symbiotic relationship, so tight that the bacterial partner ends up living inside the archaeal partner. And this is the first proto-eukaryote. The bacterium over there is actually what we see as the mitochondria today. It continues to do oxidative respiration. The archaeon provides the host environment, the cytoplasm, and all the transcriptional and translational DNA replication machinery, while the bacterium provides the metabolism. So that's it's not like a textbook where one cell went into another cell and it became a eukaryote. It happened over a period of millions of years, where a population of archaea and bacteria that were living together ended up cementing this symbiosis. So I'm interested in the period after that point. So what happens is eukaryotes then develop what we think of as the standard eukaryotic traits, not just the nucleus, but also these internal organelles, the ER, the Golgi, endosomes, and so on, cytoskeleton, the actin and tubulin cytoskeleton, changes of shape, sexual reproduction, mitosis, meiosis, all this stuff that eukaryotes invent happens after that stage. And then we reach this point called the last eukaryotic common ancestor, LICA, about 1.8 billion years ago, or slightly more recently. And that organism is called the last eukaryotic common ancestor because if you trace back all the existing eukaryotic lineages, that's the most recent ancestor that's common to all these. It doesn't mean it was the first eukaryote. In fact, it's much more recent than the first eukaryote. It's just that there are no descendants of those first organisms left today. Okay, so having given you that overview of the timeline, I'll just tell you some of the work we've done across the spectrum. So we've worked on these archaeal origins by using bioinformatic tools, using present-day archaeal genomes to see what fraction of eukaryotic genomes are made from archaeal components and what fraction from bacterial. We've looked at the integration of the bacterium with the host to become mitochondria. In particular, we've looked at the mitochondrial, so-called mitochondrial division machinery. Mitochondria actually divide inside the host, almost as if they were free living organisms. They have their own genomes, as you know. We've looked at the origin of endocytosis. And more recently, we've started thinking about how new compartments, not just the first protoeukaryote, but this whole full-blown apparatus of ER, Golgi, secretory vesicles, endocytic, endosomes, lysosomes, all those things came into being. The Golgi has been a particular favorite organelle of mine. And touching upon multicellularity, but it's not something I'm going to talk about today. Okay. So having given you the natural history, let's look at the second part of what we work on, which is the how. How do local molecular rules, small microscopic interactions, give rise to the sort of cellular state? So somehow we need to provide a theory for this, right? It's not enough just to hand wave and say, we know what these molecules do, and we know what the cell looks like, and there's a causal relationship. Often when you do genetics, you knock out a molecule, a snare protein or an ARF or whatever it is, and you get this very big phenotype. The whole organism has a big change. And if it's an animal, you see a macroscopic change just by eye. But that doesn't mean that the role of that molecule is to make that whole process, right? That molecule is working as part of a system. And when you break the molecule, you break the system. And then, of course, the consequences of that can be very huge. But the actual effect of the molecule is very local, which then has secondary consequences, tertiary, and it spreads through the system. So you want to build a theory that's able to take this local perturbation and connect it to something macroscopic. And so what I'm going to be describing to you today is an example of such a theory. There are, of course, many ways to do modeling, and I could teach an entire semester class on how you build a mathematical model of a system, how much to leave in, how much detail to leave out, how many approximations you need to make, and so on. But what I'm going to give you is an example of a theory that actually goes from micro to macro. And today's example actually, in a sense, is unusual because unlike typical models where you make the model and you see what it does, in this case, we've made the model and we can fully understand what it does. So in that sense, it's not just a model, but it's also a theory. And if we want, during the question and answer session after the talk, we can understand the difference between models and theories in that sense. So I'm going to pause, just check if there are any questions. And if not, take a few seconds. Okay, I don't see any hands up. Okay, so let's keep going. So this is a beautiful EM. If you actually look at the EMs from the 70s and 80s, they really are in many ways more detailed, more informative than what people look at today, because at that time, people were interested in exploring the structure of a cell in an open-ended way. Whereas typically today you use EM as a way to look at some particular subsystem of interest to you. And so you tend to lose the idea of the whole cell as a single entity. But so here's Pallady's very, very beautiful EMs. This is a typical eukaryotic cell. And when I say typical eukaryote, when you read a textbook, there's a book, for example, called Molecular Biology of the Cell. And immediately you should ask, which cell? And when you read that book, mainly they talk about eukaryotes. Okay, fair enough. But I already told you there's so many different kinds of eukaryotes, right? So you should say which eukaryote, which cell? Is it in a multicellular organism? If it is, then which cell type in a multicellular? And so you should never take anybody seriously when they make a claim that cells do this, unless they have some proof, which is that they've looked at lots of different cells and they all do this. Now, the features that you see in the cells I'm showing you here, every eukaryote has them. They have a nucleus, which is not shown over here. They have an endoplasmic reticulum studded with ribosomes, as you can see. They have these vacuoles. They have these small transport vesicles, which you see over here, which are these small circles. You see them beautifully budding out from some source compartment. And then they have, all eukaryotes also have this structure. It may not always look like this, but it's called the Golgi apparatus. Okay, so I mean, this is just a little slice of a cell, and you can see how much is going on, right? Now let's zoom in on that vesicle which I showed you. It's just 40 nanometers, 40, 50 nanometers. This sort of greenish yellow color on the inside is a lipid bilayer. So this vesicle is a hollow ball, and on the inside it will contain all kinds of chemicals. And on its surface, it has these transmembrane proteins, lots of them, and membrane-associated proteins. And every single vesicle will be covered with a lot of different proteins. Some important ones that will come up later are these SNAPs and these synaptotagments and so on. These are called snare proteins, which will come up later in the talk. Now, the point about this is first to show, yes, it's complicated, but also that there's some reason why these transport vesicles have so much decoration on the surface. And the reason is because these are almost like an Amazon package, right? So if you pick up an Amazon package anywhere in its delivery process, you'll find that it has a QR code or some other kind of barcode that digitally will tell you where it came from, where it's going. And therefore, anybody locally can pick it up and make sure it gets to the correct destination next. So just like the package itself contains its identity and contains its instructions, the vesicle itself contains its own instructions. So no matter where the vesicle was made, as long as you've made it correctly, it's going to go to the right place. And I'll show you a little bit more what I mean by go to the right place. Okay, so a eukaryotic cell has many compartments. These are all different. There's a hand up. Yeah, I'll take a question.
Yeah, I had a quick question. To kind of follow up on the Amazon package analogy.
Yeah.
So if like a, there's like a cell lineage, right? Like, especially in the immune system, right? They differentiate.
Yeah.
And so I was wondering, can you also find out where it is in its differentiation timeline, right?
Can you find out where the cell is? But this is not a cell. This is a vesicle inside a cell.
Okay.
Right?
Okay.
The cell is huge. The vesicle is tiny. It's just 40 nanometers. And these vesicles will be pretty much the same no matter which cell you're looking at. They'll have slight differences in composition, but it's remarkably similar between humans and ciliates and plants and yeast and so on. So these are absolutely standard across all eukaryotes. So the question of minor differences in different human cells almost doesn't even matter. I'm talking about things that are conserved between animals and plants and everything in between. Yeah. Okay, so inside a cell, there are these different locations. I think of them as places, right? You can think of them, to extend the analogy, you can think of them as warehouses where things move, right? So there'll be a source warehouse and a target warehouse. These are called compartments. They're bounded by membranes. You could think of big hollow sacs that are inside a cell. Could be the ER or the Golgi or the endosome, whatever it is, right? So they're called compartments, or they're called organelles. And there are donors and acceptors. I sometimes call them source compartments and target compartments, between which stuff has to move. And the important stuff is not the stuff in the white space, which is in the cytosol of the cell. The cytosol of the cell, everything is shared by everybody. But inside a compartment, it contains its own unique stuff. And that's the important thing about compartments, right? There's something different about its composition that allows it to have a different function. For example, the lysosome has, one of its functions is to degrade stuff, right? Or the ER, one of its functions is to make molecules. So inside the donor compartment, there'll be stuff, but also across the membrane of the donor compartment, there'll be transmembrane cargo, which is this purple thing. Similarly, inside the target compartment, there will be stuff in the gray area, and there'll be these transmembrane proteins. And the goal of the whole system is to take the right stuff from here and deliver it to here. Not all the stuff from here, of course, right? You don't want to take everything from a warehouse. When a truck shows up at a warehouse, it picks up the right thing and moves. So what I have here, so maybe the correct analogy is these vesicles are not packages. In fact, the vesicles are trucks, and these little molecules are the packages. But anyway, let's not belabor that analogy. So what happens is there's these things called coats and adapters, which create a little ball, and they actually attract the right cargo from the source compartment, and they undergo scission and they become their own little package. Now, among other things, some of the cargo that they picked up, okay, were in fact the identity markers. There's a whole bunch of other cargo which is just along for the ride, but some of these are the things that tell it where it needs to go. Most vesicles just diffuse around the cell till they hit a certain acceptor or target compartment. And then they check using a lock and key mechanism. They have a sort of key, a template molecule called a V snare or a vesicle snare, which checks against a molecule on the target compartment called the T snare. And if those two undergo the right complementarity, exact protein pairing, then this thing actually fuses and it delivers this cargo, in which case, this case is the yellow molecule. Okay, so I mean, as you can see, it's a very, very sophisticated process involving lots of molecules, the coat which recruits the cargo, some of the cargo which tests whether it's the right target, some stuff on the target compartment which tests if it's the right package, and then it fuses. So in this whole process, what we'd like to do is model this whole thing, but it doesn't even give you the right impression, because the important thing is stuff inside these compartments and stuff that are transmembrane cannot escape into the cytoplasm and go where they want to, because the energy barrier to cross a lipid bilayer is actually quite immense. So they're stuck inside, or they're stuck on the surface. So if some of this stuff which goes there has to get recycled, it has to come back on a different vesicle. It can't just exit. So for example, this red guy which started here, the V snare, it reaches the target, but how does it get back? Right? This textbook picture doesn't actually give you the solution. In fact, we don't really know the answer, but I'll present a potential answer. Okay, so that's one picture of, from one source to one target, but that doesn't even give you the right image. The right image is you have a whole cell, you have different source and target compartments, and lots of them are connected to each other by these vesicles that keep moving between them. And the vesicles move at a rate of tens per minute. They're always budding from one side and fusing to the other side. Right? And the issue is actually quite complicated, because when you bud from the source compartment, and I've shown that by representing these differential equations here, which represent the composition of chemicals X, okay, on compartment J, right? So J can go from 1 to 10, meaning the ER, the early endosome, late endosome, lysosome, whatever it is. And the chemicals, subscript alpha, can go over thousands of different chemicals in the cell. Every time a vesicle buds from a source compartment, it changes the composition of that compartment because it took away stuff. And every time it fuses to a target compartment, it changes the composition of that compartment because it gave stuff. Now, if this is constantly going on, why, when we look at a cell, does it look more or less constant over time? And the answer is because everything is balanced. Okay, so all the stuff that's coming in from some place is also going away from some place. And so the actual composition of a compartment doesn't change much. Right? And this is something which the cell actually achieves by using various types of feedbacks and so on. You can actually make mathematical models of this and show how that balance is achieved. Yeah, but the main point I'm making is that you have to think of the global system. And it's really these microscopic vesicles moving that eventually lead to compartments having the right compositions. Right? So that's an amazing change of scale. Control at one level leads to outcomes at a much higher level, orders of magnitude bigger in size. Okay, so we've done some work on this, and we've actually modeled how different compartments, using differential equations of the type I showed in the previous slide, how these different compartments arise. And you can actually show that it's possible to encode a certain number of compartments and a little bit about how they couple to each other. But it doesn't really, when at this level, the model is a model in the sense we don't really understand why it's doing what it's doing. If you put in some ingredients, it does something. If you get the ingredients wrong, it doesn't do it. Typical with many models. And so we'd like to have a better explanation. Okay, so to do that, today, I'm going to focus on something which is a very specific aspect of what's happening in these very complicated systems, because I can't imagine addressing the entire cell biology of all those molecules I showed you. I'm going to focus on one type of molecule which is extremely interesting, extremely potent, and in some sense is the linchpin of all the complexity of this system. These are called snare proteins. So let me explain what they are. Snare is actually a very long acronym. You can look it up. Okay, so what is a snare? As I showed you in the cartoon earlier, if you have a lipid vesicle, a lipid bilayer in this vesicular form, and you have two lipid bilayer vesicles, one with what's called a V snare and another one which is called a T snare, and these have the right lock and key complementarity, then they will fuse. And what I failed to mention, and I'll go back and really reiterate this, different vesicle fusions in different parts of the cell, again, use V and T snares, but there are dozens of V snares and dozens of T snares encoded in the same genome and expressed in the same cell. And V snare 1 fuses with T snare 1, V snare 2 fuses with T snare 2, and not with T snare 1, and so on. Right? So there are very specific interactions. Similarly, with these so-called GTPases that give identity, there are different RAB and ARF GTPases that decorate different membranes. So even though they are homologous proteins, so as bioinformaticians, you know about homology, the same protein family, exactly the same domain structure, but with a few changes in residues, those proteins are in different parts of the cell. And that's what makes different parts of the cell behave in different ways, right? So here you have the snare proteins. And the important thing about snare proteins, and that's what this experiment, a rather beautiful experiment shows, a single copy of a snare protein on a vesicle is enough to make it fuse. Okay, so you should think of these as extremely potent fusogens, extremely potent drivers of the direction of this process. You can zoom in on this a little bit more, but of course, once the snare fuses, and you can see that over here, what happens is you have a snare, it turns out to be a triple helix on one side and a single helix on the other. After they fuse, it forms a 4-helix bundle. Details are not important. But once you use up a snare for fusion, the energy that was just the energy of the fact that they zip together and the energy of association of those residues is what drives the force that causes fusion. Spontaneously, these vesicles tend to not fuse with each other because there's an energy barrier to overcome. But now you have a very tightly zipped up complex, right? So you have two options. You can either just throw it away. This is all just like a one-time use snare. Or, of course, cells are not interested in synthesizing huge complex proteins and throwing them away after one use. You'd like to recycle. That's what cells do. So once you have a 4-helix bundle, snare complex, there's an ATP burning machine, which actually comes in. It's called NSF, and it unwinds the snares and splits them apart. Yeah. And this harks back to the original question I had. So a snare that was brought in, once it gets split apart, needs to be taken out of this particular compartment and sent back to the source compartment if you really want to recycle it. And that's the process I'm going to focus on today. Yeah. So again, in the textbooks, you'll see this process very beautifully. The snare is unwound, but nobody tells you what prevents the red and the blue, which remember are very tightly associating proteins, to refuse. The only way to stop them from refusing is to pull out one of these things and send them back. So how does that work? Okay. Yes, question.
Hi, Mukund, this is Kasturi.
Hi.
So this snare fusion of one molecule being enough, we are still assuming that it's 40 nanometer, right? Vesicle in that ballpark, correct?
Sure, for small vesicles.
Yes, and then that's done in vitro or in vivo. I mean,
That
do
experiment
they
was
need to stay close together? Yeah.
Yeah, that experiment is in vitro. It's reconstituted.
Okay. Right. Okay. So then maybe when it's happening in vivo, the density of the cytosol or even the cytoskeletal element simplistically can have a factor where the time they're close together, the fusing partners matters. I mean, does it, or it does not matter because it happens in vitro without them?
No, it's okay. So I'm going to go back here. Okay, so this cartoon gives the impression, this cartoon coupled with that reconstitution experiment gives the impression that you just need this thing and that thing and you're done. But of course, the cell is a three-dimensional object. So for this to even start to take place, the vesicle has to be close to the target. And sources and targets are sometimes close to each other, like the ER and the cis Golgi, but sometimes they're quite far away, like the ER and the plasma membrane. And so the geometry of the cell does matter. Secondly, in fact, the problem is the other way around. So often these vesicles will take a long time diffusing between places before they get to the target. And so you have a factor called a tether, which tends to have these are very, very long proteins, which tend to sort of flop around like seaweed and bring vesicles in and test them for fusion. So the tethers will be sort of promoting the testing for fusion. But the third piece, which I will mention shortly, is that the point I'm making is that, yes, if snares were so fusogenic, everything in the cell would just fuse and you wouldn't actually have this beautifully separated cell that you see today. So there must be something preventing the fusion, which is sort of what I hear from your question, from happening inside the cell, although it's happening so quickly inside a tube. And indeed, that's the ingredient which we're going to add today, something which actually regulates snares and prevents the vesicles from fusing so rapidly and in some sense indiscriminately, because that would not help the cell achieve its goal. So the geometry, there are tethers, and then there's additional factors which are not shown obviously in this figure, which I'm going to then bring up.
Thanks.
Yep. So I do not mean to at all give the impression that these things are so simple that they're run by. In fact, this is why you keep going back to look at the old EMs, because in the textbooks they look very simple, but in practice you have to keep track, as Kasturi said, for all the other stuff that's happening inside the cell. One recent result, which is actually interesting, the textbooks. I'm sorry to keep going on about textbooks, but you may get the wrong impression by reading them. So it's my duty to correct your wrong impression. The textbooks give the impression that vesicles jump onto cytoskeletal elements, move with motors. There's this famous video of a vesicle moving along a track and pulled by a motor and walking and all that. 95% of vesicles don't do that. They just diffuse. Some vesicles do that, and they're quite glamorous, and so we tend to focus on them. Yeah, but a lot of this is quite random. Okay, so now I promised in the title of the talk that I'm going to bring in some mathematical puzzles. So I've sort of dosed you with a huge amount of molecular biology and cell biology. You've sort of, I hope, digested a little bit. You don't have to know all of it. So now I'm going to bring in the mathematics. So now our goal as modelers is to keep the important details and remove not the unimportant details, but the details that are too complex to deal with at the beginning. Right? You incorporate them later. And so what we're going to use is an abstraction, and it's an extremely powerful abstraction. It comes from a field called graph theory. So unfortunately, the word graph has two meanings in math. One is the graph of y versus x, which is not what I'm talking about. The graph I'm talking about is a structure which has nodes and edges. So this thing I've shown you is a graph. And it's not just a graph, it's a directed graph, because the edges have an arrowhead, which gives you a sense of flow. So what you should be thinking about this graph, what does this graph got to do with the cell? Well, this node, for example, is the ER. This node is the cis Golgi. Okay, or maybe I made a bad choice. So this is cis Golgi, medial Golgi, trans Golgi, plasma membrane, early endosome, late endosome, lysosome. Okay, so these are all places inside the cell. The inside of the ER, the inside of the Golgi, these are all things you're used to thinking about because they contain the cargo. One thing you may not be used to thinking about is the inside of the plasma membrane. But when you're sitting inside a cell in the cytosol, looking into the plasma membrane is not very different from looking into the ER. Right? So although the rest of the universe is inside the plasma membrane, right? For the cell, it's still something that's contained as part of the plasma membrane. So that's what this is. So this is something which it wouldn't be drawn in this way, but when you read a paper about the discovery of a new vesicle traffic pathway, they'll draw a cell, a schematic cell, perhaps like I've drawn over here, right? And they'll draw the shapes very nicely and so on. And the point I'm trying to make here is that for the moment, let's ignore the shapes, the sizes, the locations, although they're very important, as we just heard, right? But let's ignore all that and come down to the topology. Who is connecting with whom by what vesicles? So then I can just convert every compartment to a big gray blob and every vesicle to a small white blob. Okay, I haven't shown the vesicle blobs here. So these are compartments, and vesicles move not in 3 dimensions, but they flow from this compartment to that compartment. Okay, so that's the abstraction. And somebody says, this is my guess for what this special cell type, maybe that immune cell that was part of the question. I've studied this immune cell for a long time, and I think these are the compartments, and I think these are the edges after a lot of work. And now I have to say, well, from what we know about molecules, can this thing exist or not? And you may think that's too much of a question to ask, but it turns out it's not. This is where graph theory comes into it. So I've told you already that every vesicle fusion has to have a V snare on the vesicle and a T snare on the target. V is for vesicle, T is for target. Without the snares, they won't fuse. That is true in vivo or in vitro. So we posit that there's a V snare, that's a transmembrane protein traveling along this vesicle, and a corresponding T snare, the one it likes, on the target. V1 loves T1. So a vesicle carrying V1 will fuse to compartment T1. So that's great. So we're just focusing on this pathway, right? So you think that's done, and all I have to do is then make similar V's and T's for all the other pathways. But that's not so easy, because once the vesicle, the snares fuse, remember you have to pull them apart. And then you have to send the V snare back to the origin because, and this is true, it's not destroyed after one use, it is reused. So you say, okay, I'm going to find a path. There's one. There could be others, but I'm going to find a path that takes me back to the source compartment. There's one right there. And you think now you're done, because now the V snare is back where it started, and you need to fuse. Again, so you send it back along this vesicle. But now you're in trouble. And does anybody notice the trouble? The trouble is that if this is what is being carried on this vesicle carries V snare V1. And I told you that a vesicle's properties are determined purely by the cargoes they carry. We've already agreed that a vesicle that carries V1 will fuse to target compartment that carries T1. So why doesn't this vesicle fuse back? Right? And that's the major issue. We actually don't fully understand, right? But there are some patterns, and I'll show you one example of how you can do that. So the immediate thing you should think of is, well, let's inhibit it. So I make up an inhibitor. I just make it up. Yes, question.
So, like from the first compartment to the second compartment, it reached using the T1 because it had T1 for the accepting. Now,
Yes.
Now when you're saying that it's reversing back to the from where it has started, so for the moving from the second compartment to your third compartment, there should be some receptor also now.
Yes, but that's if it reaches there. But the first thing it sees when it comes out of here is its own source compartment. That's the one it's closest to. So in fact, if you do this experiment and you don't put inhibitors, these things constantly refuse. Yeah, so it is a real problem inside cells. That is, see, V1 loves T1. Right? We already established that. In which case, this vesicle carrying V1 has to go back. You're asking how it fuses here, and we'll get to that in a second. So
Yeah,
even,
I also had a similar question. Like the previous node at the top is T1, right?
Yeah.
Now the edge going down, what is this node? You know, if I can.
Good. So you guys are getting into it. So the point is that we now have to, if we're to make a hypothesis of this, we have to decorate the entire graph. I have to put all the T's, I have to put all the V's, I have to put all the recycling pathways, and then I get a solution. Right? And I'm going to work my way. This is what I'm going to work my way up towards through this talk. So hold your horses. We're going to answer those questions.
I also have a question, I'm Ranjit. So when you say steady state, is it steady state in number or flux, or what exactly it would be?
Both. So the fluxes are, the whole thing is steady. So the composition of each compartment is described by a differential equation of this type, which covers the entire graph.
Okay.
So you should really be thinking of a flux coming and a flux going, but the flux, for example, incoming flux of X here could be balanced by outgoing fluxes and incoming fluxes across the entire set. So locally, every compartment has to be balanced. And you, again, complicated, but I will get to the steady state question too, but this is great. So the point is that if you just start with a cartoon like this, then you realize, well, there's a lot of things that you weren't told. And so you ask, what exactly are the details about fluxes, steady states, fusion, specificities, and everything else? And so those demands are what we now need to address with the model. One way of saying it is that These cartoons are not sufficient. If somebody says, this is what I think, you should ask for more details. Okay, but I'm just focusing on a little mystery. There are many other mysteries. Yeah, question.
Like two possibly naive, silly questions. Like, could you have a case where globally it is stable, but the local nodes are like dynamic and changing all
Yes.
the time? And
Yes, you can. You can. And I won't talk about that today. We have done work on that. That's a process called compartmental maturation. It's now you've moved to a rather more complicated scenario, because the right model framework for that is a Markov process where the individual cell actually moves in its state, but the distribution across different cells in a large population of cells remains constant. So that's a different type of steady state, which I'm not going to talk about today. It happens, but it's not universal. The simplest type, which I'm talking about today, is 90% of what you need to understand. Second question. You had two questions.
no, and I was just thinking the converse of that, right? Could it be the case that locally they are stable, like at a steady state, and no changes, but like there is global flux or dynamism?
It is like that because one of the fluxes which I haven't shown, for example, the plasma membrane can be the source. See, of course, I take that back. There are fluxes. Okay, this is not in steady state because nothing is moving. It's in steady state because everything is moving. So this is the typical example of a dynamic steady state that you see in any out of equilibrium system.
Okay.
So it's a steady state, but it's extremely dynamic. Yeah. That's the picture you should have in your head. Everything is moving. Right, but it's the
And
composition
somehow
state.
kind of they balance, like at each
Exactly.
node, it balances itself out.
Exactly.
Okay.
And it's not somehow, in the sense you make dynamical systems models and put in the right ingredients, and they do fall into these patterns. So these attractor states are, fairly common, and you can construct them mathematically. The issue is not their existence, but to really understand their structure. Okay.
And one final question, like I always get attracted to translational questions. Like, could you make a wild hypothesis that when they fall out of steady state, like maybe the homeostasis or normal undiseased state is when they balance each other out and they're at equilibrium, and like disease state somehow corresponds to unbalance and falling out of equilibrium?
Yeah, I mean, this is very new age. It's not like that, because in the natural world, there's no boundary between disease and not diseased. If you look at, for example, a marine ecosystem, 100% of the marine microorganisms will be infected by viruses and other pathogens. And so the standard state is what it is. I mean, so there are, of course, changes. For example, if you go from one environment to another, you may want to change your traffic pathways. There are, of course, cases, and very famously, when certain viruses infect eukaryotic cells, they change the pathways in order to actually shut down host protein synthesis and make a lot of virions that go to the cell surface. So those do happen. But the connection is not with disease and not disease. It is with different environmental circumstances, one of which could be infection.
Okay. Thanks.
But this is a question that we're going to start to work on soon, is how do these pathways get rewired by viruses, which I find quite fascinating, just as a abstract problem. Okay, so back to details. There we go. So you are now the setup. V1 loves T1, but V1 has to go back. But if it does go back, this vesicle will fuse back here. So to prevent that, we're going to pause it. I'm just going to make it up, an inhibitor. And we're going to say that the inhibitor complexes with V1 to make it inert. So then you don't have a problem. Of course, you also have to have another V snare, another T snare here. Those are all happening in the background. Okay. But for the moment, I've got this V and inhibitor coming up to here. So it's great. Now I've delivered the V to the starting point. And now I can use the V again. But do you see the next problem? The inhibitor has to go back to its source node, which is the target node of V. How does the inhibitor go back? It can't go back on the same vesicle as V, because then it would inhibit V and it couldn't fuse. And so that would, you go back to zero, right? So the inhibitor has to go back some other way. Is that possible? Let's see. Well, here's a crazy path. From here, there's, yes, the inhibitor can go back. And so now we have a full system. Just to explain the fusion of this vesicle to this target, you've now invoked this large subcomponent of the whole cell. Right? And now we've got to do this for every vesicle fusion again and again and again. Let's try another one. Here's a vesicle that contains V2, binds to target T2. Cool. Okay, now you send the V2 back. Same problem. Why doesn't it refuse? You make an inhibitor, I2, which comes back. Great. And now the inhibitor has to come back to the source. But now you're in trouble because there is no route from here to here other than this vesicle. This may not have been obvious when you first drew this diagram, but now it suddenly becomes obvious that if this is the diagram that you think is happening inside the cell, and it is this inhibitor hypothesis, it can't explain what's going on here. And you see, we have to work quite hard to discover this gap in this model. Yeah, something went wrong. And I could give you this and you could play. I mean, this is how I first came up with these patterns, right? We played and played and said, could we make it work? Could we make it work? Could we make it work? And sometimes we could and sometimes we couldn't. So does everybody see why this failed? Same game we played. V2 has to go back. It has to be inhibited. The inhibitor has to come here. But if you notice, there are no arrows that go from this guy back to that guy. Because if I make a cut across this plane, you see that 2 arrows come back and only 1 arrow goes forward. So you know there's no way to go from here to here apart from this. Okay. Any questions? So that's neat, right? So we want to now generalize from this little puzzle that we started with. Okay. So for this, I'm going to teach you a bit of graph theory. So bear with me, but it is fairly simple. So a directed graph, right? A directed graph has many features. So if the nodes are labeled as X and Y, it's called strongly connected if you can go from anywhere to anywhere else by a directed path. If it's strongly connected, then there are cycles. There are ways to actually go in a cyclic pathway through the whole system. And then there are these 2 very important features, which I'm going to stress on. Remember what I said here. I cut, if you can see my cursor, I cut across the midplane here. And I noticed that 3 edges of the graph cross my cut, 2 in one direction, 1 in the other. And if you try and keep on cutting this graph, you'll find that every other cut either just doesn't go through the graph or it cuts 3 or more edges. So by cutting these 3 edges, I can disconnect the graph. So this graph has a connectedness of 3, k equals 3. And the more edges the graph has, the more its connectedness tends to be. So this turns out to be an important feature. Yeah. Are there any questions about graph connectedness? So the way you find graph connectedness is you make all possible cuts and you find the cut that has the least number of edges crossing it. In this case, it's 3. Okay. So there's a little theorem, which is from the 1970s, called Menger's theorem. And you'll see why it's related to what we're talking about, right? This graphs, graph cuts, graph connectedness are all fairly abstract. And this is the theorem that connects all this abstract stuff to a real question you may have. And I'm going to read out this theorem. Let G be a directed graph. Let X and Y be 2 distinct nodes of this graph. Then the number of pairwise edge-independent directed paths from X to Y is equal to the minimum number of edges whose removal disconnects every directed path from edge X to Y. Okay, so very complicated statement, but we can unpack it, right? We're going to pick 2 nodes, X and Y. So for example, this node is X, this node is Y. And what we're going to do is we're going to try and see how many edges you have to remove to disconnect directed paths from X to Y. In this case, oops, wrong way around. In this case, you have to remove just 1 edge to disconnect directed paths from X to Y. In this case, you actually have to take out 2 edges. Right? I'll go back to the original case. And if this was X and this was Y, you actually had to take out 2 edges to remove all directed paths from X to Y. Now, here's the thing about Menger's theorem and why it's useful. I know this graph is strongly connected. Here is node X, here is node Y. without even knowing what the paths are to get from here to here, I know that there are two paths that share no edges at all. If I know that, I don't have to know what the paths are. If I know that, then I know I can send I on one of those paths and V on the other, and my job is done. I don't even have to find those paths. Right? And that's the real importance of these theorems. From something simple, you can infer something very complicated. So I don't have to play this very complicated game. Okay. There's another theorem which I'll read out. This is actually something we proved. It's not in the literature as far as I know. I'll read it out. It'll come back later. Okay. Each edge of a strongly connected graph belongs to a distinct set of cycles if and only the graph does not have a two-cut. Keep it in mind. I'll come back to it. Okay. So I've told you, I've sort of drowned you in cell biology, molecular biology. Now I've given you sort of a textbook's worth of graph theory, Menger's theorem. And now we're going to bring it back together. So here, again, I have my, my graph, which represents a cell, but let's look at something rather simple. Two compartment graph. So here are two compartments. Here are the vesicle edges. Okay. But with one important caveat, the edges represent fluxes. Question.
Would it be okay if I interrupt right now, or would you rather I wait?
No, please interrupt. We have plenty of time.
So if you go back 2 slides, when we were considering the other one, which had, yeah. So when we were considering this example where X is is V2 and T2 is Y, don't you think this graph is incomplete? I mean, when you said we have to only cut one edge in this case, because we haven't taken care of the inhibitor here.
No. We indeed have. So this graph is incomplete in the sense, this is the graph you gave me. And I'm saying it doesn't work. So in that sense, it is incomplete, that the experiments have not yet figured out how the inhibitor goes back. For the whole thing, there has to be another edge somewhere.
Yeah, so are we right in saying that we have to cut only one edge at this point? Because then there would be 2 edges that we would be cutting.
No. So the dotted edge doesn't exist. There is no dotted edge.
But when we were, I mean, I don't get why then if we take V1 and T1 as X and Y, we're cutting 2 edges, yeah?
And that's why, so you see what happens here. There's a cycle that V follows. I'm going to draw it out with this cursor.
Mm-hmm.
There's a rather complicated cycle that I follows.
Right.
Here it is. There, and there.
Yeah.
Yeah. And the reason that V and I, except the one place where you wanted it, can go on different cycles is because of these 2 edges here, both going from left to right, turns out. If you wanted to play the same game with V2 and I2, there's only one edge going this way. So there's no way. At one level, obviously, there's no way to send it on 2 different paths from this to here. Now, what Menger's theorem says is the other direction. If there are 2 edges, you can always find a cycle. That turns out to be the hard part of proving that theorem. The easy part is obvious. If there's only one way to go from this side to that side, because if I just draw a line across here, there's only one trip that's going from right to left. It's on this bus.
Mm-hmm.
So if you want to send any molecules from right to left, you better be going on this bus.
Right.
And yet we know that I2 and V2 cannot go on the same.
Mm-hmm.
And so that's the contradiction.
Right. Cool.
Yeah.
Thank you.
Okay. Okay. So here we have 2 kinds of graphs. I'm just showing the fluxes. These fluxes are on edges E1, E2, E3, E4, and 2 compartments X1, X2. Just keep in mind, though, that the same vesicle, it could be, for example, that this compartment makes a vesicle. That compartment makes a vesicle of the same composition, and this vesicle fuses back to both compartments. It's a sort of most degenerate case you can imagine. The fluxes from this, there are actually 4 fluxes. 1 to 1, 1 to 2, 2 to 2, and 2 to 1, even though there's only one vesicle type. It's just a detail you may have to keep in mind. But we're going to focus on these transport graphs, okay? So this now, you need the tiniest amount of linear algebra to follow what I'm saying. And even if you don't remember it, I'll just remind you how all this works. So it comes back to this issue that Ranjit asked about fluxes. So now I'm going to have a hypothetical cell that has 4 compartments, X1, X2, X3, and X4. And my hypothetical cell has 6 edges, E1, E2, E3, E4, E5, E6, just so. And there are no other vesicle edges and there are no other compartments. And now I take some molecule A. And I say that the amount of A on each of these compartments is called capital A1, capital A2, capital A3, capital A4. And the amount of that A on each of the vesicle edges is little A1, A2, A3, A4, A5, A6 per vesicle. And of course, the vesicles move, right? So there's a rate, let's say 10 vesicles per minute crossing each edge. So the total amount of molecule A that crosses on edge 1 is that rate of vesicle 1 times the amount of molecule A it contains. And similarly for all the other rates. So you can write down the steady state equation. This is sort of the first day of linear algebra. You'll write down something like this. And so let me write it down explicitly. ddt of A1, first equation, is minus R1 times A1 minus R5 times A5 plus R4 times A4. That's the two outward edges, one inward edge. You see this? Minus on edge 1, plus on edge 4, minus on edge 5. That's the first row. And so you multiply this row by this column to write the first differential equation. Second differential equation, similarly, I'll just go through this explicitly. X2, there are 2 incoming and 1 outgoing. 1 and 5 are incoming, 2 is outgoing. Is that correct? Yes, 1 and 5 are incoming, 2 is outgoing. That's the equation. So you write all this down. Right, this is the connectivity matrix of the graph. And so I'm just conveniently writing this as a matrix, but this is just 4 differential equations. And this set of equations sets up a condition. It says that if you want me to be in steady state, so the left side has to be equal to 0, I'm assuming, then this matrix multiplication has to go to 0. Another way to say this, if you remember linear algebra, is that this 6-dimensional vector has to be in the null space of this matrix, the kernel of the matrix. Matrix has to project it to 0. And now you can show that if the matrix represents a strongly connected graph, the only way to be in the null space of the matrix, a little trick for those of you who remember your linear algebra, you can solve this problem. And there's something that you can use, it's called the rank nullity formula to figure out the dimensionality of the null space, okay? And it's all cool stuff. The only fly in the ointment here is that all these numbers are positive. So this is a slightly more difficult than standard linear algebra where things can be positive or negative. Here, all my rates and concentrations and quantities have to be positive. So if I put that condition, the answer turns out to be very simple and intuitive, and you didn't have to use all this math to believe me. The only way that a single molecule can be in steady state throughout the system is if that molecule moves in cycles, right? So for example, it can move on this blue cycle, or on this green cycle, or on this red cycle, and there are no other cycles, turns out, in this system. There are only 3 cycles. And there are ways to compute how many cycles there are. So just to be clear, this molecule will have a flux which is represented by the sum of all the 3 cycles. So if it was equal amount on the red, the green, and the blue cycle, then 3 units of it would go across edge 3, because all 3 cycles cross here, but only 1 unit would cross across edge 1. 2 units would cross across edge 2, and 2 across here, and 1 each, okay? So you can see the point. For a graph like this to be in steady state, all molecules have to go in cycles. Any questions about this? It's a very simple statement, okay? Turns out to be slightly niggly to prove, but it's a very simple statement. Any questions? So A1
corollary is that if there are no cycles, we can conclude that that molecule is not in steady state, right? Either it's like being depleted or enriched, right?
Sure. So the more general statement is that you either travel in cycles or you have source sink balance terms that are one-to-one connected or connected in a directed way. That's the most general steady state condition. But in this case, we're looking at snare proteins, which tend to be created at very low rates and hardly degraded at all. They go around 100 times before they get degraded. So it's a very good approximation. And the same statement is true for every molecule in this cell. So every molecule in the cell can only move on linear combinations of cycles. Okay, and there are thousands of molecules. You may worry whether cycles exist. So it turns out if your graph is strongly connected, there are cycles. There is a cycle basis for the graph. It's another little theorem. Okay, so every molecule can be associated with some linear combination of cycles. So let me pick 3 molecules which are as distinct as possible. Let me take one molecule which is blue in color and only run it on this cycle. And one molecule which is red in color, only run it on this cycle. And one molecule which is green in color, only run it on that cycle. I'm allowed to do that. I have as many molecules as I want. Okay. And that's the picture I've drawn over here. So I am using a 4-dimensional vector to represent the composition of each vesicle. The first dimension actually represents how much membrane there is on a vesicle. Okay. So a vesicle needs to be bigger to carry more stuff. So forget that one. The next 3 represent how much of molecule 1, molecule 2, and molecule 3 is carrying. This edge carries all 3 molecules. That edge carries only 1, molecule 1. This edge carries molecules 1 and 2. This one carries, 1 and 3, 1 and 2, just 3, and just 2. So now I've put on top of what was just a graph, okay, I've put now vectors of very special type. And these vectors are in the null space of the matrix. So you see the beauty of this process, right? You take graph theory and suddenly you have the enormous power of linear algebra to save your skin. Okay, very good. If I divide the vesicle by the membrane size, in some sense to normalize it, I then get the graph on the right, which is actually the vesicular composition of this graph. You would not have guessed this up front, okay? That this is the steady state of the system and so on. It's sort of fairly complicated. Why is this half and half, 1/3, 1/3, 0 and 1? It's sort of very, very weird, right? It only becomes natural and obvious if I showed you the cycles. Otherwise, it's not. Okay. Now I'm going to bring it back to this. This is all graph theory. I'm bringing it back to the cell biology. I'm bringing it back to the statement I made right at the beginning, which is absolutely true and verified inside a cell. The properties of a vesicle are determined by its composition. Okay. By assumption, every vesicle here has different properties. This vesicle fuses to this compartment. This one fuses to this compartment. This one comes from here and fuses here. They all have different properties. If they all have different properties, they must have different compositions. That's the necessary condition for this whole thing to work out. And if you gaze at this graph, you'll realize every edge has a different composition in this basis. No 2 vesicles have the same binary code. You have 1, 0, 0, 0, 0, 1, 0, 1, 0, 1, 0, 1, 1, 1, 0, and 1, 1, 1. The 6 edges have 6 different binary representations. Okay, and that's neat, because if that's true, I can make this whole thing work out. I just say that the molecules are clever enough to know exactly what the full combinatorial composition of the vesicle is. And therefore, they can behave differently depending on the composition. The only issue that would have arisen is if 2 vesicles here ended up having the same composition but behave differently. Then we would say we reject this hypothesis. So I've shown you one example of how to do this. And now the obvious question is, what are the necessary and sufficient conditions to do? What are all the examples that make this kind of thing work out? Okay, so for that, before I get into the more complicated snare business, I'm going to tell you what the answer is. The necessary and sufficient condition for every vesicle on this graph to have a different composition is the graph has no 2 cuts. A 2 cut, remember, is an edge that you cross where always one vesicle is going one way and one is going the other way, and there are no more edges, because you can remove those 2 edges and you disconnect the graph. If the graph had a 2 cut, for example, if I remove this edge, oh no, that's not the 2 cut. Let me see how to make a 2 cut here. Okay, that's true. If I remove this edge here, no, the graph still has no 2 cuts. Okay, I think what I have to do is I have to remove this edge. Let me remove this edge, which is the 001 edge. Then you notice that if I make a cut like this, there are only 2 edges crossing it, and that will decouple compartment X4 from the rest of the system. Is that right? Yeah. Now that system, just by the steady state conditions, if you have vesicles coming in here and vesicles going there, obviously the composition of this must be the same as the composition of that to make this compartment in steady state. If they are the same, then why is this vesicle behaving from that? So obviously one direction of that theorem is very simple. If it has a 2 cut, obviously you can't make it work out. That's the same failure we had in the original graph, right? You just can't make every system satisfy these cycles. The real difficult part of the theorem, which is the other direction as usual, is if it has no two cuts, I can always make it work out. It doesn't show you how, but it says a solution exists. And then you have to go and make the solution. Yeah. Incidentally, this exact type of result is what you can use to generally solve this wolf, goat, and cabbage problem. You can state it in exactly the same way and find very, very similar conditions for the solution. Okay. Now that system which I just talked about is not still close enough to the cell biology. In the system I spoke about here, sure, every vesicle has a distinct composition, but for this to work out, the molecules have to be very clever. They have to be aware of the entire combinatorial composition of the vesicle in order to modulate their own properties. And that maybe is too much to ask for, right? Molecules typically have one-to-one partners. Snares, for example, have that feature. So if you have snares, what's the result, right? And here's what I'm going to show you. And this relates to the big puzzle that I had, possible or impossible, and we went over that puzzle quite a lot. It comes down to a very simple test that you need to do. Okay, so for example, if I start from the right graph in this case, the right node, I'm sorry, and go to the left node on this vesicle, and I assume that there's a V snare that does it, and a V snare with an inhibitor that must travel back somehow through a very long path, and that's why I've drawn it as a squiggly edge. And the inhibitor is coming back to the original point through a very long path, which I've drawn as another squiggly edge. I don't care about those. They exist throughout the whole cell, right? The important thing is to make this work out, it implies those two long paths exist. Okay. So how do I test this? It turns out, just like in the previous slide, but I'm not going to prove it over here. To test it, the graph actually has to be, to make it work out, the graph has to be not 3 connected, but actually 4 connected. Okay. And the reasons are, a little bit detailed and they don't lead to much insight, but let me just finish the story. And I'm going to leave some time for questions. Finish the story with this little picture and show you how it can work for the graph that we had in mind. Okay. So, for example, if this is the source node and this is the target node of the blue vesicle, I need the inhibitor and the vesicle and the V snare to come back on this path. And I need just the inhibitor to come on this path. Okay, that's fine. But you have to make that work for every single path, for every V and inhibitor combination, V1 and V2, V1 and T1 and I1, V2 and T2 and I2, and so on for every edge. I found a way to do it for this edge, but I can't do it for this edge. You can just look at it and convince yourself, and I can't do it for this edge. But if I add 2 more edges to make the graph 4 connected, not 3 connected, then I can make this whole thing work. So there's a general theme here. If the molecules are very clever, you can get away with fewer edges. If the molecules are not so clever, you need to make more edges to make sure that there are no clashes. It roughly corresponds to longer solutions in the goat, wolf, and cabbage problem. Okay, so here are all the full set of results. Okay. If my only requirement, the left side I've written in essay type form, right, what my requirements are. And on the right side, it says what kind of graph can satisfy. If the only requirements are that the cell is in steady state, which is a good one, molecules are not created, destroyed, or modified, which for snares is true, and all membrane integral molecules move on vesicles, which is 100% true, then the only requirement is that it's a strongly connected graph that has a path from everywhere to everywhere. And one way to think about that is you have to have a path from everywhere to make sure that membrane doesn't accumulate in one part of the cell. But this works out nicely. You don't need, you can have 2 cuts and so on. It's not a problem. If on top of that, you add this property that 2 vesicles with the same membrane integral composition must fuse to the same targets, which is true, right? Then strongly connected is not good enough. You also have to have no 2 cuts. So you have to have another edge to make this work. If on top of that, you say there are snares, and snares have inhibitors or activators, then it turns out that the famous graph we were puzzling around with is not enough because that has a 3 cut. You have to have at least another edge here to make a 4 cut to make it work out. And if you go, take a screenshot of what you're seeing here, go home, take this little graph and try and work out with a different V snare, T snare, inhibitor combination for every edge and work out if you can find paths, I guarantee you will find a solution. I don't know the solution, but it exists because I know this graph has 4 strong connectors. Okay, so that's my final slide. So I hope the few things I wanted to convey are, first of all, cells are deeply interesting and mysterious, and we really don't understand how they work at a global level. We understand a lot about the molecular level. The transport system has some puzzles that are still unresolved about how molecules get recycled, in particular snares. We still don't know the answer. If you abstract this out with some very reasonable assumptions, look at this. I haven't made any assumptions about, it's some very general stuff we can all agree on. Then it turns out that it places strong conditions on the graphs that people discover using, say, imaging. And you can take these assumptions, go back to the imaging and say, you're missing an edge. Please go back and look. And hopefully people can go back and look and they can discover new edges, and you can actually tell them where to look, right? And in this particular case, it's very rare, but in this particular case, we not only have a model which gives answers, but we actually understand why, because the model behaves exactly as the theorems predict, and the theorems give necessary and sufficient conditions to make everything work out. So once I've just written down the theorems, I don't have to do this back and forth thing where you give me a graph, I give you an answer. You give me another graph, I give you an answer, and so on. I've given you the full answer for at least these 3 assumptions. Okay. Now, of course, what do we do next? There are assumptions about coat proteins and how they load cargo. There are assumptions about self edges, which we've ignored over here. Okay. There are assumptions about the level of specificity if it's not 100%. Okay. And then, of course, there's the other broader questions we had about if things are not in steady state, right? So I can scratch out each of these assumptions and make things more and more general. And you can do that. And we have done that. But at least here, there's a nice, satisfying sort of closed solution to the problem, which I found very, a lot of fun. So this is published in Journal of Biosciences. You can go read about it and email me here if you have any questions. And please come visit. We have a lot of fun here at the Simon Center, which is a bunch of physicists and engineers, mathematicians, computer scientists looking at problems like this. This is a little logo for the Simon Center we commissioned a few years ago. I don't know how many of you are old enough, but on the left, you have a Logo turtle. Logo is a programming language from long ago, 80s. You had a turtle on the screen. You were allowed to tell it forward 30, right 10 degrees, and so on and so forth. And from that, you build a whole picture. So from such microscopic interactions, you can build up trajectories. You can work at the scale which I talked about of cells and vesicles, entire cells and tissues, and indeed organisms. And you can build up on the right a real turtle, or at least a picture of a turtle. And really the goal is to span all these scales, at least be aware of them in any biology problem you deal with, because that's the fun part of biology, right? You got to couple evolution to molecules and everything in between. I'll stop. I'll continue sharing in case there are any questions. I can go back to that slide. Happy to take questions. And among other questions, we can also talk about broader issues of what distinguishes models from theories and how to choose problems to work on and so on.
Okay.
Yeah, there's a question. Yes, Kasi.
Hi, Mukund, if you would discuss a little bit about graphs that are static and not really directed. For example, arborization of the blood vessels or, webs you see in biology, right? Although information travels through them, but then their structure is kind of static. And then when they're made, possibly, there was some directionality to it. So can we deal those structures in the same manner, or would Menger's theorem apply for those, or there is another substitute for that?
No. So Menger's theorem doesn't. So strangely, directed graphs are understudied in even in engineering. In fact, this monograph, Bang, Jensen, and Gutin, you can download it for free, and it has every result that you want to know about directed graphs. Quite recent, as you see. There are no full textbooks on directed graphs. So the whole a huge amount of work has been done for undirected graphs of the type you bring up. And now there's an interesting question, right? I give you an undirected graph where between node A and B, you have no sense of direction. But globally, there may be some sense of asymmetry. And you might be able to identify such a thing. Now, there are techniques for this, and they're not as clean as these theorems, because if you can't rule out a reverse flow, let's say, of some blood vessel during at least some phase of the pulse or something, you have to have a more sophisticated view of what the edge means. The other graphs that people tend to forget are undirected, are metabolic networks, where, yes, enzymes tend to move convert substrate to product, but enzymes are actually reversible, and you can convert products to substrates. And so under conditions where the product is in excess, or under thermodynamic conditions where certain arrows don't work in the right direction, the thing can flow in completely different directions. So chemical reaction networks and the theory of that, for example, flux balance analysis, what they do is they convert the undirected graph into a type of fake directed graph where every edge has two directions. You replace an undirected edge with two edges and you treat them separately. You can't always pull that off, but if you can pull that off, then you can use the theorems that I just mentioned to check if things work out or not. But it's subtle, and it should be left to the experts.
Thanks.
So I had one question. I was just wondering, you mentioned that you studied all these results in the context of snare proteins, right?
Yeah.
I was wondering, are there other classes of proteins which might obey these, and like that you can apply these?
Yeah. So first of all, here's the way you should think about it. So just looking at the constraints for snares, here's the number of edges we need. Okay. Now you can ask whether other things obey the same constraints. If they do, you can get away with the same edges. Now you have to ask whether other things obey different constraints. Okay. Suppose I only have two coat proteins, then I should ensure that the coat proteins that are being reused in different parts of the cell don't clash with each other. But that's a whole other set of constraints which we have studied, which I'm not bringing up over here, but which will introduce even more edges and compositions and so on. So something that obeys the same as the snare proteins, we've solved for all of them. Snares, lipids, basic things that travel in cycles. The thing we're studying now are the coats, but I think the real constraint is something I haven't mentioned, which is interesting. It's the question of function. What are these graphs good for? If they're not good for anything, so let me go back to at the beginning. Okay, I have a picture of a graph that is. Look at this graph here. That's actually a directed graph where I have other kinds of edges. Okay, here I have this graph is not in a static steady state, but it's actually in a dynamic steady state where compartments themselves flow from creation to destruction. So these graphs also exist. And this part of this graph is actually the Golgi apparatus. And it turns out that this particular graph topology of the Golgi apparatus is very useful in order for the cell to synthesize complex molecules to show other cells on the cell surface. It's a very tight connection between the topology of the Golgi and its ability to synthesize complex chemicals. So just making these graphs is not sufficient. It has to be useful for a cell in order to be selected. And I think that part, people haven't even done experiments to check why certain vesicle pathways are useful for a cell. Obviously, this is a technically challenging experiment, but I think it's a huge field of new work that can be done. Actual benchtop evolutionary cell biology, where you put selective pressure and track individual fluxes and see what good it's doing. And I'm going to bring that back home to one aspect of what we've done today. Okay, so why is the graph like that? Well, because the cell needs, so for example, why is this graph like this? Well, it turns out the cell needs this cycle to do something cool. It needs it. Okay, that's what the graph is for. But what's the rest of the graph? Well, maybe the rest of the graph is there just to make sure the V's and the T snares and all that behave the right way to make this work. Right, so there's the underpinning substrate and then there's the functional end. I've only talked about the substrate part. right? So to separate the basic machinery from the functional machinery and so on, these are things which people have just started to ask. And it's difficult. Cell biology is difficult for exactly this reason. If I break an edge here, I knock out a molecule, I break an edge here, the consequences happen throughout the cell. Things start breaking as the fluxes go out of balance. And this is the reason why genetics of vesicle traffic has been so difficult to do. Okay, so I'm not saying that it's an easy thing, but it is really a thing that we need to start working on as an experimental subject. There's a question in chat. If one makes these graphs undirected and puts some probability for forward and backward paths, the same solution. Yeah, so very nice question. So as I briefly mentioned, there is another representation of the same thing, which are Markov chains. These nodes become states, the edges become transitions defined by some probability, which are columns of a matrix. Then you get some Markov chain transition, and you get a steady state. And those steady states always exist. So in some sense, if you introduce probabilities, you can always find solutions. The issue with probabilities is
how do you determine those
how do you determine the solutions? It's just the
those probabilities.
Oh, how do you determine
experiment?
Nobody's done it. Okay. And my impression is that actually it's a bad approximation, which I was just going to get at, because what you want when you get inside the cell is, again, think about it as a user of a bus system, right? You get on a bus, you want to make sure it gets to where you want to go. Okay. You're not going to get on a bus with a 50% probability of getting where you want to go. Okay. Because then you're stuck and you don't know how to get back. So inside a cell, I think the probabilities are dominated by certain fluxes, which you should think of as approximations. You should think of a pure directed graph as an approximation to a probabilistic graph with very high probability of certain fluxes. The rest will then constitute errors. And then you can ask, is this steady state robust to errors? And the answer is yes, because you can make a dynamical system with some low probability of errors, and up to some probability of errors, it will still do what you want it to do. And of course, beyond some threshold, it will fall apart catastrophically. And you can make such theories. The problem is that nobody's measured the error rates for individual pathways. It's difficult, but it's worth doing. Except for one thing, which is synaptic vesicles, which pretty much don't do anything wrong. So it's not a useful case. More questions? I'm loving the questions. Really, you guys are saying this is all too simple. I want something more complicated. So, sure. Any questions about the broad issue of what distinguishes theories from models and so on? Or we can ask other questions about cells, and let me go to a page which may have enough stuff to talk about.
Participants, don't be shy. Feel free to ask questions.
Yes, you said you can discuss about like how to choose a problem. I'm interested in.
Oh, I didn't promise that. I said how to make a model. How to choose a problem is the question if you want to go down a research career. And you learn how to do it by working under somebody as a guide. You see how they choose problems, and then you stake out your own directions while you still have some buffer inside your PhD group, and then you take a risk and you choose a problem based on some combination of your interests and your skills. Feasibility, this is a question which I think may take more than 10 minutes to answer. So what I promised was how to make a model and distinguish that from theory. How to choose a problem, you develop a taste for it over time. But please be adventurous. And the only way to try and choose problems is to try and push yourself beyond your limit, fail a little bit, fall back into your comfort zone, and then keep trying this. Yeah, sorry for such a generic answer. It's the hardest thing.
Mukund, it may help the trainees if you could discuss how to toggle between experiment and theory. Like,
that's a great experiment.
Yeah,
so
Thank you. Help the trainees, I guess.
Yeah. So actually, when I started in physics as a pure theoretical physicist. And during my PhD, I started doing experiments. I actually cloned and did some genomic insertions of genes and measured stochastic gene expression in bacteria, did some imaging. So I had, I carried out some experiment through the full cycle up to publication. And when I started up at NCBS 20 years ago, I had an experimental lab, and we were doing quantitative experiments with bacteria. So I think it's absolutely a must for everybody interested in biology to have literally done experiments with their own hands at some point. And by that, I don't mean an assay, but a proper experiment where you design it, you carry it out, it fails, you go back, you do the full cycle. Only after you do that can you have the, if you don't do that, and then you go talk to your experimental colleagues and you're just a theorist, you'll just be saying, why don't you just give me this data? And you don't realize that they would also love to have that data. And it's technically challenging, and they would love to sit with you and try and design an experiment to get that data. But until you come up with a way to circumvent various technical hurdles, it's not possible. So first, do experiments at least sometimes. Secondly, when you work, you never give up that link with experiments. Every model you make and every theory you try and see, how can it be tested? How can it be tested? How can it be tested? Go and ask people. What assumptions are too simplistic? What am I doing wrong? Go and ask people. By people, I mean experts. By experts, I mean experimentalists. Because they know intuitively a lot more about how their system behaves than you do. And they may have seen some pattern which they filed away in their minds. And they see your model and suddenly they say, this looks like that. And then you can go back and, so that back and forth has to be there. So you shouldn't end up in a department where, it's a cocoon of just modelers and theorists. Yeah. And I'm speaking for cell biology. Of course, bioinformatics is a different field, very mature, where one can deal with purely computational data sets and do purely computational work because the data sets are so well structured, genomic data, things like that. So that's possible. But if you do that, then you shut yourself off from potential new problems, which may come up by somebody introducing a new model organism and a strange new way to measure things. And so that connection always has to be there. So for me, I think what's ideal about a place like NCBS is where there's a few of us theory people or computational people, but the rest are really people who keep us on our toes. We see new talks, we see new experimental modalities, we really see data. Things don't make sense all the time, and that's the process. So that's the connection. And if you want to start a collaboration, you start by respecting the cell or whatever the actual system, and then you start by respecting the techniques and the people who do the measurements, and you slowly try and work your way toward articulating a question. You don't go in and say, I have the answer. You go in and say, what's mysterious, and you work backwards from there. My 2 cents. This has been my experience. Your mileage may differ, but I think it's roughly correct.
Professor Mukund, there's another question in the chat.
Sorry, let me open that.
I'll just read it out. Kaushik, if you want to ask, you can unmute and ask yourself also.
Oh, no, I got it. Can V-snare proteins work as T-snares for another vesicle? That is the case. How is the recycling modeled in the node edge form? Beautiful question. So it turns out this distinction between T and V snares is not a molecular distinction. The same snare can behave as one on one path and behave as the other on the other path. Then the question becomes, and how does the T snare get to where it needs to go? Which we didn't even address today, but we have some models for that. And that ends up being a very difficult constraint to satisfy. And fewer than 1 out of 1,000 graphs that you might naively write down can jointly satisfy this V and T snare constraint. So the graphs that are true cells are a very special set of graphs out of all the cells. I forgot to mention, how did the T snare get to where it's going, right? Because all snares are starting out in the ER. That's where they're made. How do they get to where they need to go? So that's a lovely question. So that's why in the field, people tend to not talk about V and T snares anymore. They talk about the snare quadruplet, the 4-helix bundle, and there are 3 that are called Q snares and one that's called an R snare because of the amino acids at the midplane. It's very understudied about how T snares get to where they want to go. Thank you for that question. Actually, I should have highlighted. There's another. Let me just stop sharing here. There is another paper which you may want to look at, which I have 3 minutes, so I'm just going to flash here this paper for you.
Feel free to go a couple of minutes above limits, sir.
Oh, is that okay? Yeah, fine. Share my screen again. How do I do this? I'm sorry for not including the citation. It's a recent paper. So can you see this paper? Yeah.
Yes, sir.
Okay, molecular and cellular cancer. So basically all the cell biological evolutionary themes which I touched upon today, this is not a mathematical paper. You'll find the recent references from the last couple of years. I do talk about this idea of eukaryotic evolution. I talk about these two things, how you maintain homeostasis, how you do long distance recycling. I talk about another piece which I didn't bring up today, which is that there's pervasive gene duplication in these systems. I alluded to it because of all these homologies, right? So how do duplicate genes actually arise and find new function inside the system? And then a few other cell biological tidbits. So it's an up-to-date reference sheet. A lot of nice papers here, well worth reading. So that's the citation if you want to just snapshot it and go back and read it later. Apologies for not including this. I should have updated my talk with this. Okay, so that's it. Actually, I have to go because I have another meeting. So, but I really love the questions. I think you guys are well on your way to becoming excellent computational cell biologists. So if you ever want to work on any of this stuff, please let me know. I'm happy to get involved. And again, thanks for the opportunity.
Thank you, Mukund.
Thanks, Alok. Thanks for being here.
Yeah, very wonderful talk.
Thank you.
Thank you, Professor Mukund, for an excellent talk and a very intricate look at vesicles and trafficking and trying to understand the math and physics behind them. So thank you once again and look forward to the next edition.
Yeah, hopefully in person. I'd love to visit. Okay, bye.
Thank you. So participants, the next talk is by Professor Amitabha Nandi of IIT Bombay at 11:45. We can take a quick 10-12 minute break and see you at around 11:40, 11:45. Thank you.
planning to talk about, think of this following situation, that suppose you have done an experiment, and the kind of experiment I'm talking about is specifically like a microscopy experiment, where you can track some observable, like a protein or a vesicle. You see when you put it under the microscope. So basically you are putting a biological system, which can be a cell or a tissue, and you using a microscopy technique, you can track some vesicles or organelles or proteins inside the cell, or you can track several cells, their location, their spatial position, and how they move with time. So basically, I'm talking about some kind of a spatiotemporal data, and you can basically get this kind of a time resolved information through the fluorescence. And now the question is, given this experimental data, how one would quantify this data so that you can get some understanding of the underlying physical processes which drives this kind of movement. And so that is number one. And number two is, can you use some of this quantified information from the experimental data to build up a theoretical model? And such that when you have this model based on this quantified, like experimental information, you can try to regenerate some of this, like what you have seen in experiments, and then make a comparison. And finally, so this is number two, like building up a theoretical model. And number three is like whether using this combination of experimental data analysis and theoretical model, can you have some understanding of the biological function? And here by biological function, I would say that it is, it is a little bit exaggerated in the sense that what I mean by biological function is like function at the level of like whatever movements you see, like what are the outcomes at the level of individual cells or tissue, not like at like a much more higher complexity. So I hope like I would be able to explain this idea. So I will go rather slow because this is a workshop, so it is okay. And I will try to, discuss this issue or this problem through two representative examples, but I will mainly focus on the first example, and if time permits, I will go to the second example. So please stop me if you have any questions. It's okay if I cannot finish it. So, and we can take questions in between. Okay. So I wanted to give you like an overall motivation of what we are interested as physicists. So I usually show this slide or this movie all the time. So this is a movie from experiments, and what you see here is the wing of a fruit fly at the early developmental stage at 16 hours after the puparium formation. And what you are seeing is this, how this wing shape evolves over time, over a very long time, over a span of 16 hours. And what you see here are these polygon-like structures, which are the individual cells, because some proteins at the boundary of each cell has been fluorescently labeled. And during this span of 16 hours, this piece of tissue, this is a epithelial tissue, this tissue is dynamically remodeled due to various things that are happening at the level of this at this cell, at this tissue. So there are cellular rearrangements. The cells are constantly exchanging neighbor. There are cell deaths. There are cell divisions, and the tissue itself, the wing, is under tension due to some boundary conditions. There are forces at the boundary. And all these together really leads to a dynamical remodeling of this tissue over this very long span of time. And what you see is a nice pattern emerging. You can see the veins formed and the shape is changing, and you see collective cellular flows from the right to the left. And everybody can see the movie, right? So it is, the movie is looping. Okay, so so the question is, if we could have zoomed inside a single cell, we will see the dynamics of a lot of proteins and organelles and things like that, which are essential for the functioning of a single cell. However, if we look at many such cells, it is because of the interaction between these cells, we see that there is like emergence of new physics. We see this emergence of shapes. And this is something which, as physicists, we are interested, like how a collection of entities, when you put interaction between them, how they lead to emergence of new physics. So the question is how one can understand biology as a collective phenomena, and what in our group we are interested is to study some of these properties using theoretical methods from statistical mechanics and nonlinear dynamics. So this was like kind of an overview. So let me come to the problem, or the problem which I wanted to discuss. And it might be a good idea to give like some kind of an overall plan. So if you think of that, as I started with, that you have, as an experimentalist, you have some experimental data. Now the question is how you can quantify this experimental data and use this quantified information to build up a model, but which can explain what you see in the experiment, but you don't want to stop there. In a sense that you also want to now use this model because you can to test some hypothesis. You can tweak the parameters, change something, and see if it leads to some new predictions. And the idea is if some of those predictions can be tested back in the experiments. So this is one part. The second part is whatever model you can build up based on this quantification of experimental data, you want to ask the question that if you can actually identify this model as a subset of a more general theoretical framework, which can be identified and applied to a large class of similar systems. So this is the main idea, and we will try to study this through these two representative examples. The first one is kinetochore capture in fission yeast, which is an essential part of cell division. And the second one is looking at axonal transport in CLE. Okay. So before I come describe this problem, let me go a step forward and try to see what happens during mitotic cell division. So what you see is again an experimental movie. So this green structure here are made up of microtubules, and this is called the spindle. And these magenta colored objects actually are the chromosomes. So one important function of a living cell is to distribute genetic material or chromosomes identically to the two daughter cells, and the mother cell does this by forming this nice spindle structure, which is made up of microtubules, cross-linking proteins, and other motor proteins. And then these chromosomes are aligned along the equatorial plane of this spindle. And once this has been aligned, this spindle actually pulls apart, stretches, as you can see, and divides these chromosomes into the sister chromatids, and then the cell divides, and this material is identically distributed in the two daughter cells. And this whole study of spindle morphogenesis, it's a very exciting field. Now, what we are going to discuss today is not how this mechanism of spindle morphogenesis takes place, but a step backward, namely the following, that once this spindle is formed, if we look at a cartoon of the spindle, what we have are these microtubules, which has formed this nice bridge-like structure. And these two objects are the spindle pole bodies from which these microtubules nucleate. And these are the chromosomes. And these chromosomes has these small dots on their body. These are protein complexes, which are called kinetochores. Now, one essential part that this cell divides is that it forms this spindle in like right time, in time comparable or less than the cell division time, so that it can divide in a realistic time. Now the question is, before the spindle forms, there is another important step, and which is the following. So I have explained this different components. So before this spindle, this structure is formed, one important part is this microtubules which grows out of the spindle pole body. They capture these chromosomes which are floating in the bulk. And the question is how these microtubules capture this kinetochore. And this is, and this is a very important question in cell biology. So depending on the kind of the cell, the mechanism by which this capture of chromosome to the microtubule happens can vary, but there are some common methods which are predominant in many cells. And one of them I'm first going to describe, and that is this something called dynamic instability based search and capture. So what is this? So you can see this cartoon. So you have this spindle pole body, and there are many microtubules which are growing out of this spindle pole body. And these microtubules are dynamically unstable. So they can grow and they can go to a state where they start shrinking. And using this, so basically what they can do is they can randomly grow and shrink in some direction. And you think that this kinetochore is also moving in the bulk cytoplasm. And whenever this microtubule comes within the close proximity of this chromosome, it attaches to this kinetochore and a capture event has happened. And one can actually write down a model for this dynamic instability of microtubules. So this has been very well studied, and this goes back in 1993. So you can think that when the microtubule is in the growth state, it can keep on growing with a growth rate V+, and then it can switch to a shrink state from the growth state with a rate F ±. And once it is in the shrink state, it can shrink with a rate V-. And again, it can go back from the shrink state to a growth state with this frequency F-+. So this is a very minimal model, and one can, using these known parameters, perform a computational simulation of these dynamics of this microtubule growing and shrinking, and it can look something like this. The simplest one, where what will happen is you have a microtubule growing. When it switches to a shrink state, it shrinks all the way. And then once it has shrinked all the way to this nucleating point, it again starts growing, but in a new direction. And that is again grows. And because this is a stochastic process, these lengths of growth can be different. And this is, as I have said, this is the simplest model. I am allowing the shrinkage to happen until it reaches the spindle pole body. One could have had something like a rescue rate where it can recover to a growth state sometime in between. And one can study these computationally, generate the length trajectory, like the length of I think there is a question. Yeah, please go ahead. You can switch on your mic and ask me the question. I cannot hear anything actually, so
maybe just is there a question? If there is, please either speak it out or type it out if you can't speak.
Okay, then if there is no question at this moment, I can continue. But if somebody types, you can tell me the question.
Will do so. Yeah.
Okay. So so this is something which has been well studied, and one can use this information and perform Monte Carlo simulation, a kinetic Monte Carlo simulation. This is also known as the Gillespie simulation, which might be already well known to many of you. One can also write down an analytical formula for the time evolution of the probabilities of these microtubules, either in the growth state, which is called P+, or in the shrinkage state, which is P-. And you can see that when it is in the growth state, it grows with this red V+. And when it is in the shrink state, it shrinks with a red V-. And this gives rise to some kind of an advection term, or which is like the first term on the right-hand side in both the equations. And the third term and the second term in the first equation is the rate of switching to a shrink state, which comes with a minus, and the gain term, which is because of switch to the growth state from the shrink state. And similarly, these two, the last two terms comes in the second equation with opposite sign as expected. So one can also study this time evolution of this length of the microtubule through these equations for the time evolution of the probabilities in either the growth state or the shrink state. So I will not spend time on this. So this is typically a mechanism of microtubule growth and shrinkage, which has been dominantly seen in many eukaryotic cells. And the idea is like you have many such microtubules growing. When one of them comes in close proximity of this chromosome, they attach to these protein complexes, which are called the kinetochore. And then the capture has happened. And then one can go to the next stage. So right now we are interested only in this capture process. Okay. I think there is a question. Yeah.
Sir, I had a quick question. So is it like kind of exploring different ones before capturing a particular one? Like, does it like come close to one, then maybe decide not to bond to it, and then like go and explore another one, and then decide to attach to that?
Yeah, so the thing is, those are more complicated processes. Yes, there can be different complicated processes, like actually like the mechanism of how this microtubule, once it is at very close proximity, it binds to the kinetochore. Right now, we are not interested with those fast time scale processes or those details. So we are assuming within this framework that whenever they come within like a very close proximity, there is an instantaneous capture. And the time scale over which this capture happens, it is much faster than the other time scales, namely the time scales over which you see this growth and shrinkage, or the time scale over which this chromosome is diffusing out in the bulk cytoplasm. But you are right that there can be more complex processes, like, I mean, what is exactly the mechanism of how it binds to the kinetochore? But at this moment, we will neglect those really fast time scale processes. So
thank you.
Does that address your question?
Yes, thank you.
Okay. So this is like a well-known model, and just to remind you, so typically in eukaryotic cells, like where this kind of mechanism is predominant, there are a lot of microtubules which are growing out of a kinetochore. Sorry, my bad. So growing out of a spindle pool body, this nucleating site. Now, recently, or not that recent, way back in 2013, this question was asked in the context of a simpler organism, namely fission yeast, that how does this capture happens in fission yeast? So first, let me give you what is the nature of dynamics of this microtubule in fission yeast. So in fission yeast, the length of a microtubule coming out of a spindle pool body, it has this following dynamics. It first grows, and then it is stuck at a pause state where the length of the microtubule doesn't change, and then it can shrink. after some time. And typically, on average, the state over which this microtubule does not change its length is rather very long. So where this length is not like, it is like a fixed, it's like a stiff rod. And what happens, it was seen in experiments that during this state, it does some kind of, so basically this rod, you can think it is a stiff rod. It does not have any kind of, like, it is not like a polymer which can bend. So because the length is small, you can approximate it as like some stiff rod, and it is like pivoted at this spindle pull body. And being pivoted at the spindle pull body, it is making some kind of a physical movement. And for the moment, let's say that we don't know what is the nature of the movement. So the question is, what is exactly like the kind of motion it is doing? And it was seen in experiments. So this is from a real experiment. And if one observes carefully, so this whole thing, if you see the cursor, this is the spindle pull body, and this movie is looping. So this object, it is a projection. So sometimes you don't see the rod properly, but this rod is doing some kind of a motion, which if you follow the snapshots from the movie, you can see and try to build up a schematic. So there are these slight movements being pivoted at this end, and the chromosome, or actually you don't see the other part of the chromosome, you see only the kinetochore, which is this magenta thing. This thing is also moving a little bit. And one can see that if you draw a line, it is with respect to some axis, it is making some kind of a pivoted motion. And whenever it comes in close proximity, this kinetochore is immediately captured. And once this kinetochore is captured, it is like really pulled very fast by that microtubule. So now the question is, as I have said in the first slide, that given this experiment, can we quantify the nature of this motion of the microtubule and also the kinetochore? And this is the question, and we will try to do that. So first, before doing that, let me digress a little bit and give like a quick overview of how we can quantify motion in a very complex environment. So let's start with something simple. So let's start with something like diffusion, pure diffusion in a viscous medium. So you have some particle, which is the yellow particle, and you have a background fluid, which is composed of these small dots. Now, the fluid is in equilibrium at some temperature T, and it is getting bombarded by this, and the bead, and this yellow particle is getting bombarded by these fluid particles, and it performs some kind of a random zigzag motion. So this problem has been studied very well many years back, and one can write down the equation of motion of this particle. And what you see here is this equation of motion of the particle. There is an acceleration term, which is the first term. And then there is a friction term. It is like a frictional force, because every time it gets bombarded by these fluid molecules, it feels a drag. And that is the second term. And the term on the right-hand side of this equation are these random kicks, which this yellow particle experiences due to the fluid molecules at every instant. And this random kicks has certain property, which is given by this equation. Basically, they are called, like, these are random forces, which has a mean 0. If you average, it is 0, and they are delta correlated. Now, as I have said, that we will consider that this background fluid is very viscous, which is typically the case for fluids inside a cell. So if it is viscous, then we can drop the first term, and this motion will be something called overdamped. And in that case, this equation simplifies further, and one can calculate something called a mean square displacement, which is this quantity. It is basically the square of the displacement of this particle averaged over various trajectories, or averaged over time. And what we see from this formula, I'm not going to the details of this formula, what we see is this average, or this mean square displacement, is proportional to the time elapsed. It is linearly proportional to the time. Now, this is for simple diffusion in a viscous medium. when we have a more complex medium, this formula will be modified, where it will be this mean square displacement will be proportional to this time to the power some exponent alpha. And this exponent will be 1 for free diffusion, as we saw in the previous example. And it can have a variety of different values. For example, if this particle is moving with a constant velocity, it will have an exponent 2. If this medium is very crowded, it is bumping into obstacles, then this exponent will be less than 1. If it is like a medium where sometimes it moves with some velocity, sometimes it bumps into obstacles, and sometimes it diffuses, it can be something in between 1 and 2. So it can be a range of values depending on the background medium. And here is the important part. So if I am able to calculate this quantity from experimental data by measuring, by looking at the variation with time of this quantity and what is the value of this alpha, I can tell something about what is the nature of this medium, and that can help us in building up a model. So this is the main idea. So what we will try to do is we will try to calculate this quantity for this microtubule doing a pivoting motion and this kinetochore diffusing freely. Let us see. So what we can do is we have this microtubule. We can choose an axis and measure the angle theta with respect to the axis and measure that angle theta as a function of time. And once we have measured this angle theta, we can calculate that square of that theta over various trajectories and plot it as a function of time. And if we plot that as a function of time, this is what has been plotted here, these dots with error bars, and you see that it actually, you can fit a straight line, which means that it grows linearly with time. So it tells you that this rod or this microtubule with a constant length, it is getting bombarded by the background fluid molecules, and it is performing some kind of a rotational diffusion. It is rotational diffusion because the other end of the microtubule cannot detach from the spindle pole body. It is always attached there. And this rod is getting kicked by these fluid molecules, and being hinged at one end, it is doing a random motion. So basically, from this information, if you fit a straight line to this data, which fits very nicely, the slope of this fit will give you the diffusion coefficient or rotational diffusion coefficient. So this is something which you can extract from experiment. Good. What about the kinetochore or the chromosome, which is diffusing freely? What we can do for that is we can look at that fluorescent point and mark the position at every time and record the position. And that will be some R of t. And then we calculate again the R square averaged over different trajectories and plot it as a function of this time elapsed. And here also we see that if we try to fit this data, of course, at large times, there are some variabilities. There are some fluctuations in the data, but still we can fit this data with a nice straight line, which tells us that this kinetochore is also diffusing freely within the bulk fluid. And if I calculate the slope of this fitted straight line, that will tell us what is the diffusion coefficient of the guy. So I have got two pieces of information from the experiment, that the microtubules are doing a rotational diffusion, the kinetochores are doing a free diffusion within the bulk space, which means that whenever they come within a close proximity, there will be a capture. So basically, using this information, I can try to build up a model. And let us see how we can do. But before that, we also need to look at some statistics. What is a relevant statistics here? A relevant statistics is that suppose we look at so many cells and look at this capture process in many cells. And we ask the question that what is the fraction of lost kinetochores? And this fraction of lost kinetophores, so these dots are the averaged values of the fraction over time. We see that this fraction is decreasing over time. Now, I should clearly explain this quantity. So this is a very crucial quantity which you get from experiments. And let me explain it a little more carefully. Okay. What is this quantity, fraction of lost kinetochores? So suppose I have each of these spheres, which is the nuclei of each cell. So in fissionist, the capture happens inside the nucleus. So each of these sphere represents, it's the cartoon for one cell nucleus, and each of these dot is a kinetochore which is diffusing freely, and each rod, this green rod, is a microtubule. There can be multiple microtubules, but I'm showing just one microtubule which is performing a rotational diffusion, which we just saw from the analysis. Now suppose we are looking at an assay where I have like n cells I can look over time together, like in this case 16. So at the beginning, so I have marked 3 different times, T0, T1, and T2. T0 is the starting time where I start doing the experiment. T1 is a later time, and T2 is, say, the final time. So at the earlier time, when the experiment has started, none of the kinetochores are captured by any of these microtubules because you have just started the experiment. So you can say that how many kinetochores have survived. Here survive means they have not got captured by the microtubule. And in that case, it is 1 because out of the 16 cells, all of them have survived. Okay. Say in the next time instant T1, you see that out of these 16, some of them has got captured. So you can see which are the ones which has got captured. So you can see the cursor, right? I cannot see, I mean, it might be good if somebody you can ask me questions in between, like if you have any doubts. So yeah, so in the second one, you can see that out of those 16, some of them have got captured. This one, this one, this one, and this one. So out of 16, 4 has got captured, which means the remaining 12 has survived. So if I take the ratio, I can say that the fraction which has survived is 75% or 0.75. Now, as time progresses, more of these kinetochores in the different cells will start getting captured. So if I go at a much later time, T2, I see that like 12 of them has got captured. So now the fraction which is surviving is 25%. So this is what has been plotted in this experimental, from this experiment. So this fraction of lost kinetochores is basically the fraction of cells in which the capture has not happened. And in the stochastic processes literature, this quantity is called the survival probability. And I will come to this in a few later slides and explain this thing a little more in details. So now I have this nice statistics which tells you that if I look at an ensemble of these cells, this is typically the survival probability. Because each of them is a stochastic event, I can only talk about these capture processes in a probabilistic sense. So that's why I need to plot this probability distribution. So now I have got 3 quantities: this survival probability, this information about the rotational diffusion of the microtubules, and the free diffusion of the kinetochores. So now let us try to build the model and let's see what do we get. So again, what are the inputs from the experimental data analysis? So this is a fissionist cell. If I zoom in inside the nucleus, I see the rod is doing a pivoted rotational diffusion, the kinetochore is diffusing, and which means that I should somehow be able to write down the equation of motion of the rod. And I should be able to write down the equation of motion for this free diffusion of this kinetochore. And all these things are happening inside the sphere of some fixed radius, which means I also need to put them inside the sphere, which means there should be a boundary condition. They cannot escape the sphere. And once I have this model based on this information, I should perform this, take this model, run this model for n different cells exactly in the experiment, and I should try to generate or recreate this survival probability which I have seen in the experiment. So let us do that. Basically, I should combine this information in the two red boxes and try to explain this plot in the blue box. So are there any questions so far? If yes, I can take this question and then go ahead. Okay, I think there is one question. You can unmute yourself and ask me the question. Kasturi?
Yeah. So I was wondering, when we took away the viscosity from the equation and considered them, like no contribution from there, is this still applicable where, or rather, what should we do where the viscosity of the cell may change and that may be, say, a disease state or a developmental state, right? So we reconsider everything or add that factor back in, or like we have to measure, I guess, the viscosity in those different situations. And that is a probable scenario, I think, in some of the cellular states. But I don't know if it's within a ballpark, that if it changes within that ballpark, you don't care. So what's the approach here?
Okay, let me try to understand the question. So first of all, here, what we are doing is we are going towards the limit where the fluid is more viscous. We are not taking away the viscosity. So basically, we are saying that, I mean, if I try to give an example, so say initially it was in a fluid which is like water. Now it is in a fluid which is like, say, glycerol, which is more viscous. Right. And when we put this bead in a more viscous fluid, which is typically like the kind of environment inside cell, this particle cannot accelerate. So you can drop this term. Now, whatever is the viscosity, based on that viscosity, one can estimate the diffusion coefficient. For example, if we know like there will be a wide range of protein molecules inside cell, and given like the protein size distribution and other parameters, and if we know the viscosity, one can calculate for those proteins the diffusion coefficient for a given scenario. Now, if, for example, the viscosity in a given situation, say in a disease state, suddenly increases compared to the normal viscous conditions, then one has to somehow use that as an input in the models, because when you see like from this equation, when we write down the simplified equation for a viscous medium, I have this quantity D. Now, D is a function of the viscosity of the medium. So as per your questions, right, the viscosity increases, that should be reflected in the value of D. Okay. Now, if the viscosity is a time-dependent quantity, that is like a more complicated scenarios. So then that has to be also captured by a change in D, I would say.
Right. So using that, it can be also predicted, I guess, if viscosity changes, if D changes, right? What would be the scenario, correct? And then that could be tested in a say disease state or in a situation where viscosity apparently changes.
Yeah. So the thing is, for example, I mean, one thing you can see clearly, for example, if you calculate this quantity, so suppose the D has changed. Okay. So suppose you do this experiment, you record you measure the position of a particle over time at two different times, at an early time when, say, the viscosity was something, and at a much later time when the viscosity has changed. Okay, now assuming that there are no crowding, which I was talking about, like there can be more complications, like assuming that this is really like a simple scenario where it is like a simple viscous fluid, you can see from this formula. So I didn't explain the formula. So the small d is the dimensionality of your problem. So if you are making this measurement in 2D, this d will be 2. If you can record the trajectory position in 3D, in 3 dimensions, this d will be 3. So for the moment, let's assume that you are making this recording in 2 dimensions. You put it under microscope. Now, if your viscosity has changed and it is still a simple fluid, then this slope, this d value will change, and that you can find out by fitting the experimental mean square displacement by this formula.
Thank you.
Okay. There is another question, I guess. Yeah. Hi, Devinder, please.
Hello, sir. So, so I would like to know that when from the fixed point, there are number of microtubules coming out. So is there any competition between these microtubules to search and capture the same kinetochore or same chromosome?
Yeah, so this is a very good question. So see, these microtubules, when they come out, they have a physical volume. So for example, these microtubules cannot come very close to each other. So there will be some kind of excluded volume interaction. That is one thing. Now, in this scenario, there is no preference for within this framework, what we see experimentally and what we try to model. We don't have any preference of any particular microtubule trying to find, like looking for a particular chromosome, a kinetochore or so. So basically what we are saying is, like, if you have like n microtubules, the kinetochore movement is completely independent of the microtubules. It is independently diffusing, and any one of those microtubules, when they come in close proximity, there will be a capture. But there are other organisms where they use more complicated forms of this search strategy. So for example, I forgot the organism. Is it in Xenophus egg? So what happens there? So there is a chemical gradient around the kinetochore. So this is a gradient of something called RAND GTP. Okay. And this chemical actually, whenever the microtubule senses that chemical, its growth state is stabilized. So it is in a preferentially more growth state than shrinkage state, which means that there is a chemical gradient around the kinetochore. When the microtubule starts sensing the gradient, it will bias its growth more towards that kinetochore. And then this actually enhances the speed of capture. Okay. But in this case, we don't have, at least in Fissionis, we don't have any evidence of such kind of a biased capture process.
Okay, and also these microtubule, each microtubule is going to have one chromosome. Once they capture the kinetochore, then it's not like that chromosome 1, chromosome 2, 3, so many chromosomes are there on the single microtubule. So how that one microtubule per chromosome, what actually is indication for that? How microtubule get to know that, okay, now the work is done and there is no more search and capture thing?
Yeah, this is a very good question. I don't have a very clear answer to, like, because there are multiple chromosomes and all these captures are happening simultaneously, right? So like once one chromosome is captured, like how the next stage of processes start, and how whether one should actually in the model simulate all the chromosomes and the microtubules together, or like number, like if there are n microtubules and m chromosomes, whether we should do like some like a lesser number of microtubules per chromosome. So this is an interesting question. And so the detailed mechanism of once the capture has happened, what happens, how they are positioned and how they are brought in the spindle, I don't know much of this. So what we are trying to do here at this level of the model, so I know in Fissionist, I think there is of the order of 3 chromosomes in total. And like there are typically 3 to 5 microtubules per chromosome you can think of. And that is what in this simple model scenario we are trying to model. We are saying that, okay, so I will just worry about like how, if there are 3 or 5 microtubules, how they are competing against each other to capture a single chromosome.
Exactly.
So does that address your question?
Yes, very
So
well.
the actual process is, yes, more complicated.
That is true. Thank you.
Okay, good. So I can go ahead. Yeah. So now I will try to build up the model, and let us try to do that. So the model for the microtubule, it has this following equation. So I won't have much time to actually go to the details of this equation, but what I can do is I can explain. So again, so what you have is, so we are modeling this rod pivoted here of constant length doing this rotational diffusion motion. And we consider at the level of this model, we consider this rod in 3D, which means you can think that you have a rod hinged at one end and the tip of the rod is moving on the surface of a sphere. So since it is in 3D, if you go from Cartesian coordinates to spherical polar coordinates, and because the radius is fixed, because the length of the rod is fixed, you need 2 variables to completely characterize the motion of the rod. One is this angle theta, another is this angle phi. So this is like the polar angle, which is the angle traced out on a plane, and this theta is the azimuthal angle. And what we have written down here is the equation of motion of these 2 angles of the microtubule, the theta angle and the phi angle. And you can see that because it is a rotational diffusion motion, as we saw in case of simple diffusion in free space, there was a noise term, there was a random kick. Here also there is a random kick with a prefactor, which has this diffusion coefficient of the microtubule. And unfortunately, there is some more complicated term in the front, which is this nonlinear term, which is a cosine of this angle divided by the sine of this angle. And the second equation also has a noise term, but a nonlinear term, which is like the inverse of sine of this. Now, one can systematically derive this set of equations by considering a point diffusing on the surface of a sphere in Cartesian coordinates and doing a coordinate transformation from Cartesian coordinates to spherical polar coordinates. And one would get this equation, which we did while we derived this equation. And this is a consistent equation. So there are more complex details in this model. For example, this diffusion coefficient of the microtubule. So this we estimate from the experimental fits, which I have shown few slides back. However, this one can show that for a rod pivoted at one end, this diffusion coefficient has a dependence on the length of the microtubule. It has a very sensitive dependence on the length of the microtubule. It depends, it goes as 1 over L cube of the length. So you can see that if this length starts growing, this rotational diffusion will become much slower. Okay, so this is the details of this equation for the rod pivoted at this end. The equation of motion for the kinetochore is rather easy. It is basically exactly the way we wrote down the equation for a diffusive particle in a viscous medium, where it is like a highly viscous fluid and it is moving freely within that fluid. And this is the equation of motion. So at every instant, it is getting some random kicks, which is given by this xi of t on the right-hand side. Now, of course, we have to put the boundary condition, and we know from experiment what is the typical size of a nucleus. It is 3 microns, which means that if we put this boundary, this rod cannot go out all the way. So when it reaches this extent, it will feel a physical barrier, and its motion is bounded by the actual bound of the nucleus. And all this like this kinetochore is also diffusing within this, so it cannot leave this whole sphere. And what we can do is we can take these two equations, put this boundary condition, and perform a simulation of this set of equations. And of course, now we can simulate for n such nucleus and keep on tracking, like over time, when there is a capture of a chromosome by this microtubule. And if we do that, we can now exactly calculate the fraction of lost kinetochores from our model. And I wanted to tell you one thing. So in this model, what are the parameters? The parameters are this D, which we measure from experiment. The diffusion coefficient of the kinetochore, which is also measured from the experiment, from the fits of the mean square displacement. The size of this sphere, which is known, the average length of the microtubule, which is also known from experiment. So while running this simulation, there is no fit parameter except for the number of microtubules which we are varying. Because if we just have one microtubule, we get this curve, which we see does not agree very well with the experimental data. But as we keep on increasing the number of microtubules, we see that when we have of the order of 3 to 5 microtubules, we get a very good agreement. And this is also consistent with the observation in experiment, which is known that for fission is the number of microtubules which are growing from a spindle pool body is roughly of this number. There are not many microtubules, unlike in other eukaryotic cells where you have like of the order of hundreds of microtubules. So this, first of all, we have a very good agreement between the model and the experiment, and it gives roughly a good ballpark number of the number of microtubules which we get out of the simulation. So this is what we have reached so far, that we started with some experiment. We analyze the data. Based on the analysis, we use them as input to construct this model and try to make a comparison with the statistics from the experiment, and we got some agreement. Now the thing is, we should not stop here. Ideally, we should now, because we have a model where we have a very nice set of equations which you can see, so we can actually play around with these different quantities and ask various questions. Okay, and that will be the next part, that can we like do something more than just proposing a model for this very specific set of experiment which is in fissionist. Okay, I mean, can we go beyond fissionist, or can we say something more about fissions, or in general more about this kind of capture processes in general? So let us try to do that. So the question is, what else can we learn from the model? And another interesting thing is, this model can be mapped to a very important problem, which in stochastic process, which physicists have been interested for a while, and it is an extension of those class of models, and that I will try to explain here. I will like try to give a general framework of the theory on which this which this model is a part of. So I need to digress a little bit and introduce something called a first passage process. And what is a first passage process? So the idea is the following. So suppose you are recording some random variable, some x as a function of time, and you ask that when this variable reaches a threshold value for the first time. And why first time? Because this variable is a random variable. So every time you start experiment where you start tracking this variable, the time taken by this variable to reach that threshold will be different. Okay, so basically what you can do is, and this variable can be anything. In our case, it was basically, say, the position of a particle reaching some target. It could be some chemical like concentration, like a protein number trying to reach the threshold, such that once that threshold value of the protein is reached, some other chemical processes start. So it is very important that it reaches a threshold, and you see that every time you do the experiment, what time it is taken to reach that threshold. It can be some stock market index also, which is a stochastic variable. So it has to be a stochastic variable. Its time evolution cannot be deterministic. So now you have this variable. So what a simple example is, you have a random walker, and the random walker cannot control its trajectory. It does a completely zigzag motion like the particle in a viscous medium. And you ask the question that when does it hit the lamppost for the first time? So if you do this experiment with this random walker n times, every time you will record a different time. And with this set of times, what you can do is you can plot the distribution or the histogram of this time, and that distribution is called the first passage probability density. And this first passage probability density is related to something which I introduced in the previous slides called the survival probability, in a way that the first passage probability distribution is nothing but the derivative of the survival probability with a negative sign. The negative sign because as time progresses, the survival probability decreases. The chances of surviving becomes lesser and lesser. So if you ask this question that in between a time instant t and t plus delta t, what is the probability that it will get captured? It is ft/dt, and that is the difference of s at t plus delta t minus s at t. And if you do like first order Taylor expansion, we'll get this formula. So it is a very simple relationship between this first passage distribution and the survival probability. Now, this class of problems has like a wide applicability in natural sciences in various fields. And again, I am digressing a little bit. So you can think of, again, revisiting this Brownian motion problem, that now I put a flag, which is a target. It is a fixed target. And you ask the question that every time this particle starts, what is the time taken to reach the target? Now, although this movie is looping, so every time I had started with doing this experiment, the times taken will be different. So what I can do is I can record the time and calculate the first passage distribution. In this case, I know the equation of motion of this particle. And if I know the diffusion coefficient, I can run a simulation and I can, again, get the distribution. There are other techniques also, which I'm not going to go into the details, that you can write down the time evolution equation for the position of this particle. Like you ask the question, what is the position of the particle at some time t, given that it started from some initial condition, initial position x0 at time 0. And from this equation, you can write out this equation, which is the diffusion equation. And you can calculate the survival probability by integrating this probability distribution. And you can, from this, calculate the first passage distribution. And again, there is another way to directly calculate this probability of survival. I'm not going to get into these details, but the point is, this is a very simple example. Now, if I try to add some complexity in this problem of the random walker, I say that I have this random walker. Now, I confine this random walker in a box, which means in the previous case, this random walker could have a possibility to escape to infinity. But once I put a box, it can never escape to infinity. So the mean time to reach this target will always be finite. Okay. I can add, and now this problem can be solved exactly with this target being fixed with whatever boundary conditions. I can use analytical techniques to exactly solve this problem. I can add an additional complexity where I say that, okay, there are more random walkers. Instead of one random walker, there are n random walkers. Everybody is trying to reach that target. And I will just consider the situation where one random walker reaches this chromosome, this target, and the the process is over. And even that problem is simple because this is a static target, and this, if we assume that these random walkers are independent of each other, I can solve it as, and I can factor out this independence. I can use this independence of the random walkers and factor out the problem, and that can also be analytically solved. However, if I now make this target also move around in this complicated problem where I have n random walkers and this target moving around, then this problem becomes very complicated, and one cannot use pen and paper to solve this problem, and one has to use some kind of a computational method to solve this. I think there is a question. Yeah, please go ahead. Also, I should have a track.
Yeah, sir. I had a quick question. So if the target is also moving, right?
Yeah.
Will like the intuitive sense I get is that maybe we cannot find a converging solution, right? Like can you guarantee that there is a solution? Like, could it, in the worst possible case, they will never reach the target? Like, if because I'm assuming that the target is also moving randomly and the random walker are also moving randomly. So, like, could they evade each other forever?
They can escape each other forever. In this case, no, because you have put an external boundary. So this boundary will never allow this target to escape forever. So you think of this, because these are not very smart walkers. They are just diffusing randomly. So because of this confinement, both of them are moving randomly. So this capture on average will always happen in finite time. And actually what you have asked is a very important component, question in this study, that if there was no boundary, even with the static walker, this could have, once in a while, this walker could have escaped to infinity, which means that if I compute an average time, this, depending on the dimensionality of the system or the complexity of system, it can be infinite. But if we have a finite bound on this problem, this is never going to happen. It will always get captured. So,
and can one write a closed form expression for the average time it takes if the target is also moving and the walkers are also moving?
No, so that is the problem, and that is also a very good question, because, so as I was telling, I didn't give you the details. So let us try to see. So if I have this problem, I have a target and I have a this thing, no boundary, a single target is fixed. In this case, I can solve any of those equations which I showed in the previous slide in a semi-infinite boundary, and I can calculate the survival probability, and the survival probability has a power law solution. The moment I put this boundary, this survival probability will have some dependence on time with an exponential cutoff. Means, what I mean by that, that the survival probability at large times will be something like an e to the power exponential of minus t by tau, which means that the survival probability will always, like, everything will always get captured because of the boundary with a fixed target. And there also you can calculate the mean time and you can have an exact closed form solution for the survival probability. Now, when you have this second problem, you have n walkers. Now I am assuming, since I can solve the problem for one walker with a fixed target in a closed confinement, for n walkers which are independent of each other, I can say that I know the solution. For n walkers, it will be whatever the solution for the one walker to the power n, because they factor out because they are independent of each other. If they are not independent of each other, if there is an interaction between the walkers, then the problem is more complicated. You cannot solve. But if you assume that these walkers don't see each other, they can cross each other, then this problem is still solvable. The moment you put movement of this lamp, this target, then you cannot have a closed form solution. All you have to do is, you can have a closed form solution in a simple case of one worker and one target moving, but not for n workers. So that is a very simple case where you can get a closed form solution. And but otherwise, this you have to solve using computational methods.
Thank you.
Okay, and this is very important because now you can see when I have designed this problem, this has been designed as a physics problem, and the physics community or the stochastic processes community had been interested in the problem, and there has been several papers in the study of this problem without the confinement. The moment you put confinement, it becomes very complicated, and you can clearly see that with this simple picture, now you can relate to another problem, right? And this is nothing but our problem, which of course has a more complicated like picture where you have rods diffusing and kinetochore, but roughly in spirit, it is exactly the same problem. You have this random motion of the microtubule, random motion of the kinetochore, everything happening under confinement. So this is the kinetochore is the target. These things are the workers. They have a more constrained motion. And what we have found is that because of this mapping, some of the general results, which we see in this case, in the left-hand side case, is also seen in this more complicated biophysical problem. And these were results which were discussed in some of these papers we wrote in 2020. I will not go into these details. And so this was a general extension. The other direction you can ask is, recall in the one of the early slide when we introduced the problem of search and capture or capture of kinetochores in fission yeast, we discussed that in eukaryotic cells, the capture happens in a different way. They don't do, the microtubules don't do a rotational diffusion. There are many microtubules, and each of these microtubule is dynamically unstable. They are doing dynamic instability along a given direction, growth and shrinkage. And that leads to capture. So now with this model, and because we know also the model of dynamic instability, we can create a hypothetical model, a toy model, where we use the parameters for fission yeast and ask this question that in fission yeast, why fission yeast is not using dynamic instability based search and capture, and instead it is using like a mechanism where the rods can do rotational diffusion. I mean, it could have done a dynamic instability also like the other organisms, and why this is not happening? Is there anything we can find out by comparing these two? So the question we can ask is whether to pivot or not to pivot in the context of fission yeast. Why fission yeast is pivoting? So this is kind of a hypothetical question, and basically at the level of model, we can compare these two scenarios. We compare a model where we do dynamic instability and compare a model we do rotational diffusion and vary the number of microtubules and see, we compare the time efficiency, which one happens faster. So yeah, so I wanted to discuss the second model. So this study is quite involved, and it will go into much details. So I can continue on this problem, or rather go to a different example, but let me first give you like the key result. So what we found that with this comparison, that we found some like from the physics perspective or the stochastic process perspective, from the perspective of getting some general understanding, what we found that, and it is a very crucial crucial understanding what we gathered during the study, that if you are looking at a capture process, a stochastic capture process inside confinement, one should not, one should take the mean capture time with a pinch of salt. So because this mean, any kind of average behavior could be a little misleading. And that is introduced because of a very compact, because of confinement. So I'm not going into these details, but if somebody is interested, I can go and show you what I mean by this. So this was an observation. And then the question is, so typically when we do some experiment or some study of some stochastic time evolution, typically what we do is we calculate the average, the second moment, and so. So it is very standard to calculate a mean. Now, if we cannot calculate a mean, what should we calculate? And what we found was, in these problems with confinement, there is an additional time scale, which is very robust to any other variation, and this is called the characteristic time scale. Again, I'm not right now going into the details. And this characteristic time, we can also use something called extreme value statistics and quantify it from experimental data. Okay, so this is a very important and robust quantity. And what we found by running this comparison between these two different models, it gave a prediction that if you have less number of microtubules competing for, like, trying to find a target, a moving target, then rotational diffusion is more efficient. If you can grow many microtubules, if you have like a large number of microtubules which can, like, you have a larger source of tubulin, then it seems this search and capture or dynamic instability based search and capture is more efficient. And this is somewhat interesting because in fissionist, we have noticed that the number of microtubules growing out of a spindleable body is quite less, where we found this was the dominant mechanism. Whereas this search and capture, which is observed in higher order eukaryotic cells, there the number of microtubules is very large compared to fissionist. So this actually gave us some idea or some hypothesis about the biological function. And this is this was roughly the problem which I wanted to tell you, or give you an overview of how you can use the combination of experimental data analysis and modeling to get insight about the biological processes or that is happening inside, at the level of cells or tissues. I don't know how much time do I have. Do I have sufficient time? Also, the thing is, I have skipped a lot of details here, and I can also continue and go into the further details of this problem, or discuss briefly about another problem where we can take a similar approach. So I don't know. So if somebody has any questions, I can take some questions. For the moment, take a pause and address the questions.
Professor Amitabha, the scheduled time is till 1:15, but maybe we can go till 1:20 or so. And I personally would say we can look at another problem, so that gives us more variety. But I leave it up to you to make the final call.
Okay, sounds good. So then we can discuss the other problem. So are there any questions so far on whatever we discussed until now?
Hello.
Hi.
Hello?
Yeah.
So I wanted to ask, in this, while doing the simulation, so to to apply the boundary condition, as in the confinement, like in the simulation, how do we do it? Do we put some potential, like some bouncing potential or something like that?
Yeah, it is something like that. But it is like a very hard potential. So you can think of something like a reflecting boundary condition at the boundary. But I mean, we exactly don't know how the microtubule in reality will interact with the nuclear envelope. So if we know, those can be taken into consideration, but for the sake, at the level of this simple model, we just it is a hard wall. So whenever it hits the wall, it feels it gets reflected back.
Okay, thank you very much. Another thing is like this, the characteristic time that we're talking about, like from what physical quantity are we seeing the characteristic time?
Yes. Yes, this characteristic time, this characteristic time, I have to spend at least 5 minutes to tell you what is this characteristic time. So maybe I can digress and then very briefly discuss the other problem. Yeah, should I? Yeah, maybe. So it requires some I will first let me see. Okay. So again, I'm showing this through this cartoon. So consider I have these two random walkers. One is a random walker, another is a target, and they are diffusing freely. Now they hit each other at some time t, and if I do this experiment again and again, I can find a distribution of this t. Now, as I have said, in free space, one can show that this survival probability will have, at large times, have power law dependence on the time. So it scales as t to the power minus beta, and this beta can depend on various things. It can depend on the dimensionality of the problem, and if you introduce more complexities in the problem. But for the moment, like if it is in free space where you don't have any bounds, it at large times it will be some power law. That means now if I calculate the average time is nothing but whatever is the survival probability, it is the total cumulative distribution of the survival probability or the area under the curve. One can show that. So one can also, so if this is the first passage probability, the average time is t times ft, and this relates to, it just gives you an integration over all time of the survival probability. So you can see that if you have like a power law, and depending on the exponent, for example, in like simple problems, this depending on the exponent, this mean time can diverge. And the intuitive way to see it is, because once in a while, every time you do this experiment, once in a while, this will escape to infinity. So the mean time will be finite. Now the thing is, the mean time will be infinite. Yeah. Now when you put a box, mean time will always be finite, and one is guaranteed to get an exponential tail of the survival probability. So whatever is the survival probability form, it can be very complicated. At large times, it will decrease exponentially fast. Basically, it will have all the polynomial powers in its form. And if you have such an exponential tail at large times, you can see that I can write some t by tau, and this tau is nothing but the characteristic time. And this tau depends on the like the intrinsic parameters of the problem. And for some simple cases, one can calculate tau, but otherwise you have to estimate it numerically. So this was another thing I will briefly tell you. So in our problem, because it was a numerical problem, it was very difficult to find tau, because you see S, this survival probability only has an exponential tail at very large time, which is very small when this survival probability has become very small. And to computationally extract tau is very challenging. So it is associated with the rare capture events. So what we had was, we actually found out an algorithm. I will skip that. We found out an algorithm to find out this tau, and which is like using some kind of a replication. Again, this is, I'm not going to discuss. And we found that we can estimate tau up to a fair degree of accuracy and generate the survival probabilities up to a very high precision. So the bottom line is this characteristic time tau is associated with this exponential tail of the survival probability. So I gave a longish answer, but I hope that addresses your question.
Yes, thank you, sir.
Okay. So let me go back and spend the next 10 minutes or so to discuss this other example. So I will, now that we have discussed at least the first part, at least the formulation of the problem rather in details. So this second problem, I will try to like go a little faster. So in this problem, what we are interested in, we wanted to see transport of cargo or along these very specific neuronal cells, which are called axonal cells. So this is a warm body, a C. elegans body, and this is a cartoon, and what has been labeled inside this C. elegans schematic are these different axonal cells. So what we are interested particularly in our study is this particular axonal cell, which originates from the posterior side of the worm body. Now, if I try to zoom in and show a schematic of this axonal cell, it looks like the following. So it has a very long shaft or axon, which is made up of microtubules. It has a very condensed cell body. And this axon has a polarity. And it is the polarity of the microtubule, because the microtubules themselves has polarity, which is a structural polarity because of the way these alpha and beta tubulins are arranged. Now, what we are interested here is how these specific motor proteins, namely the kinesin proteins, which always walk from the minus to the plus direction along microtubules, they move along this axon in a directed fashion. And so these are the different parts. So this is cell body, this is the distal teeth. And so basically what these kinesin motors will be doing, they will be carrying cargoes or nutrients from the cell body, and they will walk along the microtubule and try to reach the distal teeth. Okay, so there are also connections to the other axonal cells through the synapses and whatnot. And just to give you a recap of what these kinesins do, in case like I'm sure everybody knows. So these kinesin motors, they are able to hydrolyze ATP, and whatever energy released, they can use this chemical energy to do mechanical work, in a sense that they can walk along the microtubules carrying a cargo. So this is like a GIF of this movement of a kinesin carrying a cargo. So they move with a constant speed. And this has been very well studied over like the last 2 decades or so. And like typically, if you have this is a typical speed of a motor walking along a microtubule, 1 micron per second. And if you know the diffusion coefficient in free space, one can see that why for traversing large distances, it is always time efficient to use this kind of ATP hydrolysis and transport material using this kinesin motors. Okay. So one can also model the dynamics of this, like attachment, detachment of these motors to the microtubule and all these things. So these are like has been all been very well studied over the last last 2 decades or so. Now, given this motivation that we are interested in studying how kinesin assisted transport along these very long microtubule axons made up of microtubules happen. So we are interested in this following question. So this is like a biologically relevant question, is the following. See, think of this, that you have some distribution of kinesin motors. Now these kinesins will carry vesicles or nutrients from the cell body to the distal tip. Now you have to strike a right balance of how many kinesin motors you want to use, so that you don't deplete all the kinesin motors. Like you don't use up like by taking all of them at the same time from cell body to the distal tip, so that later there are no kinesin motors available here to carry further nutrients. So regulating the kinesin distribution over the whole axonal length, and particularly near the cell body, is very crucial. Otherwise, this can lead to large-scale disorders in a grown-up, like a proper developed organism, and neurological disorders. And one interesting thing is, so basically the question is, what is the mechanism of cargo transport? And can we build up a model? And how it is biochemically regulated, how the distribution of kinesin motors or cargo is biochemically regulated. So in this context, what our experimental collaborators, they were interested was to understand that understand the role of a particular protein, which is called ubiquitinin, and basically it is ubiquitous everywhere. And this protein is known to play an important role in degrading many other intercellular proteins. So for example, ubiquitinin, like, I mean, if you take proteins and redistribute and accumulate like at a different place, so at some point this ubiquitin will play a role in degrading those proteins at the other side. So this is known, and it is also known that if you somehow mess around with this ubiquitin protein concentration, this can lead to neurodegenerative disorders. And so the question we are interested is, does ubiquitinin also play a role in cargo transport along the axis? And if yes, how? So this is the main question. So now clearly you can see the only way to see what is the role of ubiquitinin in cargo transport is to first do an experiment in the normal wild type, and then repeat the experiment where you have somehow knocked down ubiquitinin or increased ubiquitinin. Okay, so this is what is done here. So these images are snapshots of the whole axonal cell at steady state, and the first one is for the wild type. The first one is for the wild type, and the fluorescence what you see here is that of the kinesin motors. So anywhere you see a bright fluorescence, that corresponds to a larger number of kinesin motors at that point in space. So you can see that this is the cell body. You see some brightness because of the kinesin motors, and at the distal end you see a larger fluorescence. However, if you knock down ubiquitinin, you see that there is an increase in this fluorescence, which means that there is more kinesin at the distal end. So somehow the number of kinesins has increased. So there were some other experiments also, so which is a more direct control of the ubiquitin pathway. This is this MPXP knockdown, and in that case also you see that there is an increase in the number of motor proteins compared to the wild type, these kinesin motors. So I think I'm almost like at the verge of like my time is kind of over, but let me just quickly go to the so we have several quantifications, which I won't have much time to discuss, but one can do because these motors carrying cargo are always moving from the cell body towards the distal end. So you can do experiments like make a cut, a laser ablation on the axon, and see what kind of distribution you see after the cut. And as expected, so you see that as you make the cut on the right hand side of the cut, there is an accumulation post ablation, whereas on the other side you don't see any increase, which is expected because these motors are source is in the cell body, and because this is cut, they cannot go to the other side and they get accumulated here. While on the other side, they actually ideally they should drop, but here what you see is they are roughly constant, and one should take this distribution with a pinch of salt because this intensity level, this normalized intensity level is almost comparable to the background noise intensity. So this is not very reliable, but on the other case, you clearly see an increase. So one can do all these statistics, but the important statistics is what is known as the chymograph. And I will very quickly tell you what is this. So this is the wild type, this is the mutants, the ubiquitin mutant and the BXB. So the y-axis is the time axis, the x-axis is the position axis. So one should only focus on these lines which are diagonal, or well, not diagonal, which has a slope non-zero, which are not constant, which are not vertical or horizontal. Because any line which has a non-zero slope, they indicate cargoes driven along the microtubules by the motor proteins with constant speed. So if I try to find those diagonal lines, these are these blue lines, and you see if I compute the number of, if I compare the number of lines, the number of lines has increased as we go from the wild type to the UBA1 or the FBXB, which tells you that now there are more cargoes or more motors binding the cargo and the microtubule. That number has increased from the wild type to the mutant. And if I further plot a distribution of these motors attached to the cargo, this is the steady state distribution of the motors attached to the cargo for the wild type, and this is for the UBA1 and the FBXB. And clearly you see they are plotting on the same normalized scale. You see that as I am going from the wild type to the mutant, the distribution is getting broader and the mean is also shifting. So it is an indication that the number of motors that are binding to the cargo is increasing with ubiquitination. So now I can try to ask a question. So I have this observation that these are the steady state distribution. Now I can ask a question that can I construct a model which will at least give me some insight about what is happening. So can I take 2 or 3 more minutes?
Yeah, sure, sir.
Okay. So and that is what we are going to do. So what we will do is we will write down something called a master equation. So I thought I will explain master equation, but I won't have time. But the master equation is basically you write down the time evolution equation for like the probability of some configuration of some observable with a configuration n, and you ask the question that how it came, like what is the probability of invert transition where it comes from a configuration n prime to n, and the probability in the configuration n prime was p n prime t, and any loss term, which is the second term. And this is a very standard equation, and one can derive it systematically using these ideas of stochastic processes for time evolutionary probabilities. And I will skip this example. Now, using this methodology or formalism, what we are going to do is we are going to write down an equation, master equation, for the probability that there are n motors bound to a cargo in between cargo and micro. And using the master equation formalism, I can actually write down that time evolution probability. And this time evolution of this probability, it depends on basically a gain term where a motor has attached to the cargo, which is the first term, and or basically there were more motors and one motor has detached and reached that number n. And this minus is basically the loss term from that state. And so this is using very general formalism, and these are basically the binding rates. These U plus and U minus are the unbinding rates. And for this problem, I will have the boundary conditions. And with all this, I can calculate the probability distribution at the steady state, which means the probability is not varying with time, which gives me this recurrence relation, which is within the blue box. So now the question is, here I still need to know the binding and the unbinding rates. And here I will make an hypothesis. And that is the most important parts, that we say that the unbinding rate is unbinding of a motor happens with some rate beta. So if there are n motors attached, depending on that number n, it detaches with some rate beta. However, the binding rate is given with this rate equation. So if there were no motors attached, a motor can attach to the cargo with some rate A plus. However, I can have a cooperative binding, which means that if one motor is already attached, that will increase the chances of another motor attaching. If there are two motors attached, the chances of a third motor attaching is even higher. And that is captured by the second term. And this second term is a hypothesis which we put in the model. And with this hypothesis, we can get an analytic expression for the probability distribution at steady state, which has this form, exponential form. And what we find is it gives a very nice agreement with the single model for all these different cases where these red lines are from the model. And what we have prediction or a hypothesis that only if we have cooperative binding, we can explain this change in the distribution. Otherwise, so you can see, so if the B plus was 0, this mu will be 1, and all the exponentials will be of the same, will have the same decay with number, and you cannot capture the variability in the and the thickness. So the second term, this B plus n, is very important. So this is a hypothesis from the model. Now one can go ahead and ask questions that I will skip all this, that what is the molecular mechanism of such cooperative binding, how it is controlled at a very microscopic scale. And one biological function is very clear, that in the wild type, you have less ubiquitination, less motors are binding. And it makes sense because this ubiquitination regulates cooperative binding, and you don't want many motors to bind in a cargo and get depleted from the cell body. So what we see is this ubiquitination, or this method of ubiquitination, has like a nice way to regulate the motor distribution along the axonal length. So I will stop here, and let me thank the collaborators. So the first work was this is a continuation of work which I did as a postdoc long back, and then we continued with our PhD student Indrani and my collaborator, my colleague at IIT Bombay. And the second work is an ongoing work with experimentalists from TIFR, from Sandhya's group, Vidur and Sandhya, and my theoretical collaborators, Debashish Chaudhary from IOP and Amir. And the first part, the work done during postdoc was like the experiments are done in the lab of Butterbeach. And Nina then Benjamin were working on the theory part. So yeah, thank you for your attention. So yeah, please let me know if you have any further questions. I can already see somebody I know. Hello, Professor Bhattacharya, how are you?
I'm fine. Very good, Amitabh. And thank you for coming and giving this talk.
No, thank you for the invite.
Because we are going to make it a yearly affair, the major event in this area. So hopefully next time it will be on campus.
Yeah. And Professor Amitabh, are you traveling? So especially appreciate you.
Right now, no. I'm not traveling. I'm back to the campus.
Okay.
So next time, hopefully, we'll have it physically.
Okay, great. I'll be very happy to, like, I have never visited.
Ashoka, no?
Yeah.
Good.
So sorry, I think I have taken like a lot of time. So
No, that's fine. That's perfectly all right. Are there any more questions?
So let's take a moment to thank Professor Amitabh for an excellent talk. And hopefully, as Professor Alok said, we'll get to see you at the next edition on campus at Ashoka. And look forward to that.
Thank you for the invitation. And yeah, so yeah, I look forward to next year. Great.
Right.
And
thank you.
Professor Bhattacharya, I will be in touch with you. So I will talk to you.
Yeah, we should talk.
Yeah.
For others, Amitabh and we did some work together long back.
Yes.
So actually, was a student.
One of my earlier biology works was with Professor Bhattacharya.
I think one of the first forays into biology that you did.
Yeah, exactly, on microRNA regulation. Yeah.
Good to see you.
Yeah, great to see you. So yeah, I guess you will have like a break or next session. So should I just log out or?
Yeah, I think log out.
Okay. Thank you very much.
We'll share the recording with you, sir. And yeah, and participants, the next session will begin at around 1:55. So maybe tune 5 minutes before. And talk is by Dr. Sumain Basak of NII, National Institute of Immunology. Right. So let's take a quick lunch break and see you at 1:50, 1:55. Thank you.
Window properly. The window format is prepared. Let's see. Yeah, I can probably see the participant. Just let me check the format. I think it should be fine now. Yeah, I think it looks to me I'm fine, I guess, here. Yes, that only thing that I need to change, I believe.
Yeah, I'll let's give it a couple of minutes for people to come back from lunch, and then I'll introduce you and we'll get started.
Sure, no issues. And I believe I just have a quick question. I know last time also asked. I believe these students are mostly undergraduates. Am I correct?
Sir, it's a mix. There are some PhD and master students also. There are a couple of people who are professors and practicing, like somebody from the Dairy Institute, somebody like we have a mix, but yeah, it will be fair to say that there's a mix of mostly students, but a mix between undergrads, masters, and PhD. And we have some faculty from Ashoka and other places as well. So
yeah, I see all of that is here. Bittu has also joined.
yeah. Hi, Bittu.
I know a few of them, so it's wonderful and to be back here. We'll wait for a few minutes. Yes.
So I think let's begin. Welcome back, everyone, from lunch. We are excited to have Dr. Somen Basad from National Institute of Immunology, Delhi. So Dr. Basak obtained his PhD degree from the University of Calcutta. Subsequently, he carried out his postdoctoral research at UCSD, University of California, San Diego. In 2010, he joined NII, National Institute of Immunology, and set up the Systems Immunology Lab. Since then, his research group has been combining bottom-up and top-down systems biology tools to address how inflammation is controlled in physiology and what causes dysregulated, deregulated inflammation in human ailments. His talk today is titled Probing Dynamical Crosstalk Between Immune Signaling Pathways Using Mathematical Tools. Welcome, Dr. Basak, and please take it away. Quick question, would you like questions as you give the talk or all of them at the end?
I'm fine with both. I guess I am, honestly, I have no reservation if you ask questions in the middle. It might make it more interactive. It can be very useful because if you move along in the presentation with some confusion, sometimes it becomes, right? So I'm fine.
So we can, participants feel free to ask questions, either tap, raise the hand in the Zoom reactions or just unmute and ask. Or okay, please take it away, sir.
Okay, so I think first of all, thank you, Ashish, and we have to really admit this wonderful program that you guys have set up in Ashoka, and with all of and Gautam, of course, a lot of credits. This is indeed a very important initiative in the NCR region. I'm sure that it's already flourishing and it will go to a level which it where it can actually feed back into the important research program that NCR carries out. And this is my laboratory. This is the laboratory that I have, Systems Immunology Laboratory. And just a minute, I think I just can you see my, just a minute, I think I have to go back to the paint color. Yeah. Do you see my cursor? Hello. Can you answer me, please? Do you see my?
Yes, sir. We see the cursor, but not in red. Are you trying to make it red?
I can make it red. Yeah, I think it is supposed to be in red. Yeah, I think it will be look red now. Oh no, this is the color. Yeah. Okay, it will not be. Yeah, I think this is fine. Let's keep it this way, right? You can see the cursor, right? So
yes, sir.
This is a laboratory, the systems immunology laboratory, and today I'll be talking about probing crosstalk between immune pathways, the bottom-up system biology approach. The work that I'll be describing, some of it has been done a long time back, and some of these are still continuing in some form and format. And we have a huge number of different collaborators. Of course, this kind of initiatives require involvement of multiple set of peoples. Particularly the discussion today, we'll have. I mean, Professor James Groms from IIT Delhi. He has collaborated with us in a variety of different programs where we have combined competitions and experimental studies. And students, Balaji was my first PhD student on this lab. He has left the lab long time back, but I think still discussing the work that he has done with Udaditya. He was a guy who I hired in collaboration with IIT as a PhD student. He did most of the computational biology work. And Pyle also has contributed in those earlier days, formative years of the lab in experimental analysis. And then, of course, a lot of students began to contribute, particularly Alvina and Navin. They are currently with the lab and doing a lot of wonderful work. But today, I think we will probably discuss immunology and system biology. So maybe, more of a systems immunology. So we will try to sort of focus on those, how computational, what computational tools one can use, and how they might have been useful. Before we go into that, first, I think because there are a lot of undergraduate students, or some of them are at least, maybe we should start with some very basic question. Why study immunology? And this is a couple of things. After a little investigation, one might find. Microbial pathogens, a total number of viruses on Earth, if you put them next to each other, they would stretch for 100 million light years. And total number of bacteria in ocean is 100 million times more than the number of stars we see in the known universe. And if you take a spoonful of soil, you'll be surprised. Number of microorganisms in that spoonful of soil would be almost equal to the number of human beings living in the continent Africa. So it seems that we're submerged in a sea of microbial threats from outside. And from within, we have issues too. An individual, the time we're born and the time we'll die, between these, there will be around 100 new mutations in our genome, each of us. Will they be causing cancer? Not necessarily. But hey, see the next information, that oncogenic mutations are far more prevalent in healthy human tissues. A study was done to look into breast tissues. You see a lot more frequency of precancerous lesion in healthy individual population than the frequency of breast cancer in human population, suggesting that you do develop precancerous lesion every now and then, but you do not necessarily develop cancer. As if the same thing, right? When you have submerged in a sea of microbes, you do not get infection every now and then. You are, acquiring mutations, but even precancerous mutations, but you don't necessarily get cancer all the time. And this is the answer. Who is protecting us from those external threats and those from within? The answer is immunity. The answer is immunology. And of course, COVID has made it very obvious why one should study immunology, but do not forget the internal threats also. That our immune system survey external threats like microbial infections, also look into malfunctioning cells from within, and plays an important role to limit them and eradicate them. That's why we should study immunology. That's an interesting and the most important aspects of our life. And if you see immune system, of course, this is very rudimentary of innate and adaptive immune system. Tell you what, if you look into the evolutionary tree, some of my some of my colleagues here in Ashoka would be more interested. You'll see the adaptive immune system came a lot later in early vertebrates. By the time you begin to lose your exoskeleton, some says. But if you do look into the evolutionary tree, you see that innate immune system was there even in arthropods and nematodes. It's evolutionary conserved system. And if you look into the scale of functioning, when you get a microbial infection here, you have within minutes, you get, of course, within minutes, the one you probably imagine is operating is your physical barrier. That's a part of innate immunity, right? Skin, for example, stomach pH, for example, you have heard all of this. But after that, you get an inflammatory immune response, innate immune response, inflammatory immune response that involves macrophages, neutrophils, so on and so forth. We will come to that in a minute. And then only you see within hours to days scale, adaptive immune system picks up. And as an imaginary figure here, I mean, I do not have, this is not experimental data. One might really speculate what would happen if you do not have innate immune system and you do not have adaptive immune system. If you look into the number of microorganisms in y-axis and duration of infection in the yellow line, says what happens in a normal human being. Infection, there will be accumulation of microorganism in a given tissue or in an anatomic niche, and they will probably be eradicated, and then you get a yellow line. If you lack adaptive immunity, you will see that there is a green line where this eradication is not happening. But if you lack innate immunity, soon after infection, you will see profound replication, profound multiplication of microbial pathogen for the want of innate immunity and inflammatory immune system threat, and microbes will accumulate too quickly. So this is again innate immune system. You have inflammatory response that plays a very important role in this innate immune systems. And I'm going to really talk about those inflammatory response. What are they? It's an of course form of innate immune response, and I'm going to try to pictorially present it. It would be important for us to later understand what we do and why computational methods were at times necessary. And of course, one can orchestrate things much beyond the complexity that I'll be capturing here today. So when you have these tissue resident cells, for example, it's a macrophage that is sitting in your underneath your skin in the epidermis in that layer, endothelial layer. When there is pathogen, they will of course also cause a lot of cells to die. So microbial ligands and necrotic cells that will give you danger-associated molecular patterns, where pathogens will give you pathogen-associated molecular patterns, and they will signal through TLRs. There are receptors that would recognize these threats. This would lead to activation of intracellular pathways, and that would transmit a signal in the nucleus, provoking expression of genes, particularly chemokines and cytokines. Neutrophils, these neutrophils are very important mediator of inflammatory response, but they are normally in the bone marrow. They are only few neutrophils that would be in the circulation, and they will patrol. They will, in the blood vessel, they will come in and they go by. But once these chemokines, cytokines are secreted, neutrophils will begin to adhere and then extravases into the tissue, where they will secrete a plethora of neutrophil molecules, serine proteases, myeloperoxidase. They are potent. Myeloperoxidase, for example, is of course a mediator of reactive oxygen species. There are iron chelating protein that will deplete the tissue anatomic needs of iron, so microbes cannot replicate. And these are very potent. Neutrophils, they have these granules where they keep those potent molecules, and they will help in microbial killing, and thereby allow us a chance to then trigger adaptive immune response and then completely clean up the mess that may have been caused because of microbial infection, right? So far, you have tissue resident cells. They must sense microbes and secrete chemokine cytokine to drag neutrophils and other potent effector cells at the site of infection. Even macrophages, another thing those effector cells would do is phagocytosis. They actually, this is a very nice thing, hopefully it will come up to show you.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
That was quite a burp, isn't it? So neutrophils are coming at the site of infection because the tissue resident cells. Yes, we will take up a question now. Yeah.
So I had a quick question. So this engulfing behavior, right? Macrophages also tend to do that, right? They also
Yes.
like almost eat up the invader, right?
Yes. So the point that you're making is true. In fact, neutrophils, the major mode of their microbial killing involves those granular proteins, reactive oxygen species, iron chelating proteins, so on and so forth. Even ionophores, they secrete proteins that would make membrane penetration and disrupt bacteria. Actually, phagocytosis is a minor component of neutrophil-mediated microkillings. Macrophages are indeed more potent in phagocytosis. Yes. Does that answer your question clearly?
Yes, sir. Yes, sir. Thank you.
Okay. So let's move on to the next one. What is it that we're getting here is that one question, remember that I was telling you that neutrophils are in the bone marrow, very few are in the periphery, patrolling the body. And once you got infected, tissue resident cells need to call them. That please come to the site and kill the bacteria. The question, of course, is a philosophical one, that why they are in the periphery. That is the question, right? Why you have neutrophils, those are so potent, are to be in the periphery, and why not are they? Why are they in the bone marrow and not in the periphery? Why? And that, I think, is a cartoon that really clearly make you understand what goes on in the immune system. Is this very striking contrast? Look at this person giving a parking ticket, right? And then beyond that, below it, you see these are our most prestigious Black Hat Commando. You can one day ask in parking lot, why don't we see Black Hat Commando? The simple reason that is, if you make a mistake in merely parking, they'll shoot in your head. They're trained to do that. Neutrophils are killer, and small perturbations in the body system, they would not recognize, and they would be alerted and secrete all those damaging molecules. Those are not only, there's, as I say, innate immunity. Those are not only directed to microbes, but they can also cause tissue damage. So one learning is that neutrophils are deadly. They have to be recruited. We have kept them in the barrack like we do for Black Hat Commando. Unless there is a real emergency, we don't deploy them. And second, that neutrophils do not discriminate when they act. Their action might cause harm to body cells, tissue cells also. And that's a very important point, which will come us to the next part of this. So this is, you see, the response, expression of pro-inflammatory cytokines, thereby must be transient. And pathways controlling them must be dynamic. You see that you have a pathway that must go up, and then there must be post-induction termination of the pathway, very controlled. Otherwise, you will have too many of those pro-inflammatory cytokines and chemokines, and too many of the neutrophils coming into the anatomic niche where infection has occurred. And this will cause damage to the body. So this is the very basic of bringing back to computational model, because of this very notion that the very heart of the immune system, there is a requirement that it must evolve better dynamical control. And, you know, I think this is not going to be, and this is the signaling pathways that transmit signal from peripheral receptors that are present in the tissue resident cells to the nucleus. And that signaling pathway must be dynamically controlled, which controls the expression of cytokines and chemokines. And then, obviously, how signals are transduced from pathogen recognition receptor to the nucleus, and how this signaling is dynamically controlled. That's something we will study in a minute. But question is that what exactly we do study in cell. I mean, this is of course a general question, and a lot of people have studied a lot of pathways that actually is involved in regulating expression of chemokines, cytokines, and people have, their own study, they have shown how dynamical control is mediated. Let's see, in our laboratory here in the National Institute of Immunology, Systems Immunology Laboratory, SIL, what do we study? So this is the concept that I'm going to, we have been nurturing it for over 10 years, 15 years, I guess, almost. And that's the key thesis, that's the key notion that we have been nurturing to claim a fundamental contribution to immunology research. Now you have signals, right? Pathogen 1 and pathogen 2, one can say, well, pathogen 1, for example, a virus would elicit different gene response than a microbe, let's say bacteria, right? So you can imagine that there is a pathway A for virus and there is a pathway B for bacteria, and that leads to response A and response B. So we used to, once upon a time, maybe many years back, many decades back, we used to think there are individual pathways and that control distinct cellular responses. The problem is there are not that many pathways. You have, as I mentioned, you have hundreds and thousands of different microbes. You don't have that many pathways. And then how do you achieve such an curated response that is curated for individual microbial entity? So this individual pathway-centric model began to challenge. And by last decade, I think 2005 to 10, the first decade of the century, was a very meaningful journey in this. People started doing mass spec analysis. They would use a protein and then pull it down and see what else interacts with him by mass spec. And you'll get a huge number of different proteins. You see that pathway A proteins, many of them interacts with the molecules that belongs to pathway B. That was confusing. You don't see insulated pathways anymore. And this sort of experiment, this is one of my favorite papers published in Molecular Cell in 2006. It's a, by the genome-wide shRNA. So you have begun to learn how to do small hairpin RNA-mediated gene knockdown. Once you learn, you begin to make a library for the entire genome. And this is one experiment where they are actually in the x-axis what you have, one or the other gene of the Drosophila genome that has been subjected to knockdown. Around 24,000 entities could be found in the x-axis. And y-axis what you measure is activity of MAP kinase, ERK in case, right? There are 3 MAP kinases. This is ERK. You measure average G score. To simplify it to my audience, if G score is 0, that shRNA did not impact MAP kinase activity upon knockdown of the target gene. And a non-zero value means this has substantial impact, right? I mean, if the knockdown led to increase in the G score, that means that this particular gene must be inhibitor of MAP kinase. And if the knockdown led to decrease in the G score, those might be stimulator of the MAP kinase activity. And I see this is the extreme left of the graph and the extreme right of the graph. They look pretty familiar. These molecules, these genes people later identified, they are all known regulator of MAP kinases. What was astonishing though, that is a continuum of this line, and it was later reproduced with many such shRNA screens. This is actually a typical what you get in a shRNA screen for whatever you measure. What does it say? It's very interesting that every gene in the Drosophila genome may have a non-zero contribution to MAP kinase activity. And that's very different than those A and B, 2 pathways doing things for us. It appears that entire genome is expressed and forming a network and controlling a particular phenotype, in this case MAP kinase. So sooner or later, we began to have these depictions that there are multiple pathways, they're interconnected. Sometimes they share molecules, sometimes they're connected to feedback loops, meaning one pathway is making some protein that actually is acting another path. Sometimes there is feedback, negative feedback regulation. And sometimes there are simply as simple as a functional connectivity, which is not physical in nature, but we know that there is a connectivity. So we slowly realized that pathways are interlinked through spatial and temporal relationships, and it's not a single pathway or network of pathways which integrates multiple signals, and that is responsible in complex cellular responses. And from then onward, we began to see the system biology. Of course, the term came up, and we began to see there are two components of the system biology. I'm going to keep this again. One, that is omics-based system biology. You have heard of it, transcriptomics. I'll talk just 2 slides today on transcriptomics. Genomics. These are called top-down system biology. You, of course, understand the same thing in system biology. It's a network of pathways. What you do, you take the genomoid readout. And from there, you put forward a hypothesis about the network, how it might be working. And another one started working at the same time, the end of the last decade or middle of the first decade of the century, the bottom-up system biology that began to get nurtured. That I understand about pathway, I understand about pathway, I know how they are interconnected, and I do see, I aspire to test if this interconnectedness changes the way pathway works or pathway B functions, and thereby pathway A and B together can form a subnetwork, and I begin to understand the property of the subnetwork. When I'm done, I add another pathway, that is pathway C in this process, right? And now, going back to our microbial response, we have done enough of ABC. So this is your pathway. This is S1 is nothing but a microbial signal, and let's say this L1, I mean, we will get into this L1 together just in 2 slides from now. That's an instructive pathway, signaling pathway. Remember, I was talking about how tissue resident cells receive signals through TLR, and there has to be intercellular signaling pathway that allows them to secrete chemokines and cytokines. Let's call that is this magenta color pathway is the one that directs inflammatory response, that provides for expression of chemokines and cytokines. And you see these cartoons here, the dynamical activity of this magenta pathway. That is the instructive pathogen responsive pathway. For pathogen 1, there is one dynamic activity. Pathogen 2, there could be a bit different. And then you begin to understand that there are these green pathways around. Unlike cells cultured in a petri dish, in our body, cells are sitting in an anatomic niche. That tissue resident cell that I was alluding to, they are in a tissue. They are not only experiencing signals from pathogen, they are also experiencing tonic signals that are present within a tissue microenvironment. That tonic signals would also activate pathways. Let's call it green pathway, right? And what you see, that green pathway, of course, activates certain other factors. We will not have to get into it. And activate certain genes. That's a Venn diagram I'm trying to do. GRM is gene regulatory module, right? There is a magenta genes and there are red genes. And then, of course, all these mass spec studies, genomic studies, and many other bottom-up system biology approaches that are very biochemistry driven led us to realize these pathways are intimately interlinked. Magenta and green, they're interlinked physically and functionally. The question becomes, that is what we have been studying, that these ancillary pathways that are activated in a tissue microenvironment because of microenvironmental derived tonic cues, do they alter inflammatory response elicited by the instructive pathway? What we do now, we add computational biology, cellular biochemistry, experimental model to understand how signal processing by an integrated network, or in other words, how crosstalk between cellular pathways tunes immune responses. Now, what is the specific question? Oh, okay. Give me a second. I think the PowerPoint collapsed. Yeah, it came back. Okay, any questions while I'm setting up the PowerPoint, right? Any questions, anyone? That's a good time. Good. So now going beyond this pathway B, let's get into something that is real biology. this magenta pathway I was talking about, the instructive pathway, that must be activated in tissue resident cells to induce expression of chemokines and cytokines. That is nothing but the NF-kappa B pathway. This is called canonical NF-kappa B pathway. What you have here is these TLRs. These are called toll-like receptors. And you have at the heart of it this transcription factor. It is relay P50 NF-kappa B transcription factor. Relay and P50, these are the two subunits that forms this transcription factor. In resting cell, this transcription factor is in the cytoplasm held inactive by this inhibitory protein I-kappa B alpha. So they're stuck in the cytoplasm. When this tissue resident macrophages, for example, would experience TLR signaling, that would activate a kinase complex called Nemo-IKK kinase complex. So it is called a canonical NF-kappa B pathway activating kinases, or you can call magenta pathway activating kinases. These would phosphorylate I-kappa B, leading to its proteasomal degradation via ubiquitin pathway. That would liberate this bound relay 50, which will now go into the nucleus and mediates expression of dozens of chemokines and cytokines. And that would drag affected immune cells at the site of infection. That would ensure limiting microbial infection and microbial eradication. Interestingly, this I-kappa B, this inhibitor of NF-kappa B, relay P50 NF-kappa B dimer, that mediates expression of chemokine and cytokine, and that is held inactive in the cytoplasm by this inhibitor I-kappa B protein. Interestingly, I-kappa B protein is encoded by this gene NF-kappa B IA, which is in fact an NF-kappa B target gene. So you get a negative feedback in the path. NF-kappa B is activated. It is mediating expression of chemokine and cytokine. Follow my cursor. There is this curve here. It is mediating expression of chemokines and cytokines, and then it is also inducing expression of its own inhibitor. So it will lead to post-induction attenuation of this NF-kappa B activity. So this is the negative feedback. That's at the very heart of the immune system. Intercellularly, in its molecular architecture, you find a dynamical code that instructs dynamically controlled inflammatory response, and lack of which can lead to tissue pathology and destruction site. So I'm going to, this is just for some students. I think what we do to do this nuclear NF-kappa B measurement, I'm going to really pass it a bit quickly. What we do an assay called EMSA. So if your NF-kappa B is present in the probe, we actually incubate nuclear extracts with a DNA probe that has kappa B size. If radio level DNA probe, if the nuclear extract has NF-kappa B in it, what will happen? That DNA protein complex will migrate slowly. This is called electrophoretic mobility sequence. If there is no NF-kappa B in your extract, the DNA probe will migrate faster and come at the bottom of the gel. If there is DNA probe in your nuclear, if there is nuclear extract as your NF-kappa B, then if you incubate the nuclear extract with radio-labeled DNA containing the NF-kappa B binding site, NF-kappa B will bind to radio-labeled DNA, causing the DNA to migrate slowly. And this is called electrophoretic mobility soft assay. And you can now do a radiogram and determine that there is a shift. That means that NF-kappa B is there. So this is called NF-kappa B EMSA. And I'm showing you the NF-kappa B EMSA that we have gotten using, I mean, of course, not the full gel. I'm showing you the crop gel from the top. What we have done here, we treated cells. These are called mouse embryonic fibroblasts. We treated them with LPS and collected nuclear extracts in a time course. We harvested cells and collected nuclear extract. And what you see, and then those nuclear extracts were analyzed for the nuclear NF-kappa B DNA binding activity by EMSA. And what you see is that there is a time-dependent activation, 0.51. You see there is an NF-kappa B nuclear activity that falls down a bit, and there is a next peak that progressively deteriorates up till 24 hours. So you see the biphasic NF-kappa B response coming up, little falling down, and there is a progressively deteriorating second phase. And this is owing to the negative feedback, right? And now, and again, I-kappa B alpha mediated negative feedback is very important for the stringent control. And you know, if you lose this control, there are many other mediators in addition to I-kappa B, I'm not mentioning them today, that ensures this dynamical control. When you lose them, you will have NF-kappa B. Look at this red line. You have NF-kappa B, that's not falling down, that's staying there. That's disease. That's uncontrolled relay activation. That would lead to so much of expression of chemokines and cytokines. You will have too much of neutrophils and they will begin to destroy your tissue. You will have disease, inflammatory diseases. You have heard of a few of them. One is inflammatory bowel disease. Exactly what happens. You have a very high NF-kappa B activity. You cannot be controlling it, right? On the other hand, you think of it, if you induce too little NF-kappa B activity, you won't have enough chemokine cytokine to drag neutrophils at the site of infection or any effector cells for that matter. Pathogens will win. So you'll begin to have vulnerability to microbial infections. You'll be immunocompromised. So that is the very essence of this magenta pathway. What we are calling them is a canonical NF-kappa B pathway is the transcription factor relay P50. And we are saying now that this dynamical activity of transcription factor relay 50, there is one, of course, I-kappa B alpha. Is there more? That is, of course, an important question. And we are saying that this dynamical control must be there, otherwise you will have disease. And then we were also discussing, remember, the ancillary pathways. In a tissue, you will have a lot of different cells, a lot of different cues, a lot of different tonic signals, and they are there. They're impinging upon tissue resident cells. While that tissue resident cell is exposed to microbial challenges, for example, in macrophages hitting in a tissue and it is staring at a microbial infection, bacterial infection, but that macrophage in this tissue anatomic niche is also experiencing signals from neighboring cells via a variety of tonic cytokines that host secretes to maintain tissue homeostasis. And that activates the green pathway, the ancillary pathway. And this is another pathway. Within the NF-kappa B system, we call this non-canonical NF-kappa B pathway. That's an ancillary pathway, green pathway. What you have in this pathway is this molecule called PNO100. This is a protein. It's called an NF-kappa B precursor protein. It is expressed by this gene called NF-kappa B2. And it sequesters this is an alternate NF-kappa B subunit, rel B, in the cytoplasm. Non-canonical pathway is activated by select set of host-derived ligands that activates, for example, lymphotoxin beta receptor. Lymphotoxin beta receptor is important for lymph node biogenesis during early embryonic age. It is also important for maintaining lymph node architectures in adult. The secondary lymph nodes that are important for orchestrating adaptive immunity. But lymphotoxin beta receptor is actually also present in a variety of different tissues, not only lymph nodes. We do not know what they're doing. What we know, it activates the non-canonical pathway. Here you have a kinase called NIC-IKK1. It's a slow-acting kinase. It activates very slow in response to lymphotoxin beta receptor. That's a non-canonical pathway activating kinase, which will again phosphorylate this P100 protein that leads to partial proteolysis of P100 into P52. P100 is the precursor. P52 is a mature NF-kappa B subunit of the alternate pathway or the non-canonical pathway. That will make rel B P52 dimer, which will translocate into the nucleus. P100 C terminus is I-kappa B-like. P100 N terminus is NF-kappa B-like. So when this entire protein is present, it sequesters rel B, and because of its C-terminal I-kappa B-like domain, it sequesters rel B in the cytoplasm. Non-canonical signaling leads to cleavage of the C-terminal, removal of the C-terminal domain from P100. That not only produces the mature P52 subunit, but also removes the inhibitory domain. Now rel B 52 is ready to go into the nucleus where they mediates expression of plethora of lymphokines and immune differentiating genes. rel B 52, it is not involved in inflammation. It mediates expression of immune differentiation genes. And what it does, this pathway signaling, the dynamic is a bit slow acting. As I said, it comes about a little late, but very persistent and a very low amplitude. Low amplitude, but persistent activity is achieved in this pathway, right? So you have these two pathways. And of course, what I just mentioned in the beginning, we have done, people have done, a lot of people have worked on it. They have found out there are a lot of molecular processes these processes connect these two paths within the NF-kappa-B system. There is the canonical NF-kappa-B that is involved for inflammatory response, the non-canonical NF-kappa-B that is probably involved for immune differentiation. However, non-canonical NF-kappa-B signaling is present in a variety of different cells, and now we know these pathways are connected. So in this laboratory, what we have been asking is, because of this interconnectedness, maybe non-canonical signaling modulates inflammatory immune response mediated by relay. So that is what the genesis is, that there are two pathways, they are interlinked, they might be doing crosstalk with each other, and that would impact inflammatory relay activity, and thereby that would have a tuning effect on inflammation. The question is, how do you probe crosstalk between cellular pathways? And why would you even imagine such crosstalk is happening? One interesting clue is this tissue intrinsic signals. I was mentioning about this microenvironment. Indeed, that microenvironment, a lot of signals come that may activate lymphotoxin beta receptor or another receptor to activate non-canonical pathways. So our proposal is that within a microenvironment, non-canonical signaling is activated in the resident cell, and that tunes inflammatory response that the resident macrophage would elicit or resident cell would elicit, and that has ramification for overall immunity. So that's the knowledge. My question is that there are two pathways, 1 and 2, canonical, non-canonical, right? How do you study crosstalk between pathways? Well, you take cells and treat them with ligand for both the pathways and see if there is an alteration in the cellular response. As simple as that, but it is not as simple as that. And that is where the computations are coming in. I'm going to take a couple of slides to clearly outline why we did what we did and combined computational studies with our experimental research. Although we had a very solid theory that there are those two pathways, they are cross-talking with each other, and this might lead to changes in the way these cells and body respond. But to understand crosstalk, we had to bring in computational model. So why, when you say that two pathways are cross-talking, you give ligand 1 for pathway 1, ligand 2 for pathway 2. The question is, how do you give them? Will they be concomitant or will they be sequential? If they're sequential, which one should come first? And what would be the separation between two sequential signals? So that's one problem. I mean, even in a culture cell, if I want to do that, this is a genuine problem. And I do not know, or no one would be able to so easily gather, how crosstalk would happen in the body. All possibilities needs to be looked into to derive conclusions about what would be happening in the physiological settings. And that's the first question. We're talking about a huge different combinations, simply how to add two ligands. And now I'm adding another more problem. Dose. How will you dose them? Equal ligand strength? Or one would be high? Or one would be low? Or maybe the one would be in different pulses. Cytokines are short-lived. In the body, they sometimes give this kind of pulsatile signal to resident cells. Cytokines that are derived by tissue residences. They also give an autocrine signal, but that can be pulsatile. Lots of theories are there, and you won't be able to just simply brush them aside and say, okay, this is the way to do it. You consider those and figure out in one of those conditions, do you get a crosstalk or not? There's a limitless possibilities that now one has to do in cell culture settings to simply understand if the core pathways are cross-talking with each other. And of course, the emergent properties of the system. I'm sure a lot of people have talked about it here. I'm going to give two slides here. What is emergent properties? Non-linear properties. What is emerging out of the system? Not intuitive, non-intuitive, right? I'm going to give you an example, which is very nice here. You have glass, you have tungsten, you have inert glass, you have metal cap. What do you make of it? Anyone? Any answer? If you see these four things, can you come up with any word?
No, what was the question?
So,
what is the odd one out?
No, yeah. If you think of these four components, what do you think as a sum of them? Any object that could be a sum of it?
Inert keeps coming back.
So, a sum of it would be I can give you some clue, right? Let's go forward. Even that is intuitive. The sum is intuitive. An electric bulb can be considered, right? If you take a little bit piece of it, you can make an electric bulb, right? That's not emergent property. That's additive. Now, you give an input, electricity, you get light out of it. That's emergent. Looking at these 4 pieces of material, no one would imagine this will light a bulb. You will get light out of it, right? So that's an emergent property. That's a non-additive property where it is more than the sum, provided a right input is given. So that's another issue with the crosstalk. Crosstalk is an emergent property where you have interlinked system that will generate nonlinear emergent properties. And you do not know what to exactly record for your experiments. And then another issue is, of course, one problem with that is if it is non-emergent, then effect can be early and late. You see signal 1 and signal 2, if you combine these 2 signals, signal 1 gives a response like this, signal 2 gives a late acting response. If you combine signal 1 and signal 2, there could be an additive response. But there could be a lot of crosstalk response, right? They are non-additive in nature. Look into the other examples, right? Now, these are, property of emergent system. You won't be able to speculate so easily. And not only that, it creates a lot more problem for you. If you measure early, the top and the bottom example, you will not conclude there has been a crosstalk. Look at the first shaded bar here. If you go back to the late phase, very late, you will also not conclude necessarily that there has been crosstalk. Experimental measurements are done in time points. If you have taken the first time point or third time point, I would hardly make a conclusion there has been a very good crosstalk between signal and 2, leading to emergent behavior. But if I take the second time point in the gray shaded area, you will clearly see that the activity is a lot more when signal 1 and 2 is combined, right? Versus the individual activities in the middle phase. So what is it that you will be measuring? When exactly will you do the recording of your experimental measurement? That becomes challenging. We're just simply adding 2 ligands. We have no idea what to expect and when to measure them. And the mechanism, and that's a very nasty slide. I was talking about NF-kappa. I definitely have been a bit overwhelming about talking about this canonical pathway and non-canonical, how Rayleigh 50 versus Rayleigh 52 dimers are formed in the nucleus. Now we're talking about a few, at least a dozen, honestly, of the pathways that are involved in pathogen response. Look what it looks like. And if you see a system and cell behaving differently when ligand 1, 2 are both added, you would not know what mechanism is probably working there. And to a possible way out has been the computational biology. What you do, you can actually construct a mathematical model. I'm going to discuss how it can be. To describe pathway 1, you can construct a mathematical model to describe pathway 2, and then you actually can simulate pathway 1, simulate pathway 2, and I can then simulate combinatorial regimes. All different combinations that we were articulating. Sequential, simultaneous, one first, one second, all possible combinations to identify the regime that might give crosstalk, and then you can substantiate those predictions in experimental studies. If the predictions are not matching up in experiment, of course, the model can further be refined, and this can be an iterative loop. And this is a picture, right? I mean, Apollo missions, you must have seen this picture, right? It's a historic day for mankind. You must have not seen this picture. It's a lunar module simulator at the NASA facility, lunar landing research facility. They were simulating thousands times the lunar landing before we actually landed on the moon. So how about this one? This is the important way of thinking about it. You can simulate conditions and then get to a point where you know enough about the system to gather adequate predictive capabilities. And I'm going to move it out from this and going to directly to the NF-kappa model that we have built. So this is the NF-kappa model we have built. Remember, I described you about this canonical pathway, the molecular processes, association, dissociation, synthesis, degradation, nuclear translocation, gene activation in response to IKK kinase activation. That's the canonical path. The major dimer is really P50. And there is this non-canonical path where in response to NIC, RLB52 is known to be activated. Now, what we did, we constructed a mathematical model what these molecular processes has been described in equations, mass action kinetics equations. We got around 37 species, hundreds of parameters, and we did a lot of parameter estimations. I'm going to show you how these models can be parameterized. For example, a degradation rate of, for example, rate of change in the NF capability mRNA level. Remember that NF capability gene encodes this protein called P100, which is a precursor protein, and upon non-canonical signaling, it becomes P52, which in association with RLB forms RLB52 dimer and actually allows for expression of immuno differentiation genes. Now, question is, in the model, you have a rate constant that is required to be fitted to describe the change in the NF-κ2 mRNA level. So, what is the change in the NF-κ2 mRNA level? You can probably say the change would be proportional to or equal to constitutive synthesis rate, which is irrespective of anything but inducible synthesis rate. NF-κ2, just like I-kappa-B alpha is encoded by NF-kappa-B IA gene, which is NF-kappa-B target. We knew NF-kappa-B2 is also NF-kappa-B relay P50 target. So you can have a inducible synthesis rate, you have a constitutive synthesis rate, and you have a degradation rate. And this is the equation that can describe change in the NF-κ2 mRNA. Now, question is, how about measuring, let's say, degradation rate constant? How will you do that? I mean, you have rate constant, you have to find out those values so that you can put in the model, and then you can start simulating by one after another equation. You get the rate constant, and you can now fit into the model and start simulating. How about you measure degradation rate constant? This is what we did. What we did, we used actinomycin D. Treated cells with actinomycin D, it blocks, it obstructs mRNA transcription, and then measure the NF-κ2 mRNA level over time. What is happening? In this cell, no mRNA is produced, only mRNA is degraded, and I'm measuring the level of this mRNA. Of course, after normalizing with actin, and then I put a zero-order kinetics for mRNA decay. That's an assumption. mRNA decay requires a lot of different involvement of proteins, but we, for the simplicity, model requires some degree of reductionist approach. We considered that less consideration half zero-order kinetics and fitted this equation, and then we got an mRNA half-life. From there, we can calculate the degradation rate, and that rate can be now fitted into the model, right? So this way, we have generated a huge amount of values from our own experiment literature. The field is nurturing for quite some time, 30 years almost. NF-κ2 fields, you got a lot of literature. You can survey the literature, look into the data, and from there you can get an idea about the rate constant. And also modeling requires fitting, although there will be some stringently fitted, determined rate constant. There will be some that are in the gray area, and you can fit them and see if the model behaves properly, and then change the fitting, right? So this is our model for an input of the model. Remember, the electricity bulb require an electricity to get the light. That's the emergent property. Here, the inputs of the kinase activity. You can measure the kinase activities. We can treat cells with, let's say, TNF, and measure the IKK kinase activity, harvest cells in a time course, and measure the IKK activity. Treat cells with ligand that can activate lymphotoxin beta receptor, that would activate non-canonical pathway. And now you can measure the NIC-IKK1 activity, right? So you can measure the activity of the activating kinases of the canonical and non-canonical pathways respectively. So what you can now do, you can input, you can use the IKK that is experimentally derived, plug it in the model through the IKK arm, and simulate the model. You have all the parameters are given, simulate the model, and simulate nuclear NF-κ2 activity. We have done what we did. We treated cells with TNF, measured IKK activity, used the IKK activity, plugged it in the model, simulated TNF-induced NF-κ2 activity. And we also performed experiment by EMSA, remember, electrophoretic mobility shift assay. We performed experiment to derive nuclear NF-κ2 DNA binding activity and compare them. And that's an iterative process. You can continue to do this better fitting for better fitting. So those are the parameters where you probably have to keep refining to get a better fit. So arrived onto a parameter space where you could get a very good fit for TNF-induced NF-κ2 response and LT-beta-induced NF-κ2 response as well. right? And LT beta, this NIC kinase is a very boring. It comes late and stays there forever, and a very weak NF-kappa B response. This is not a relay, but very weak relay response is also observed, and this is modeled here. It's only relay activity has been modeled here. So we got a model, we asked, and I'm going to show you an example where model has helped us. And then there are 2 or 3 more examples. Yes, I will take the question. Yes.
What you just mentioned, was that RELU? What activity you just said?
Rel A P65, Rel A P65. Yes, that's the major transcription factor that drives the expression of inflammatory gene.
Okay, thank you.
And now we use this model. Remember, the main issue was that when this probably will crosstalk with each other, there are numerous possibilities, and which one to pick? I mean, how will you direct your experimentalist? And this is the way we did it. At least this is one of the way to do it. We did this, this is what you see here, a typical kinase activity profile. You have an amplitude, you have an onset time, that is A, and you have a fall time, that is B. So A plus B is the duration of the kinase activity, H is the amplitude. It's a more of a typical kinase activity profile that would be probably true for any pathways, right? Now, what we did, we keep changing the value of H and A plus. Sooner we got a library of around 600 or 700 profiles. These members of this library, each of them is actually a specific kinase activity profile. And members differ from each other with respect to the amplitude of the kinase activity and the duration of the kinase activity. In a way, that's all possible kinase activity profiles that can be there has been taken in this theoretical library. Now what we are doing, we are plugging in the library, of course individual with each member, through the NIMO-IKK, the magenta, the instructed, the canonical pathway, or through the non-canonical NIC-IKK, the green, the ancillary arm, or both. So NIMO is magenta, canonical, NIC green, non-canonical, NIMO plus NIC together. When I am plugging in both, we are calling it black. We are putting a black color here, right? And we are doing here, at least in this case, we just narrowly focus on the simultaneous activation when you are doing both. And downstream of this, we simulated NF-kappa B activity and determined crosstalk index. Crosstalk index is that the NF-kappa B activity that you get when both the arms are engaged in relation to the one you get by adding NF-kappa B activities going to individual engagements. So NF-kappa B NIMO plus NIM means when both the pathways are engaged, what NF-kappa B you get when both the signals are coming versus individual signal derived NF-kappa B activities. If it is more than one or less than one, that means there is some degree of crosstalk and you are getting non-additive NF-kappa B response, right? And from there, we did a huge, of course, this is 600 or 700 kinase activities, a huge set of simulations were done. And we started saying, give me crosstalk proficient profiles, top 10%. And of course, some profiles, they engage to mnemonic, but that combination of mnemonic never gave crosstalk more than what you would expect additively. And some combinations gave crosstalk more than what you would expect coordinatively. I wanted to know among those crosstalk proficients, what the top 10% and what do they look like? And this is what they do like. Each spot is a profile characterized by their peak duration and peak amplitude. Interesting thing is if you see here, I think the this thing is gone. If you see here, what you see here is this area. NIMO-IKK and NIC-IKK, when you see, there is a duration control. For a varied peak amplitude, they got engaged into crosstalk. These are all crosstalk proficient profiles. There is no such thing. Then for a variety of different amplitude, they are engaging in crosstalk. But they needed a critical threshold. For NIC-IKK, when you see, only crosstalk proficient profiles appear after 8 to 10 hours. But NIC-IKK1 is Non-canonical pathway is slow acting. I mean, nobody cares about it. For pneumo-IKK2 also, there was an interesting thing. We see that only after 2 hours, these crosstalk proficient profiles begin to appear. But then it's a very monotonous. Once they appear, they're always there. But first 2 hours, there is no crosstalk proficient profile. Why was it so interesting? Because pneumo-IKK is activated in inflammatory regime in response to microbial ligand LPS, where you get pneumo-IKK activity that persists beyond 2 hours, long lasting. But cytokines also activate this inflammatory relay pathway, relay NF-kappa B pathway, this relay NF-kappa. But cytokines typically induce this relay NF-kappa B very transient, not going beyond 2 hours. So indeed, we knew that there is a disparate kinetic control of pneumo-IKK2 by inflammatory mediators. Cytokines only activate transiently, and microbial ligands activated more persistently. And our results suggested that short-lasting pneumo-IKK2 that are produced by cytokines, those would not engage in crosstalk with the non-canonical pathway at the level of downstream NF-kappa B activity. But long-lasting pneumo-IKK2, for example, that is activated by microbial ligand, will conveniently engage in the crosstalk with the non-canonical pathway if you also activate them, and produce synergistic NF-kappa B activity at the level of downstream NF-kappa B response. And this is the simulations actually. We took IL-1 kinase activity, we took LPS kinase activity and simulated NF-kappa B response. What you see here is this. I think the red color is LPS-induced NF-kappa B activity, early peak, progressively deteriorating next phase. Of course, clearly matching experimental data. Green is when you treat cells with the non-canonical ligand alone. A little bit activity, very slow, but very low amplitude. You put them together, model says there will be a lot of crosstalk. Look at the black line. When you put them together, particularly the late phase would be augmented. But with IL-1, with respect to the IL-1 alone, that is the red versus IL-1 along with non-canonical signaling, there was hardly any difference, suggesting that the model not only directed us to understand crosstalk, directed us to a very interesting prediction that crosstalk between canonical and non-canonical pathway would alter nuclear dynamics of the relay NF-kappa B transcription factor, but the crosstalk would be stimulus selective. The crosstalk would happen, non-canonical signal would amplify, augment inflammatory relay response generated by microbial ligands, but not generated by cytokines. And this is the experimental data. I'm showing you the IMSA that I have just discussed around 45 minutes back. IL-1 response, because the kinase activity is very transient, you see very transient NF-kappa B response in the nucleus. Lymphotoxin beta receptor non-canonical pathway, very weak NF-kappa B response. Together, you don't see much improvement. LPS, again, that IMSA I have shown you. Alone, you see an early peak, fall, and a second phase, which is progressively deteriorating. LT beta, very weak NF-kappa B response. Put them together, crosstalk happens. The early peak is not altered, but the second phase is augmented. So, non-canonical signaling promotes or sustains LPS-induced relay NF-kappa B activity. That is what we gather. And once you have model, this is, I'm not going to go into the detail, but once you have model, you can do things. You have all the parameters in the model. Then you, of course, ask, why is the crosstalk happening? When your model could predict that crosstalk would happen, that's a very good beginning, because I have all the parameters in the model. I can perturb them one at a time. This is called parameter perturbation analysis. To determine which one of them would affect, would impact crosstalk. So here you are generating a crosstalk sensitivity score. Higher the score, meaning more the perturbation of the parameter produces more effect on crosstalk, right? And the top one was K9. That is NF-kappa B2, NF-kappa B induced NF-kappa B2 transcription. We started parameter perturbation, and this NF-kappa B induced NF-kappa B2 transcription came up as an important parameter that determines molecular crosstalk between canonical and non-canonical signaling. And NF capability is the one that encodes the gene P100, which is thought to be important for RailB P52 regulation in the non-canonical pathway. Here we are rediscovering NF capability as an important determinant of relay-driven inflammatory activity. We actually, this is what model did. It pushed up to this experiment. We did the experiment. We used NF capability to knock out cell, and look what happened. This late NF-kappa activity that was amplified because of non-canonical signaling, which is written here is B is green, non-canonical signaling alone, LT-beta receptor alone, L is LPS, LB when you are stimulating cells both with LPS and the non-canonical ligand. You see here, the late NF-kappa activity that is induced by LPS at 24 hours was not augmented much when you took NF-kappa to knockout. And then we even made a cell line. We used NF-kappa-B2 knockout to put back NF-kappa-B2 gene into it, but from an NF-kappa-B insensitive promoter. That's a constitutive promoter, because our model parameter sensitivity analysis indicated that NF-kappa-B induced transcription is important. So we made a cell line where NF-kappa-B gene is no longer induced by NF-kappa-B2. It is constitutively expressed. Again, the crosstalk is gone. You see the early activity in the canonical regime falling down at 24. When you co-stimulate, the late activity is not induced in a constitutively expressed cells. But when you co-stimulate, when you do the NF-kappa gene put back in NF-kappa knockout from an inducible NF-kappa responsive promoter, you see the late activity is augmented, all right? So this is what it is. Now summarizing what we discovered, that you get signal to cell, relay 50 is activated, that induce I-kappa-B alpha production, that is a negative feedback, right? But you also make a lot of P100 NF-kappa-B2, which normally would make LB52. But if you have a concomitant canonical, non-canonical signal, you generate copious amount of P100 and thereby P52, a part of which then forms an alternate dimer called relay 52. So it's a positive feedback. You are making P100 and NF-kappa-B2, that became NF-kappa-B itself in the form of a relay 52 dimer, which has exactly same function as relay 50 with respect to inflammatory chemokine cytokine expression. So it generates a positive feedback when two pathways are cross-talking with each other. That sustains LPS-induced activity, right? Now, I'm going to take 5 minutes to express you something about the gene expression. And that's about the top-down system biology that I was alluding to. So LPS, this is LPS-induced relay activity by nuclear EMSA maybe, and this is LPS-induced gene expression. Let's say this is an RNA-seq analysis, right? Genes are up, magenta, genes are down, green. More green means very down, and more magenta means very high up. LPS-induced these genes. My question is, how do you determine which LPS response genes are regulated by relay? What will you do? If I ask that LPS-induced around 900 genes, I want to know how many of them are induced because of NF-kappa-B relay. What experiment will you do? Anyone?
Maybe try knocking down or something?
Yes. So this is exactly what you do, right? You will take a relay knockout cell and treat them with LPS. You see this first cluster of genes, they are not induced in relay knockout. They are definitely relay target genes. Second cluster genes are also somewhat affected, right? They are also relay target genes. The third cluster, although they are induced by LPS, they are also induced in relay knockout cell. So they are relay independent, but LPS induced. That's a very easy thing to do. But this becomes an interesting question for us. Crosstalk responsive genes. How do you determine if a change in the relay activity that I was showing by EMSA, that was signaling studies. Now we are asking the question that we see that, of course, LPS gives this kind of relay profile. If you do LPS plus non-canonical pathway, you get the black profile. So there is an increase in the nuclear relay activity, right? And you see, this is the gene expression data. We have done this, RNA-seq. You will see these are LPS response genes. Now, when I do L plus B, meaning L and B, meaning crosstalk regime, you see some genes are even more induced. Now, question is, how do you determine that they are related because of, how do you determine this difference in relay activity indeed is causing these gene differences? Going back to the answer that you gave, if I used relay knockout, remember, both LPS response genes will come to the basal level and LPS LT-beta response genes will come to the basal level. These two play along. If you do relay knockout, the activity will no longer be there and gene expression would look like exactly basal. I will not
you won't be able to delineate the crosstalk.
Yes. See this L and LB, the second column and third column, there is an increase in gene expression. I would never know, was it because of this increase in relay activity for something else? Because the very basis they were induced, they are no longer induced in L alone. So I would not know if their increment was because of more relay or because of another pathway that I am not examining today, right? That the star means the crosstalk sensitive culture. I know this is a crosstalk sensitive cluster. I also know these genes are induced by relay, but I do not know if their super expression or hyper expression is because of increased relay or for another thing. Because in knockout, all of them are gone. So that's what we do. We find out these by an analysis called gene set enrichment analysis. That's a classical example of when you can use top-down to derive conclusions, meaningful conclusions. What we did, we found out a list of LPS responsive genes and then find out their crosstalk score, meaning their expression in the co-treatment regime vis-a-vis their expression in the sum of their expression in the individual regime. And we also collected a predetermined list of bona fide NF-kappa B relay target genes. Someone has determined it. You know, you can use LPS response in relay knockout to determine what are the bona fide relay target genes, but there will be many that are not direct relay target. Then you can look into the promoter, find out if there is an NF-kappa B binding site. Then you look into relay chip-seq data, combine all of these to screen out and generate a bona fide list of NF-kappa B direct targets. So we had a list. Someone gave it to us, 290 genes. And we actually rank the genes in the crosstalk, right? This is what we are doing now. This is called gene set enrichment analysis. What you have, here you see these are 900 genes. We have ranked them based on their crosstalk score. Here, genes are very much hyperactivated during crosstalk. Here, genes are reduced their expression during crosstalk as compared to individual cell regimes. And we have passed this 290 gene list, predetermined list, in an enrichment analysis. And this each line, these hits meaning they are NF-kappa B targets. So there is a match between this 290 list and there is a list of 900 genes that you have experimentally determined. These are crosstalk sensitive, hyperactivated. These are crosstalk sensitive, downregulated. And these are NF-kappa B targets. You see all the targets are on the left side when they're upregulated. And you can generate an enrichment score that is coming pretty high, 0.4, suggesting that genes that are hyperactivated upon crosstalk are likely to be NF-kappa B target with a statistically significant enrichment score. That's called gene set enrichment analysis. That in a global scale with hundreds of genes and in a global scale provides for a very authentic conclusion that NF-kappa B crosstalk that leads to hyperactive LPS, hyperactive NF-kappa B response as compared to LPS or LT beta response alone is meaningfully converted into heightened gene expression, relay target gene expression that are inflammatory genes. And once you know that, this is what we did finally. And we are doing a lot of these experiments these days. This laboratory, this observation, non-canonical signaling amplifies canonical response. Non-canonical signaling amplifies inflammatory relay activity. Has been core of this laboratory for the last couple of years. Because we then looked into my animal model. This was the first paper we published, and we published many others I'm not going to talk to. But we infected mice with Citrobacter rodentium. It's a gut pathogen, collected the epithelial cell, gut cells, and we see a lot of NF-kappa B activity. You take NF-kappa B to gene out, NF-kappa B is normally thought to be a regulator of immune differentiation. But you see, if you take NF-kappa B to gene out, this inflammatory relay activity is so timid in the colon, they do not express enough chemokine and cytokines. We have measured this leads to a defect in the microbial management. You have a lot of bacteria in the colon and it leads to systemic dissemination. You will see bacteria in liver, lung, everywhere, and those mice will succumb. That was a major discovery showing not only discovering crosstalk between pathways, stimulus selective crosstalk between pathways, figuring out the mechanism that may link them, for example, NF-kappa B2, but to show it has physiological relevance with respect to gut immunity. And since then, we have shown its role in inflammatory pathologies in the bowel and so on and so forth. And I will show you 2 more 5-minute slides. And this is very, very crowded, right? At the crosstalks. We will come to that in a minute. Let's get here. we got this dilemma soon after this publication. We, of course, saw crosstalk is happening in, I mentioned these cell types, mouse embryonic fibroblasts, right? And we also saw that the crosstalk is important for gut cells, intestinal cells. And when it comes to inflammation, we discovered that crosstalk controls inflammatory NF-κB response, and macrophages are the important thing. They are very important for crosstalk. So we asked if macrophages, the crosstalk is happening. And we have repeated this data in primary macrophages as well. Unfortunately, the first few days we were doing it in cell lines, and that is what we published. Look into this data. L means signal 1, LPS, microbial ligand. B means signal 2, the tonic signal, lymphotoxin beta receptor signal, and LB means when both are circulating. You see LPS alone 0, 1, and 16 hour. Already NF-κB activity is falling out. B alone does not give too much of an activity anyway. If you put them together, you see this LB regime, the 16-hour activity is much higher. So you see the quantification below. L16 versus LB16, there is a statistically significant enhancement of the late activity when cells are co-stimulated, and this is from fibroblast, as obviously we have presented before. And in this macrophage cell line, you see B is working. Non-canonical signaling is working. It is inducing, albeit weakly, the NF-κB pathway. Be it in J774, be it in raw macrophage. What you see is not working. And L is of course working. You see L treatment, you see lot of NF-κB activation at 1 hour is falling down. B is working. Look into the J774 cell, the middle one. 0, 1, 16. LPS treatment, lot of NF-κB, it is falling down, not unexpected. B, yeah, you see some activation of the non-canonical pathway, albeit weakly, it's known to be weak. You combine them, you see that 1 hour treatment, of course, there is an NF-κB, but 16 hours, there was no enhancement of the late activity. True for also raw macrophages. The late activity was not enhanced despite co-stimulations of cell with the non-canonical pathway, despite showing that non-canonical signaling was working alone. One may speculate, well, you know, maybe raw macrophages of this myeloid cells, they may not have enough NF-κB to mRNA. Even if they do, maybe remember, NF-κB induced NF-κB to mRNA production was an important sensitive parameter in our experiment and model. We said that maybe macrophages, microbial signals would not induce, LPS would not induce NF-κB to mRNA expression. That was not true. We compared with fibroblast, we see almost equivalent NF-κB to mRNA induction in these macrophage cell lines. So what's going on? It seems like that pathway crosstalk is not only stimulus selective, but also cell type specific. We did something very interesting. It's a Monte Carlo based analysis. We did a parameter sensitive analysis, the one that I showed you where individually each parameters were changed. That's called a local sensitivity analysis that sometimes masks complexity of the system. This is a global analysis. We had too many parameters based on their co-regulated biological processes that they are participating into. We grouped them into smaller cohort. And we looked into the parameters that are supposed to be more subject to cell type specific variation, that are supposed to have more variation between macrophage and fibroblast. Remembering that our mathematical model and its all kinetic parameter values were derived based on experiments performed in fibroblast, we hypothesized in macrophage certain parameter values are different. If I can nail that, I will know why macrophage is not showing crosstalk. So we did this. We grouped the parameters into a smaller cohort and did this Monte Carlo sampling, where we actually, each cohort, we simultaneously took values from the nominal value, 25% up and 25% down, and with the and combined and expressed this effect of this parameter uncertainty on the NF-κB crosstalk in this total effect index. Another very interesting thing came out, that is group 6. That says NF-κB IA mRNA expression rate. NF-κB IA encodes IκB alpha. Our modeling studies indicated that in addition to NF-κB induced NF-κB to mRNA expression, NF-kappa B induced I-kappa B alpha mRNA expression could be also a very important determinant of NF-kappa B crosstalk. Indeed, our results shows that this I-kappa B alpha expression I-kappa B alpha was far more responsive to a given dose of LPS in macrophage than in fibroblast. That the chromatin, the I-kappa B alpha gene was poised for hyperactivation in macrophage cell line. Within 1 hour, you see a lot of I-kappa B alpha expression that was not normally seen in fibroblast. And we use these values, this macrophage specific parameter, particularly the I-kappa B alpha parameter. You see the model F. This is the model that we are showing for fibroblast. You see there's a high crosstalk, LPS induced early activity, falling down low activity, I-kappa B alpha synthesis, NF-kappa B to mRNA induction, and crosstalk. And when he started changing the values, particularly the values that are governing these two important parameters, NF-kappa B to expression and I-kappa B alpha expression, to those that were obtained using macrophage, we again see that same NF-kappa B activity we can recapitulate. We see that hyperactivation of I-kappa B alpha once we take I-kappa B alpha specific macrophage specific reaction constant rate parameters, and the crosstalk is gone. So again, pictorially on your left, it is shown very well. Fibroblast, the model, this is PF, a parameter ensemble derived from fibroblast. And we have seen key changes in the key parameters. One is NF-kappa B driven I-kappa B alpha synthesis rate. We use those parameters to arrive on macrophage specific parameter ensemble, PM, and the crosstalk is gone. You see the magenta and black line is coming close to each other. And this was very useful because I now know that I have a parameter ensemble in macrophage that can explain why macrophages are not causing crosstalk. Maybe the I-kappa B alpha synthesis rate. Indeed, that is what we found. To pictorially present what we found, that TLR or even in the singular regime or co-treatment regime, of course, the early delay 50 activation is happening. That gives an early I-kappa B alpha synthesis rate. But there is also a late relay 52 activation that happens, right? Because of NF-kappa B2 that would be induced by early NF-kappa B activity would be converted into relay 52 dimer, giving a positive feedback. That's a late activity. But in macrophage, the I-kappa B alpha promoter is so hypersensitive that would lead to a late phase I-kappa B alpha synthesis. That would not happen in fibroblast. In fibroblast, this late activity would conveniently locate into nucleus, giving a lot more late NF-kappa B response. In macrophage, this late activity would go into the nucleus, but will end up triggering this hyperactive alpha promoter, causing a late I-kappa B alpha synthesis that would take down the crosstalk. And indeed, when you actually make a parameter ensemble within macrophage, you remove relay 52 mediated regulation of I-kappa B, you see crosstalk is coming back. An experiment that can only be done computationally, cannot be done experimentally to make such a important mutations. It is not possible. So we see that the crosstalk that are stimulus selective, cell type selective, and that's the last slide that is called cell-cell heterogeneity. If you look into that cell in the petri dish, you treat them with LPS, you see NF-kappa B response, but this will show cell-cell heterogeneity. Not all cell will respond to similar extent. Some cell, this is a trajectory, this is a live cell imaging, and what you see in the color is NF-kappa B response. Some cells show very short early response, some cells show moderately long early response, and some cells show a very oscillatory second phase, and that is for a wide range of ligands. What we did in the meantime, and we looked into this issue using our model. The model is a kinetic model, it's a deterministic model. We introduced heterogeneity, we introduced noise in the parameters basing on our literature survey. And then when you use that noisy model, and these are, and to really, we could recapitulate this heterogeneity that is experimentally observed. This is the experimental data from other people. This is our own data. And this 0.3986 is the number, this is the central variance. So we have now arrived onto a model. This is a work that has not been published yet, with our deterministic model introducing noise in the cellular parameters. We could recapitulate experimentally observe cell heterogeneous NF-kappa B responses. And we did this very interesting study just to finish this. The magenta is, of course, signal 1, LPS. Green, LT beta. And LB is co-stimulation. And we are not measuring signal response. we are measuring log variance of response, meaning how much variations were there in a population of models that recapitulate a population of cells in a plate. LPS response, of course, there is a lot of variance. LT-beta response, lot of variance, very peculiar response. When you add them together, you see noise is falling down. Something that we are yet to fully understand, but what we think is that NF-kappa B crosstalk not only amplifies inflammatory response to microbial ligands, but also synergizes them for the benefit of a accelerated innate immunity that are important for eradicating bacteria. But I am showing you here why one should study immunology, and I am showing you that important mechanisms, cell signaling, pathway 1, pathway 2, they crosstalk. We are showing you that this crosstalk happens in stimulus and cell selective manner, and such crosstalk is indeed a way to integrate signals and tune immune response. You can amplify, you can dampen, you can insulate, you can cause heterogeneity or synergize heterogeneity and then give an amplified response. And we are proposing that such interlinked signaling pathways, such crosstalk regimes, you cannot, of course, target hub pathways. You cannot target NF-kappa B, you cannot target P53. They are central to our biological processes. Therapeutic targeting, although they are involved in disease, direct therapeutic targeting would cause devastating side effects. On the other hand, these cross-regulatory mechanisms appears to be stimulus and cell type specific, and thereby may provide for expanding therapeutic opportunities for inflammatory diseases. This is where I will close. I think I've been talking quite too long. And again, it would have not been possible without those collaborators, a lot of them. And indeed, the aspiration is to reach to this era of system medicine, where this bottom-up systemology, where you're integrating pathways, pathway 1, pathway 2, maybe tomorrow is another pathway we'll be integrating in our concept, in our mathematical model, in your cellular experimental design, with the hope that this might provide for, this insight might provide for disease-specific intervention, cell-specific intervention in human elements. And I will again thank you for your patience, and it was wonderful to share some of our key research findings, particularly that pertains to computational modeling and experiment combined approaches with you. Thank you very much. I'll take up questions if you have.
Thank you, Professor Somen. Any questions? Feel free to unmute and ask. So I'll ask a quick question. I see you are collaborating with somebody from AIMS. So I was just wondering, you mentioned this at the end of your talk. So have you, like, do you have a target pathology or disease in mind when you're looking at translational application of this, crosstalk work?
Yes, it's a good question. Actually, I did not, because we developed a molecular insight that pathway crosstalk may tune inflammatory response in the colon. And in the first few years, we established that this is important. If you do not amplify NF-kappa B response, you become susceptible to gut pathogen. That one, Citrobacter rhodesium, we studied. But in retrospect, we begin to think that such amplification loop that we have discovered that is achieved through crosstalk could also lead to inflammatory disease of the colon. And that we published a year back, I believe in a year or two, I do not remember, in PNAS. That was not involved any mathematical model. I mean, that sort of model was not involved in that study, but the insight was used. We used human IBD sample, inflammatory bowel disease sample. We found out engagement of the non-canonical pathway in human IBD. We used animal models where this crosstalk was disrupted specifically in epithelial cell. to discover that in the absence of such crosstalk, animals are far more resistant to inflammatory insults of the colon. For example, DSS, that's a cholitogenic insult. You can induce colitis, but if you do not have the crosstalk, you see that the animals are resistant. At this point, this is where we are. We are currently studying if such crosstalk in dendritic cell is also orchestrating colon inflammation. That work is in a pretty advanced stage. But involving multiple cell types and studying is giving us some kind of confidence that molecularly the crosstalk should be or could be targeted, something that we need to look into in the coming years, particularly for inflammatory bowel disease.
Thank you, sir. Any other questions? If not, I and all the participants would like to thank Professor Dr. Somen Basak for this enlightening talk. Thank you, sir. We really enjoyed it. And
thank you very much. It was a pleasure to be here, and wish you all the best.
Thank you. So after that, we have lined up our final, but certainly equally exciting talk of the day by Professor Ranjit Padinatiri from IIT Bombay. So the scheduled time is 3:45, and I see Professor Ranjit is on the call. So, sir, if maybe we can
wait for, maybe people would need a break. Do you think they need a break and start after 10 minutes? I'm okay either way.
Yeah. No. I was just saying we can do a quick check, audio check. We'll start at the scheduled time.
Sure. Yeah, I'm going to share my, if you let me share, I'll just share my screen.
Yeah, you should be able to.
Yeah.
So participants, take a 10-minute break. We'll start around 3:45.
Can you see this?
Yes, sir. Yeah, and can you go forward a few slides?
Yeah.
Yeah, perfect.
Okay, great. Okay, we'll wait for the time and then I'll be around.
Sure, thank you.
Yeah.
So welcome back, everyone. Welcome to the concluding talk of this two-day virtual workshop on computational and mathematical modeling of biological systems. We have an excellent speaker to conclude our two-day workshop. I'll introduce Professor Ranjit Padinateri from IIT Bombay. So Professor Ranjit is a professor in the Department of Biosciences and Bioengineering at IIT Bombay. He has research interests in understanding self-assembly in biology, focusing on nucleosome positioning and 3D organization of chromatin. Professor Ranjit uses theoretical and computational physics tools to understand biological systems. He studied physics at IIT Madras, where he got his MSc and PhD degrees, examining how semi-flexible polymers like DNA fold. He then went on to do postdoctoral research at Northwestern University, working with John Marco on chromatin. Later, he moved to Institut Curie in Paris to work in the group of Jean-François Joly, investigating the kinetics of filaments like actin and microtubules. Since 2009, he joined IIT Bombay, and currently he's the Dr. P.V. Sukhatme Chair Professor at IIT Bombay. Please take it away, sir.
Thank you. Thank you for the invitation to talk in this workshop. I'm going to talk about physics-based model to study chromatin organization. And I'll describe how chromatin is organized in space and time and how it is kind of broadly related to function. Can you all hear me? Is it clear?
Yes, sir. We can hear you fine. Participants, are you able to hear Professor Padinateri fine? Please do a thumbs up or yell out a quick yes. Can everybody hear Professor Ranjit? Please say yes,
yes,
unmute and say, okay.
Okay, thank you. Yeah, so good. So please feel free to stop me in between any time. This is this kind of a workshop. I plan to have it kind of an interactive session. So I will give a kind of detailed introduction on chromatin, how chromatin is organized, because a lot of new things have come up in this area, which has kind of even changed our understanding of chromatin itself. And a lot of things in the textbook a lot of information in the textbook is rewritten, and a lot of new ideas have emerged, how chromatin is organized and how is it related to function. So the work that I'm going to do, the part of the work that we did in the lab is done by some of these students, PhD students, whom we can see in that picture, which is the nice, beautiful Western Ghats when we went for a trek last year or so. So as you can see, the picture on the right-hand side is a chromatin, is a cartoon of chromatin. And we will see how to understand this and how is it related to function. Okay, so we all know that DNA is the molecule that codes cellular processes, right? All the living organisms have DNA, and DNA is a sequence of A, T, G, C organized in some way, and this sequence encodes some information. This is something that all of us know. Now, some of you might have heard that DNA is very long. If you take, for example, human DNA, if you stretch it out, it is like 2 meters long. And this 2 meter long DNA, which is like 23 pair of chromosomes, this whole 2 meter long polymer, so it's a kind of a set of 23 pair, 46 chromosomes, 46 polymers. These polymers have to be stored in a small nucleus, typically is like of the order of 10 micron size. So there is indeed a packaging problem, that how to package this very long polymer into a tiny space, which is 10 micron, which is somewhat like, for example, if you have a very thin wire, which is about 30 kilometers long, and you need to pack this 30 kilometer long wire into kind of a tennis ball. That's the kind of packaging that DNA is packaged into. So it's a 10,000-fold packaging. But this is not just a packaging problem. This is also an information management problem, but because we all know that DNA encodes information. So this information has to be managed in an interesting way for the function to happen. So, and let me explain that. We know that we all start from a single cell in our mother's womb, and then that's one cell divides and divides and divides, and an organism develops, right? So in that single cell, you have some DNA, 23 pairs of DNA, and this DNA is copied over and over again to form all these cells. But we know that if you take two different cells, for example, think of cells in our skin and cells in our eye, these two cells have exactly the same DNA. The DNA sequence in the cells in our eye is the same as the DNA sequence in the cells in our skin. But the skin cells function very differently from the eye cells, right? How do they function differently? The only difference is essentially the chromosomes, the chromatin, right? So the chromatin is very different, is differently folded, but DNA sequence is the same. But the organization of the DNA, which is chromatin, is very different. So just to connect you with what I said, those who are new to this, DNA in cells, DNA is not bare. It's not like a bare polymer. It is covered by large number of proteins, and it is folded to form this polymer called chromatin. So if you think of a thin polymer chain, it is folded. And if you look under some electron microscopy, it would look like a highly folded thick filament, like somehow. So there is a folding, and we are trying to understand how. We still do not know how exactly this is folded. That's not something we are trying to understand, how this DNA polymer is folded to form chromosomes and chromatin. But what we know is that this folding is very important. This folding is what essentially decides how our one cell type is different from other cell type, how skin cell is different from brain cells or bone cells or eye cells and other cells, right? So we have many different cell types. The cell differentiation and the cell type formation of different cell types essentially is a process of folding chromatin in different ways, and then as a result of it producing proteins by regulating the gene expression, right? So that's what the idea of differentiation and development of an organism, and that's what the crux of this process. So as I said, DNA is assembled differently in different cell types as chromatin, and chromatin organization, which is the self-assembly of DNA, is different, and how different this is something that everyone is trying to understand, and we have some recent understanding of this, which is something we'll discuss. So the point is basically the DNA is organized, the DNA sequence is the same, but it is organized in space, in 3D, differently in different cells, different cell types. So this is the basic idea. Now, how is it organized? So, of course, so it is organized into chromatin, and chromatin is a long polymer. And just like DNA encodes some information which is in the sequence of ATGC, chromatin has to encode some extra information, because now the information that this is a skin cell, or this is a brain cell, or this is a cell in liver, or whatever the organism it is, whatever the tissue it is, that information has to be encoded extra on top of the genetic information. And therefore, there are extra layers of information encoded in chromatin, which is called the epigenetic information. So how this extra layer of information is encoded in DNA is also a part of understanding the chromatin folding of DNA and formation of chromatin. So the epigenetic information is encoded by folding the genome, and different cell types have different, something that people in the engineering and physics call something called microstates and macrostates. This is some particular organization of molecules, and in this case, proteins and DNA and all the chemical marks have organized to different, and this can lead to different microstates and macrostates, different states of the polymer, different states of chromatin. And that's what essentially decides the state and the function of the cell, right? So the microstates and the macrostates in the chromatin essentially decides the function and the fate of the cell. Now, by folding this, what does it achieve? It basically regulates genes. The gene regulation depends on how the DNA is organized, because the genes that are expressed in a skin cell is very different from the genes expressed in our brain or the genes expressed in our heart. Heart cells can beat while skin cells don't, right? So the function and the underlying gene expression, all that is basically encoded in this epigenetic code, and the three-dimensional organization of the genome, that is chromatin assembly, essentially decides how this extra information is encoded and how it affects the function. Another function is, for example, recombination, something that you have heard of. Two different parts of the DNA or come together, and when there's a DNA break, they repair, and you can even essentially change the gene positions, right? So all of that is gene recombination, translocation, etc., are also decided by the three-dimensional proximity of two different parts of the DNA, right? So how the DNA is organized in space and time decides the function and the fate of the cell, essentially. So this is something that we are going to try. We are trying to understand how chromatin is organized in space and time. So space and time often called 4D, because there is 3Ds in space, X, Y, Z 3D space, and time as a fourth dimension. So how is chromatin organized in space and time? And then this epigenetic information is organized through this folding process. How is this epigenetic information organized through this folding process? And not only just organized, when the cells divide, they are inherited. For example, if you have a skin cell, okay, there's a question. Yeah, please ask questions. Sorry, I missed the question. In case there's a question, of course, feel free to unmute and ask me in case I miss the hand that you raised.
Yeah. Yeah. So I had two quick questions.
Yeah.
One was, so you said different cell types have different organization of chromatin, right?
Yeah.
So I was just wondering, within a particular cell type, like within skin cells, could my hand skin cells and feet skin cells differ in how they fold and how chromatin is organized? That was one question. And the other was, I'm a bit of a biology beginner, so I'm more on the computational side, but like in cancer, we say, right, that it is caused by mutations in the DNA, right?
Yeah.
So one is, so there are two distinct things, right? The code in the DNA sequence, right? When there is a aberration, we call it a mutation.
Yeah.
And the kind of orthogonal thing is how that DNA is folded, which is chromatin, right?
Yeah.
So, so when we talk about pathology or diseases like cancer or other genetic diseases, is there an interplay between mutations in the DNA sequence and how it's folded?
Yeah, because the second question, so the second question, quick answer is yes, I'll come to it. The first question that you asked, whether skin cells in different tissues or kind of different tissues, but let's say epithelial cell, but different tissues, would they have different chromatin organization? So I would say, yes, we don't know fully the details of this, but if we have to have different gene expression levels, then certainly chromatin organization can be different. So that's the generic answer, that whenever you need gene expression levels slightly different. So if you want to have, let's say, epithelial cells in one tissue versus another, like liver versus like some other organ, if you want the gene expression to be changed, the chromatin organization typically would be changed. So that's, you can have, yes. And the second part, just to a little bit more to say about, for example, in disease, in cancer, for example, it's like a lot of cancer studies are now also to understand how chromatin is organized in cancer cells. And what is the idea? The idea is that, for example, let me say very broad things. Let's say somebody has a mutation. Very likely that that person will develop cancer, but it's not that that person will 100% have cancer, right? It is possible that people have mutation, but the cancer is still there, the cells are not cancerous, or cancer doesn't manifest. So why is it, right? So there is, people would generically say, okay, mutation plus environment would play some role. So I would say the folding of chromatin would play some role, and you would have a mutation, and when the gene is expressed, only then that mutation would manifest in some function. But if you can fold it and then not have that genes expressed or accordingly regulate the expression, then maybe this disease may not manifest. So therefore manifestation of the disease certainly has a chromatin folding angle. And a lot of, for example, a lot of, let's say, lifestyle diseases certainly have chromatin folding as a big contributor. So yeah, that's the answer. Is it clear? Other more question?
Yeah, so just a quick follow-up. In certain kinds of cancer, there is this thing called HRD, homologous recombination deficiency. So is that related to chromatin? Because I saw the recombination.
Yeah, so the answer is I don't know, but I don't think so. People are just studying right now. People are just studying how does the folding affect recombination itself. It will be the next step to study. Does it have a role in cancer and things like that? So probably naively I would think yes, but I would say we still haven't studied those things in the context of chromatin folding.
Thank you.
Yeah. Okay, good. So let me go ahead. If there are questions again, please stop me. If I did not see your recent hand, please unmute yourself and then ask the question. Okay, going forward. So what I just said is that how chromatin is organized in space and time is crucial, and something that we want to understand. We want to understand how chromatin is organized. Now, if you take textbook, many of the textbook pictures are like this. So this is a popular textbook picture where DNA is folded. So we start with DNA, which is then folded in a particular by the help of histone proteins. So that is histone octamer forms the nucleosomes, and you have a string of nucleosomes. So, and then this string of nucleosomes is somehow folded further. And there are all kinds of pictures like so-called 30 nanometer fiber and so on and so forth. And finally, to form, for example, mitotic chromosome. Now, mitotic chromosome is something that you can see under microscope. So we know that this exists and this is true. DNA is something that is kind of well studied. And this 11 nanometer string of nucleosomes is also kind of well studied. But everything in between, where I have not written anything, all of these are kind of cartoons based on some kind of in vitro experiments, reconstitution, etc. And as we understand what is going on in vivo more and more, it appears that these are not accurate. So for example, 30 nanometer fibers do not seem to exist in vivo at all. Why? If you reconstitute chromatin under particular conditions, they form 30 nanometer fibers, but in cells, in living cells, there isn't any evidence for having any reasonable amount of 30 nanometer fiber that would do some function or something relevant. So all of this in between 11 nanometer string of nucleosomes to mitotic chromosome, all of these are cartoons. And right now, the research in the last few years is basically trying to understand how the chromatin is organized in this scale. And we have a kind of a new understanding of this. So that's what something which I would say today. So the question now is, how do one experimentally measure the 3D organization and understand this? Because I just said that those in between, after 11 nanometer, all of those organizations are not accurate. So how did we know this? How did experimentally one study this? So as you know, the popular methods to study structure of anything, structure of a protein. For example, the most popular method is X-ray diffraction, X-ray crystallography, right? That's how we kind of studied how protein is organized in 3D, how protein is folded. But chromatin is not a crystal. So therefore, X-ray diffraction is not very effective. You wouldn't get a nice diffracted pattern, and therefore we cannot study chromatin very well using X-ray crystallography. Then the other popular method is, of course, cryo-EM. Cryo-EM is, of course, being used to study chromatin, but there are some issues. For example, staining DNA is an issue. If you just stain the DNA, the whole nucleus would get colored or will have some signal, and one cannot easily regulate and study this contours of chromatin or polymer very nicely. So there are various issues in using cryo-EM to study chromatin. There are some partial, there have been some recent attempts, and kind of most successful attempt is by this OSHIA group, which published in Science in 2017. So if you are interested in this, go and have a look at this O et al. From this OSHIA group in Science. There is also Elstow, Maishma, many others try to study chromatin using cryo-EM, and there is a partial success towards that. That's how we kind of understand the absence of 30 nanometer fibers, etc., in cells. The most popular method for studying three-dimensional organization of chromatin is called chromatin conformation capture method, which is kind of a biochemistry and sequencing method, which I will describe in detail a little bit how this method works. So it started as a method called 3C, which stands for chromatin conformation capture, and then kind of a modified versions of this chromatin conformation capture was later known as high C, micro C, and so on and so forth, started by a person called Job Decker in 2002, and then this Lieberman, Aiden, and others took it up, and then there's a very detailed study in the last 10 years or so. So this has been kind of the most popular method, which gave us a lot of insights into how chromosome is organized in 3D. Of late, recently, last 5 years or so, microscopy has been also very kind of successful. There's again, microscopy has the same issue as cryo-EM in some sense. Microscopy also, we cannot label the DNA. If you just label DNA with some fluorescent marker, the whole nucleus will lit up, and you cannot know which part is where. Therefore, people have to develop new chromatin tracing methods. So there is some idea called DNA paint or oligo paint and so on and so forth. And using those ideas, people now started studying chromatin organization. And one of the popular papers and something became very successful is Bintu et al., which is published in 2018. So if you are interested in this, I would recommend you to have a look at these papers. And I would just a little bit describe how this chromatin information capture method works. So the way it works is, if you imagine a polymer, as shown in the cartoon here, And this polymer is kind of cross-linked by many proteins and so on and so forth. Now, what we want to understand, or what these experiments try to understand, is the contacts between different parts of the chromatin. For example, if you think of a polymer like this, wherever there is a red dot, two parts of the genes are coming together and forming a loop or a contact. So identifying these regions where they contact each other is the aim of this chromatin conformation capture method. And once you understand where these red dots are, then we know that there is a loop in between. So the way this is done is you add some chemical, which is formaldehyde, into the nucleus, and that would cross-link DNA and protein together. Once you have cross-linked chromatin, you can use some kind of enzymes to cut certain sequences. So these are like nucleases that would cut the DNA. Once you cut the DNA, you can separate out pieces like this. For example, if you imagine 4 regions being cut and X, a cross-link region, the cross-link region is red, and then you would have 2 pieces of DNA coming together. These are the regions that were in contact, and one of the piece here in the X is a region on some side of the chromosome, while the other part could be far away on the genome, along the genomic contour. So these two, then once you have this X-like pieces, you can separate them, isolate them, and sequence them. Once you sequence them, you can figure out the X part of this coming from which part of the genome by comparing with the available human genome sequence available to us. If you compare, we know where the X is coming from, we know where the Y is coming from, and then we can say that X and Y were in contact. So this is the idea. You figure out these 2 parts of the genome were in contact, and then you make a contact matrix. For example, what is shown on the right-hand side is a contact matrix of X chromosome, for example. So what does it say? If you just look at, for example, around 98 megabase to 103 megabase of X chromosome is shown here on the right-hand side, the data of contact. So this is called a contact map. And wherever there is a red dot or darkish red dot, that means if you take 1,000 cells and 1,000 X chromosomes, in what fraction of those X chromosomes you found, for example, let's say 101 MB and 102 MB were in contact, right? 101st megabase region and then 102nd MB region were in contact. How often they were in contact is marked in this color map, in this contact map. If they were in contact very often, there will be dark red. If they were not in contact, then they would be whitish color. So you can see a color bar here on the right bottom. Dark color means most of the cells they were in contact, and white color means they were rarely in contact. So this is what this contact map means, and this is how the contact map is generated. So this is the experimental data of chromatin conformation capture, and this can be converted to a probability of contact between 2 segments X and Y. So essentially, if you normalize this map, this becomes the probability of contact between any 2 segments X and Y. And this is a symmetric matrix because contact between X and Y is same as contact between Y and X. So this is a symmetric matrix. P of X, Y is same as P of Y, X. So I will take a pause here and let you ask any question if you have to understand this contact matrix and how they obtain this contact matrix. Because I would use this contact matrix for the remaining parts to explain what we did to understand chromatin organization using this contact matrix. Are there any questions?
So could you talk a little bit, explain this a little bit again, like these X and Y, these are what segments, like what is the relationship between X and Y? Could you?
Yeah, so let me just go back one step here back. So let's say you have a chromatin genome, which is a polymer. Now our aim is to know, for example, if you look at the red dot here, one part of the genome is in contact with other part of the genome. We want to know what are these regions, what is the base sequence locally here of these two regions. For example, if you know the human genome sequence, you know this is, let's say, this is 100th MB and this is 102nd 102 MB location, then we know what the sequences are, right? So we want to basically know which two parts of the genome are in contact. So we know all these, we want to know all these points, and we want to know which two parts of the genome are in contact, so that we know that how it is folded and organized in 3D. So that's the aim.
So when you say contact, what do you really mean? Like, do you mean like physical contact?
Yeah,
or
physical contact.
Okay.
They are in proximity and they are actually in touching each other.
So are there like forces and
polymer.
stuff between them? Like, do we think about, are there physical interactions between those?
Yeah, indeed. Let me show this 1 second. If you take, so any polymer, like if it is folded, right? If you something is folded like this, like there would be different regions coming in actual physical contact. For example, this region and this region are actually in physical contact. This region and this region in physical contact is in physical contact. So once you tell me these regions are in physical contact, I get some idea about how it is folded in 3D.
Got
so
it.
That's the idea.
Thank you.
So this is something that you can experimentally obtain. So take it and then reconstruct how this is probably how it is organized in 3D. That's the idea.
And then there might also be like, like two regions which are spatially close but overlap, and then two regions which are spatially far apart but still overlap, right? They would lead to very different interactions, right?
Yeah, so there could be two segments which are very nearby, they can locally fold, but they could be very far away and they can also fold and come nearby. Yes. So to get this, what one does is you throw in some chemical, formaldehyde typically, so that there will cross-link and fix all this 3D organization. And then you cut using an enzyme. Once you cut, it will be pieces, but these red dots regions will be cross-linked. Therefore, it will be stuck there. Then you can separate out through some process. And then at the end, you will have this X-like segments. Many X-like segments is what you will have, because imagine this cutting at different locations. You will have many X-like segments and many straight segments. Straight segments we'll throw away. We are not interested in this because they are not in contact. Wherever there is an X-like segment, that means there is a contact. We take it out and sequence it. So you then, if you have an X-like segment, little X and little Y, by sequencing those two pieces, you know where that they come from by comparing with the available genome data. And then we say that, okay, I took out a sequence and I found 100 times X and 100 times Y. That means in 100 cells, this X and Y were in contact. That's what it means. Right? So that's how this contact matrix is obtained. So contact matrix is essentially like a frequency matrix. How often, in how many cells, and how many chromosomes you had this region X and region Y were in contact. And for example, you can ask the question, what is the contact between 100th base pair and 700th base pair, or 100th base pair and 7000th base pair? Okay. Any other questions? So if there are no questions, I'll just go ahead. Feel free to stop me. So this is the first understanding, the new understanding, that chromatin is organized into these domains, which is shown here, which has a name. It's called topologically associating domains. So this is the paper which is in 2012 first proposed this name, Nora et al., where they call this topologically associating domains. And what does this domains mean? These domains mean, the red color means locally they are folded. So this is as if locally chromatin is folded here. Then another local fold, another local fold, another local fold, and so on and so forth. This far away, for example, here, this means this region and this region, 99 and 101, are in contact, but rarely. Not that this region is, there is a color, but not that red as this region. So they are occasionally in contact, but not all the time. So this basically gives you a kind of a mathematical representation of the polymer structure. So this is that the chromatin is such STAD-like structures is the one of the new idea that came out. The STAD is also represented in this way. So if this is because it's a symmetric matrix, so only one side of the diagonal, either upper diagonal or lower diagonal matters. So if you just take the upper diagonal part of it and rotate it, it will be like a triangle. So that triangle part is what is represented. So this square, if you look at only this part of the square, it would appear like a triangle. So that's what another representation. So this A, B, C, D, E, F are all different TADs or different domains. So this is D domain, E domain, F domain, G domain, H domain, and so on and so forth. So this is kind of manually annotated in some sense and shown here as different domains. So this is the first understanding of these domains. Now, the question that we are interested in is, how do we get the 3D configuration of chromatin from this domain? Once you get this such a matrix, such a picture on the left-hand side, how do we get back the actual chromatin picture? Because this is experimentally what you get is only what you have on the left-hand side. Now you have to reconstruct what is on the right-hand side. Now, if the probability of this contact were almost 1, then it is relatively easy to reconstruct. We can just put a loop and say that, okay, they were in contact, therefore I have a structure. But this probability of contact is not 1. They are very small. In other words, any dot here, if you take, that probability is only like less than 0.1. So that means any contact between any two regions, only you see those contacts only in, let's say, 10% of the cells. In 90% of the cells, those regions are not in contact, suggesting that the chromatin is highly dynamic. There is some structure, but the structure is dynamic, but still in overall there is an average structure. But the probability of any contact is small. Therefore, you cannot easily go from left-hand side picture to a right-hand side picture. You need to have a algorithm or a kind of a theory or a model to go from left-hand side to a chromatin polymer picture. So this is something that we are interested in, to understand how to go from this left-hand side picture to a right-hand side picture. And so one idea that we used, I'll discuss today, but there are other ideas as we have in this paper and other papers. So let me describe one part of how to go from this picture to this picture, which is basically how to make a polymer picture of chromatin that is consistent with this kind of contact map that is obtained from experiments.
And I had a quick, another quick question, sir. Like whatever technique we use, how accurate it is? Like, is there some notion of a ground truth? Like, can we experimentally determine like the ground truth 3D conformation of chromatin?
Correct. So good question. So now it depends on what you call as ground truth, but this is one experimental data that you have, which is the contact matrix. But there is another experimental data which is kind of microscopy data, which you actually see with your eyes, right? So you put chromatin under a microscope, like super resolution microscope. Then you can say, I can fluorescently label gene 1 and I can fluorescently label gene 10. And then you can ask the question, how far gene 1 and gene 10 are? Like, what is the distance between gene 10 and gene 1? And this is something that experimentally one can measure. And I will just show some data and compare with some experimental data. That is some sense kind of, if you would call it ground truth, I don't know. right?
And one final, sorry for interrupting you again and again. So reminds me of the protein community, right? The protein community has PDB, right? I was wondering, is there a similar database to PDB for the chromatin community?
So there is, I'm not, so there is this ENCODE, which is the epigenetic database. It's called ENCODE, ENCODE. A lot of data is actually uploaded in this ENCODE. Most of the data is there in the ENCODE.
Okay.
So that's the database for our epigenetic chromatin community in some sense. Yeah. Okay.
Yeah. Thank you.
Okay, good. So then we're going to try to understand a little bit of 3D organization of the chromatin, which is essentially converting chromatin to a polymer. And if you want to draw a polymer picture of chromatin, how will you draw this? That's the question that I'm going to understand, tell you, and drawing that in a kind of a quantitative manner. Now, in the last kind of 20, 30 years or so, people were trying to understand DNA as a polymer, and there's a very good understanding of how to describe DNA as a polymer in a very quantitative manner. And one of the picture is this. So if I have to tell what DNA is to a kind of a lay person, the model used is something called a worm-like chain model. You can think of the contour of the DNA like a chain, kind of a worm-like chain, which has some stiffness. It's not like completely floppy. It has some stiffness. It has some twist. So there's a twist persistence length. It has some bending persistence length. It has essentially no intrinsic curvature. For example, if you, typically short length scale DNA would be a straight filament-like chain, right? So we have a good understanding of how DNA, to describe DNA as a polymer, but we don't have a good understanding of chromatin at all, how to describe chromatin as a polymer. So that's what we are interested in. So this is again, going back to what we know that DNA, if you draw DNA as a polymer, then chromatin is nucleosomes. It's like a string of nucleosomes, right? Histones, DNA wrapped around histone proteins, forming a kind of a string of nucleosomes, and which is further folded. So this is what we broadly know. Now, if you want to think of this picture for the entire chromosome, and if you want to draw chromosome as kind of a string of nucleosomes, and let's say if you want to simulate chromosome as a string of nucleosomes, it's almost impossible because there are millions of nucleosomes even in a single chromosome. Human chromosome 1 is more than 200 megabases, like 250 megabases, which has more than 1 million nucleosomes. So it is too big to draw, too big to simulate. So therefore, even you cannot study and organize this on a computer. So we need a more coarse-grained picture of chromatin. A coarse-grained picture of a polymer is something like this. You want to convert this kind of left-hand side picture to a right-hand side picture, which kind of a beads connected by springs like a polymer with some kind of contacts. So this is what we want to think of. Why am I saying this? Because you can just a little bit think about this. The only way of simulating a polymer, or the easiest way or the simplest way of simulating a polymer, is to think of it as beads connected by springs. Any other way will have some difficulty. Obeying the rules of thermodynamics, etc. There would be, so this has to obey, when you simulate chromatin, it has to obey the rules of thermodynamics and rules of physics and chemistry. So obeying all these rules, the simplest way to simulate any polymer is to think of it as a bead spring chain. So if you want to think of chromatin as a bead spring chain, this is the picture on the right-hand side. We know that there are some contacts between faraway regions. So you would want to put some red contacts on a beads connected by spring chain. Now the question is, what is this chain? What is the quantity? For example, if you put a spring, how stiff the spring should be? If you put a contact, how stiff this contact should be? Should it be always in contact? How often they are in contact? How do you make this chain? And if you have these beads, how big is this bead? For example, this bead, if you just say 5 nucleosome is a bead, what is the size of this bead in nanometers? Is it 20 nanometer, 50 nanometer? How many nanometers is a bead? So these are the questions that is required to draw or to simulate this and have a quantitative picture. So these are the questions that we want to understand. What should be the spring constant of the springs so that you can simulate it in a quantitative manner? What should be the size of this beads? For example, if this bead is 1 kb or a 5 kb chromatin, how many nanometers is it? How bendable is chromatin? Is it like very floppy or is it difficult to bend? How is there some kind of intrinsic curvature for this, which is a polymer quantity, and how bendable is, and what is the interaction between different parts of the chromatin? For example, you know that you cannot, the interaction between these materials are known to us, right? What is the interaction between different parts of the chromatin? These are the questions that one needs to know to simulate this. The only thing that we know is nucleosomes, because nucleosomes is kind of reasonably well understood. We have crystal structures. We have been studying over many years. So we know if you draw a nucleosome picture, the distance between 2 nucleosomes is 11 nanometers. And if you draw a linker DNA, this is about 25, 28 nanometers. So these are the things that we know. So since we know some information about nucleosomes, what we decided is to take some recent data which came out in 2020, which is a nucleosome resolution contact map. So this contact map experiments progressed, and in some cells, for example, mouse embryonic stem cells, they could generate a contact map of different parts of the chromatin coming in contact at nucleosome resolution. What does it mean is that this experiment could tell how often 10th nucleosome will come in contact with 100th nucleosome. What is the probability that ith nucleosome will come in contact with jth nucleosome? So this data was available recently. So we use this data to reconstruct the chromatin organization because we know something about nucleosomes already. So for example, what you see on the left-hand side is the contact probability data at nucleosome resolution from this experiment by C et al., which is published in about 2-3 years ago. This red dots means high contact. So you can, this is about 10 power minus 1 is the darkest red, and it could be as small as 10 power minus 3 and 10 power minus 4. So this is the contact map that one got, for example, a particular region, which is about as shown in this here. Now the point is to convert to a polymer picture. So one way is the following. You take a polymer, wherever there is a dot, you put a spring, but put a spring in a statistical way. So that's the crux of it. You put a spring with a probability Pij. Don't put the spring always. For example, if the contact probability is 0.1, put the spring only in 10% of the conformations. But we didn't put randomly, we put some extra constraints, which I wouldn't discuss right now, but it's there in the work that we published. But I'll be happy to explain this to you at the end of it if you need. But the basic idea is you take this, put springs in a particular way, so that in Pij percent of the Pij fraction of this configurations, so we have an ensemble of chromatin configuration. In Pij fraction of the configurations, you have a contact between 2 nucleosomes, which are as per the information that we obtained from this contact experiments. Once you have this, you can run a simulation called Brownian dynamic simulation. So we ran a Brownian dynamic simulation, which would equilibrate these polymers with these constraints of this connection between spring connections. And then we can get 3D configurations. And then we can ask the question, in the configuration that we got, are we getting the same contact as you saw in experiments? For example, look at the bottom right-hand side and left-hand side. So this is, for example, this is a TAD-like domain. Experimentally also, there's a TAD-like domain. In our simulations also, there's a TAD-like domain. There is this arm like this, which is a faraway regions in contact. For example, 70.35 is in contact with 70.26. So that's what this means, right? So there is a faraway contact. Experiment also have faraway contact. So we generated a bunch of configurations which essentially has similar contact as experiments. So this is, for example, a heterochromatic region on chromosome 7 in mouse embryonic stem cell, euchromatic region on chromosome 5. There are TADs, we also have TADs. So what we essentially did was to put springs and equilibrate such a way that it satisfies this contact probability. Once we have this, as you were asking, we can then measure the 3D distance between any two regions. We can ask the question, for example, what is the distance between this bead and this bead? This is a particular region on the genome, and this is a particular gene on the genome. What is the average distance between these two regions? We can ask this question, and that plot is this. So the green curve that we have is what is the 3D distance given the genomic distance between two nucleosomes. So if given the genomic distance between two nucleosomes is about 100 kb, the average 3D distance is about 260, 270 nanometers. So what is the 3D distance on an average given the genomic distance along the genome? Right? That's the question that one can ask, and there is a microscopy data. For example, this is red dots. So this particular data is for alpha globin gene on mouse embryonic stem cells. The same alpha globin gene, same mouse embryonic stem cell. There is also the experimental data from microscopy super resolution experiments. So they took 3 pairs of regions and then measured what is the average 3D distance between those 2 pairs. For example, between roughly like 120 or so kb away, and then that's what this red dot. So this actually matches with what we predicted. So we predicted based on our simulations without any fitting parameter, because we already know the size of a 200 base pair chromatin. So since once we know 200 base pair, it's a nucleosome plus a little bit of linker DNA. So we know the size of this. Once we know the size of this, there is no more parameter for us. So we can actually predict this green curve without any fitting. And then you compare with experiments, you they fall on top of each other, giving us this nice match between experiment and simulation prediction. These blue dots are for some other chromatin. Of course, we don't expect it to match because the chromatin is different. There's different contact map. But the order of magnitude you want to check, if you have an X chromosome, this is from an X chromosome. If you have an X chromosome, what is that 3D distance is something that is measured here. And that's what this is shown for that particular experiment. Of course, it wouldn't exactly match because that's not what we simulated. We simulated an alpha globin region, and it matches with the alpha globin experiment. And if you take some other gene, it would have some other 3D distance. And that's what shown here as a kind of a comparison, as an order of magnitude comparison. Okay, so this is a red and blue comparison. Sorry, red and green comparison is the first indication that we have reasonable structures. So we have 3D distance comparable as well as contact map comparable to what is experiments. So once we have this, we can take the structures and then ask a question that if you take, for example, 10 nucleosomes and call it a big bead, a spherical bead, we can ask what is the size of this bead. So that will be the size of, let's say, if you take 10 nucleosomes, which I call a coarse grain bead. So if you take 10 NB equal to 10 means 10 nucleosomes. 10 nucleosomes is about 2 kb. So if you take 2 kb of chromatin as a sphere, what is the radius of the sphere? That's the question that would tell you how big is 2 kb chromatin is, or how. Then you can take 50 nucleosomes, which is about 10 kb, and think of that polymer as a sphere. So it's a kind of a spherical. If you put a spherical shell around it, how big is the sphere that would contain this 10 kb chromatin. So all of this question we can systematically ask based on this configuration that we have. That's what we did. So we have a radius of such spheres as a function of NB. We can also ask if you have a polymer made of such spheres of 10 kb or a 5 kb or a 2 kb coarse grain beads, what is the interbead distance, how they are fluctuating. This fluctuation would give us something like the spring constant that we had seen earlier. We can also ask what is the angle. So many of you are aware of Ramachandran angle in the protein configurations. So just like if you think of protein chromatin folding like a protein folding, you will have to know what is the angle between different segments of the chromatin. So the angle can be also measured. This is this bending angle and dihedral angle, something that we can also measure, which we measure, right? So this is the answer that we get. If you take the radius of these spheres and vary the number of nucleosomes, so x-axis is the number of nucleosomes. So 5 nucleosome is 1 kb, 50 nucleosome is 10 kb, around 380 nucleosome is about, it's about I think 400 nucleosomes would be about 100 kb if I'm not wrong. 3 something will be around 100 kb. So right. So different kb of chromatin, if I take, what is the size of the sphere average? That's what is shown here. As a control, if you just take a polymer without any interaction, polymer theory already predicts how that curve should behave. There's a green curve, which is a control. If you take some experimental data and compare it with, so this data is not available for all kinds of cell types. For example, for Drosophila, data is available, so which is recent experiments. And if you take the data, it comes roughly comparable to what is we predict. And this is for very many different genes and different curves for different genes. And along the gene, of course, the sphere size will vary. So the radius as a function of genomic distance at different locations on the genome. If you take 5 nucleosomes as a sphere, you would get the size of the sphere varying, which says that at some regions the chromatin is expanded and some regions the chromatin is compact. And this compaction means small rg. Extended chromosome would mean large rg. So this fluctuation means along the genome, chromatin is compact or extended. That's what this means. Where all chromatin is compact, where all chromatin is extended. So that's what we predict on the left-hand side. And the average behavior of this, the average of this is shown on the right-hand side. And kind of compares with microscopy experiments from available data. It is certainly order of magnitude correct. For example, if you take 100 nucleosomes, there will be approximately around 100 nanometers, right? That's what this RG this says, right? So yeah. So if there are questions, I'll be happy to answer so far. What we just took is to take a bunch of chromatin configurations and characterize it by drawing spheres around this polymer pictures and asking the property of this. Are there any questions so far? Okay, so if there are no questions, so as I said, this means that some areas chromatin is compact, some area chromatin is extended, and this is, if you compare this with the contact map, which is on the top, there are at this TAD boundary size, these triangles are called TAD, topologically associating domains. At the TAD boundaries, as if chromatin is kind of extended, interior of the TAD chromatin is compact. So this is the kind of picture that one would get by comparing the RG, the radius of gyration, with this contact map. We can compute other quantities, as I said, the distance between two neighboring spheres, how much they would fluctuate, and that fluctuation is quantified as a kind of a distribution So this is the distance distribution between two spheres. And that distribution, the width of the distribution is related to the spring that I drew earlier. So if you want to think of chromatin as a beads connected with springs, the spring should have a stiffness, which is what is shown here. It's about 1 to 10 piconewton per micrometer is the stiffness of the spring of chromatin. If you think of chromatin as a beads connected by springs, this is the stiffness of the spring in real units, should be approximately 1 to 10 piconewton per micrometer, right? So these are some numbers that we are putting out, which can be used for anyone wanting to simulate chromatin, would use beads having bead size given by here, and would put springs with a spring constant given by these numbers, right? So you can connect beads with the spring constant and simulate chromatin. And that's not enough. It's not enough to have beads and springs. You need to have these beads interacting with each other. What kind of interaction would you have? If you think of one interaction as steric repulsion, for example, these two parts of the cable cannot cross or overlap, so they would sterically repel. So in protein literature, you know that Ramachandran angles emerge from some kind of steric hindrance. So if you think of coarse grained beads like this, so if you think of 5 kb of chromatin as a bead, which is the red color, another 5 kb of chromatin as a bead in blue color, do they sterically repel or can they kind of overlap? If you imagine polymer picture, certainly the polymer can mix. Therefore, there can be some overlap. So there is some, so this interaction is not like a hard sphere. There are some soft spheres which can a little bit overlap. There is some softness, and we quantify this softness in this work. And we have a particular way of quantifying this overlap, which I wouldn't go into the detail. I'll be happy to answer this if you have questions. And then we quantify the angle. So this is the angle between, for example, if you have spheres like this, you can draw a vector between these two spheres and another vector between these two spheres. And you can ask the question, what is this angle? This is called the bending angle. Or if you have 3 vectors like this, 3 segments like this will make 2 planes. This is called dihedral angles. This is, for example, these are the Ramachandran angles in proteins. Equivalently in chromatin, if you think of dihedral angles, the phi is a dihedral angle. What is the dihedral angle is a question that we can ask. And we also have answers to this question. At nucleosome resolution, this is the angle one would expect, where there is a peak at 60 degrees, which again, experimentally, there is some hint of a 60-degree angle between nucleosome resolution chromatin. And if you have coarse-grained chromatin, this is the angle distribution. So P theta Cg is the coarse-grained bending angle distribution. So all kinds of angle between 30 degree to 150 degree is possible, which means that things are quite flexible. The chromatin in vivo can have all these angles. This is because there is all kinds of proteins binding and dissociating, making the chromatin bend and open up, making all of these angles possible. Unlike protein, which is kind of folded and sitting there to have a function, chromatin is folded and unfolded dynamically. Therefore, if you look at over a period of time, one should see all kinds of angles, with mostly angles between kind of 30 and 120. That's the prediction that we have. So the details of this is available in this paper, which recently published by Sangram Kadam et al. You can all see that if you want to know the details. And this kind of things can be also experimentally measured, primarily through microscopy experiments, because there are the so-called chromatin information culture experiment. There's the data that we take, and then we can predict what one can observe in microscopy experiments, right? So all this, for example, this says that the RG or the 3D distance at the TAD boundary is large compared to interior of the TAD. Experimentally, you can see that between the TADs, there is larger distance, and within the TAD, there's lower distance. So this is something that experimentally can also be measured. And what we predicted, the hint of what we predicted is already seen in some early experiments that is already recently measured, right? So we then go on to actually study the detailed interaction between different parts of the chromatin. So what, as you know, in physics and chemistry, if two parts of any system interact, they interact through some kind of potential energy. So there's an energy of interaction. So VI is the energy of the interaction between some bead, right? Which are, so if you have a bead of chromatin, ith segment of chromatin, and you have another segment r distance away, how do they interact? Do they attract or do they repel? This is the question. And that interaction V of R is something that we kind of derived through a method called iterative Boltzmann inversion method. This inversion method, because we want to generate this potential that is consistent with what is seen experimentally. So we take the experimental data and do some kind of an inversion and obtain this potential energy. So this V is the potential energy, and P is something that is already we obtained in the polymer models. So V is obtained such that P is correct. So this is the mathematical framework using which this is done. So we have this particular form for the interaction energy. At small lengths, they repel, but the repulsion is not very strong. They are soft. And when they are a little bit nearby, they attract, and far away, the energy is zero. Far away, energy is zero. There is some attraction when they are nearby because of the proteins would bind and make them attract. And when they come too close, they would repel, right? That's the interaction. And there is a particular mathematical form we propose for this interaction, which is here. And using this potential, if we simulate chromatin, we get 3D radius distances like radius of gyration of the polymer, etc., which is reasonable and compared with something which is known from a full detail simulations, right? So we can predict 3D distances using this potential energy that we propose. So basically what we just wanted to say is that we have a bunch of properties of chromatin: RG, interbead distance, angle, overlap, which all inconsistent with contact map is what we have. And there are other polymer properties which I wouldn't go, but I'll just summarize that if you want to think of chromatin as a polymer, the textbook models of chromatin organization is not accurate. And beyond the 10 nanometer picture, there is new picture emerging, new chromatin models emerging, which is basically chromatin is made of topologically associating domains interacting in a particular manner. And this new understanding of 3D organization in the last few years is something that I described. And using that, we propose a model to describe chromatin as a polymer, and one can predict all the properties of the polymer and how to use those properties to simulate chromatin, or even to draw a picture of chromatin in a quantitative manner, and which would involve some kind of bead spring polymer model with flexibility and softness. And we provide some real numbers and real units that can be used in those simulations. And the details are available in these papers. So that's the summary of the work that we did, chromatin as a polymer part. I would again take a break here, and then I'll stop here temporarily. And I have one more small part on some epigenetic aspects of chromatin. But depending on the question and interaction, I can take it up or not take it up. I will just wait for a pause for a minute and take questions if you have.
We would love to, at least I would love to hear a little bit about the epigenetic stuff, maybe briefly.
Yeah, good. I'll do that. Others, are there any quick question on chromatin as a polymer part? Okay, so let us just go to the second part. Now, I said that the organization of the chromatin, the folding of the genome, contains epigenetic information. So chromatin is folded into polymers, its nucleosomes, and so on and so forth. What can we say about this epigenetic information and copying this information? Because as I said earlier, if you take a skin cell and if the skin cell is dividing, the skin cell would remain a skin cell even after cell division. So that means it's not enough to copy the sequence of the DNA. It has to also copy something beyond the epigenetic information as well. So how do we copy this epigenetic information is an open question. People are trying to understand. And a part of it, something that we try to study, and that's something that we will just describe. So as I said, when cells divide, DNA, the genetic code is copied, the sequence of the DNA is copied. What happens to the epigenetic information, which is beyond the DNA sequence? So how is the epigenetic information stored? One way it is stored is in those folded nucleosome structures. If you think of chromatin as a bunch of nucleosomes, along the genome, along the contour of the chromatin, nucleosomes have certain chemical modifications. For example, there is acetylations and methylations of the histone protein. So they are post-translational modifications. So I just showed as a cartoon. For example, if you take a histone and look at a particular residue, if there is a chemical mark, let's say it's a methyl mark. If there is a methyl mark, I would put a star here. If there's no methyl mark at that particular location, there's no star, right? So this is kind of a code. There is a methyl mark or there is no methyl mark, right? So similarly, it's like a flag. There is a red color flag or there is no red color flag. Similarly, there can be an acetylation mark and so on and so forth. So there are sequence of this modification along the genome. It's like it's forming a pattern. And this encodes some amount of information on how to fold chromatin itself. For example, this decides the interaction strength that we said, the potential energy that I talked about, is basically coming from this kind of histone modifications, which essentially through some proteins lead to those kind of interactions, which eventually fold the protein. Now,
I had a quick question.
Yeah, sure.
In cancer, we read about this thing called CNV, right? Copy number variation. So is that also epigenetically related?
Copy number of the chromosome itself, is that what you're saying?
Yeah, not copy. So there is this term called CNV, copy number variation.
Okay.
So it's often mentioned in conjunction with methylation.
So I am not fully aware of this, but there are two. So of course there are some things like ploidy, which is the number of the chromosome copy itself changing, which is also affect cancer. That is not directly related to epigenetic in some sense. It is related to cell division and all that. But the gene copy itself can vary. And that is related to translocations and so on and so forth, which is related to epigenetic in some sense. So yes, so that way, if the copy of the gene itself, translocation can affect those kind of sequence itself getting copied, probably. I am not an expert on that part of the study of the chromatin aspect. So I do not know, but here what we are talking about is the histone modifications. If you have a chromatin, along the nucleosomes, there are some methyl marks and acetyl marks, and that form a particular sequence, and that can decide whether how a particular region is folded. For example,
like, is this like you said, this methyl mark, no methyl mark, methyl mark, no methyl mark, like this pattern, right? That is that like a binary code which is encoding information?
Yeah, that's what this slide says. This says that the typical histone modification part essentially is like a binary. If you average over many cells, that's what experimentally one would measure. What you get, you can get this kind of pattern like in the red color here. So let me explain this. So what is here is written as H3K27Me3. So H3 is histone 3. On the histone 3, which is the H3 protein, right? On the 27th amino acid is a K. So on the 27th amino acid, there is 3 methyl mark. So as Me3 means, right? So this is an important mark for heterochromatin. So wherever you have this particular mark on the 27th amino acid on H3, if there are 3 methyl groups added to it, very likely that will be a part of a heterochromatin. This is a code. That too, not just any heterochromatin, some particular heterochromatin. And for example, what is shown here is an experimental data on human chromosome from this base pair to this base pair. So you can see some regions very often you find. So the peak, the height of this means how often you find a mark there. That's what this peak means. So this region, this mark is seen very often, while in this region, that mark is not seen very often. Right? So this pattern means how often you see this mark if you take 1,000,000 cells. That's what this pattern means. Is that clear? Now,
and there could be associations of particular patterns to disease and pathology, right?
Yes, there can be and there is. And these are the things that people are currently trying to understand, how this is correlated to certain diseases, because this is certainly directly correlated to gene expression, and the gene expression is certainly correlated to diseases. So, yes. Now, these patterns, if you convert to a single cell, in a single cell, at any nucleosome, there is a methyl mark or there is no methyl mark at a particular location. For example, at 27th acetyl K, there is 3 methyl mark or not, 1 or 0. So in that sense, it is a binary code. So this data is basically taken from this paper. You can have a look at it if you want. The data is here. So if you get such binary code, which is an epigenetic mark, you can ask the question that when cells divide, this genetic code is copied, what happens to this mark? What happens to the S3K27Me3 mark? So this recent set of experiments from many labs show that just after replication, even the mark is copied. So if you look at the top part in this picture, you see this mark before replication and after replication. So during the after the replication, DNA is copied certainly, but not only the chromatin is formed at replication, and if you look at the epigenetic mark, that's also kind of predominantly copied. So how does it happen? That's the question. And one theory is that imagine a DNA which has 4 nucleosomes. Okay, these are 4 nucleosomes, the 4 red spheres here. Now, this DNA is divided into 2. When you replicate, it becomes 2 copies, right? Now, when you copy, you very well know, like all this lagging strand and leading strand and all the copying mechanism, something that you are aware of. Now, when you copy, what happens to the nucleosomes? So what is known through some very interesting experiments are that nucleosomes are randomly handed over to either of the strands. So you take this nucleosome, you hand over into this bottom strand. So you get a red color there. But correspondingly in the other strand, other chromosome, you have 2 copies of the DNA. So one copy of the DNA will get the nucleosome from the parent, but the other copy will not have a nucleosome from the parent. So you have to make a new nucleosome and put there. So that's the blue color. So the blue color means it's a newly made synthesized nucleosome. Red color means nucleosome inherited from the parent.
But is the blue identical in conformation and shape to the red?
It is identical except it doesn't have this chemical mark.
Is it? Can we think of?
This, yeah.
Can we think of it as complementary to red, like, or am I mixing up DNA and chromatin?
No, for all the functional purposes, for all the it is same as red. As far as protein is concerned, it is exactly the same as red. Only some chemical modification on that. The post-translational modification is different. Otherwise, red and blue are, as far as protein sequence, as the protein structure is concerned, they are the same. This is a nucleosome, this is also a nucleosome. But the only difference is this was there from the mother. Therefore, whatever historical chemical mark on that would remain. For example, if you think of this star on it, would remain as a star there, while a new one would not have the star. There's the only difference between red and blue. Blue will not have the star, red will have the star.
So one final thing, if we know the marks on the red, can we predict precisely what the marks on the blue will be? Like it will be absent?
No, at t equal to 0, it will be absent. Now there are enzymes that put these marks. So there are many enzymes that actually come and put these marks on this blue and make it red-like. Okay? So now the question is, as far as the cell is concerned, it would want to put the mark such that it would look like the parental chromatin, right? If you have 2 daughter chromatin, you want to put those marks on those blue, newly synthesized nucleosomes, such a way that overall pattern of the marks should appear exactly like the parental. Because that mark, the pattern, has some information on how to fold the chromosomes, and this folding would affect gene regulation. So for example, if a region is heterochromatin, you put those heterochromatin marks there such that that region will remain heterochromatin. If some region is gene coding and expressing, like those genes are highly expressed, you put, so typically the highly expressed regions have some kind of acetyl marks. So you put some kind of acetyl marks there such that that region will continue to be expressed. If you wrongly put methyl mark there, that genes will not be expressed anymore. That chromosomes will be folded and that genes will not be expressed. So somehow the cells have to figure out and put appropriate marks on these blue nucleosomes. Now the question is, how do they do this? How do these enzymes know what mark to put on this newly synthesized genome? And we have a model and we have a very simple answer. So I wouldn't go into the detail of how we did it. So since, as I said, this is like a binary sequence. So one can use some ideas from information theory to do this. Why information theory? Because think of this sequence, the top sequence is the mother sequence, which is 11100. This one would go randomly to, this is a parental nucleosome, which has a modification. That's what 1 would mean. This second line is one particular strand of replicated chromosome. Okay, one particular replicated chromosome. So this nucleosome came to this strand, but the second nucleosome went to the other strand. Therefore, this became a newly synthesized nucleosome without that mark. Therefore, it is 0. This nucleosome came here. This nucleosome went to the other strand. Therefore, 0 here, 0 here. So this is a newly synthesized strand which has many zeros because some of those nucleosomes went to the other strand, which I am not showing in this picture. Now you have to fill this 0 with 1 or not. Is the enzyme have to make a decision. Now, this binary sequence, if you think of, so what we did was we did this work in collaboration with, since I am in IIT Bombay, we have people working on mobile communication, information theory, which are completely different field from biology, but they have a problem which is somewhat similar, where, for example, if we communicate over a mobile phone, and if I say something, the information is encoded and sent somewhere. But due to some noise, a part of that information will be lost. And only as I speak from here, some part of the things that I speak could be lost as noise, but your computer gets all those information through Wi-Fi. And you get a sequence. And your computer somehow has to reconstruct what I'm saying and give to you, right? So even if some noise makes it to lose some information, even if some noise comes into the come into the data that goes from my computer, your computer should be able to faithfully reconstruct it. So how to do that is something that is kind of well studied in the field of information theory and mobile communication. So we use those ideas, reconstructing back such sequences. Non-noisy version of the sequences are kind of a problem in communication theory. So I collaborated with some guys who are experts in that. So Nitya, who worked with us, with Shiviraj and I, did this work, and the details are available in this paper, which you can read. And we use these ideas, and we use something called a Markov model. I wouldn't go.
I had a quick question on
sure,
the slide. Can I ask?
Yeah, sure.
So if you look at the second and third rows, right? Everything, like our goal is to reconstruct the first line, right? The first row, right? Which is the parent sequence, right?
Yes.
So I was just, if you look at the fourth circle in the third row from the right.
Yeah.
Right. So we ideally want that to be a 1, right? A blue 1.
Yeah.
The fourth circle on the third row, right?
From the left or from the right? From the right.
From the right.
Okay.
So why does it say uncorrected nucleosome to that? Like, shouldn't that ideally, we wanted to correct it to a 1, right? Because it was a 1 in the parent. When it divided, it became a red 0, right?
Yeah.
And we would want to correct it to a blue 1, right?
Yeah, that's a good point. So yeah, the answer is there is nothing called a perfect correction. So these are either done by enzymes. So enzymes will not do. Enzymes can make mistakes. So there is some noise still. There is some mistake. So this is the question. The question we are asking is, if you know some statistical information about the sequence, so that's what is written here. If you know some statistical information about the pattern, what would the best known algorithm do? Can the best known algorithm get back the original sequence faithfully? How good can it be? That's the first question that we asked. Even if you take the best algorithm known in the information theory, mobile communication field, what can it do? Can it reconstruct the best? How good it would be? That's the question that we asked, right? So if enzymes were some computing machine, that knew how to reconstruct this patterns using these complicated algorithms that is developed by these engineers in communication theory, what would that lead to? Enzymes likely, they are not computing machines like the computers, right? So that's the first question we asked. And this is some statistical information. So when I say known some statistical information. What is the statistical information? So if you think of this sequence as a Markov chain has two parameters, alpha and beta. I wouldn't go into the detail. Alpha is the probability to have 1 followed by a 1, and beta is the probability to have a 0 followed by a 0, and 1 minus alpha and 1 minus beta is the probability is to have 1 followed by 0 and 0 followed by 1, and so on and so forth. Right? Once you have two parameters, alpha and beta, in a Markov chain. So given this sequence, one can get these parameters alpha and beta, and that is the statistical information. So if I give this statistical information of alpha and beta, what can this algorithm do? So this algorithm can reconstruct. So what is a good algorithm? So one of the good algorithm is called map decoding. Map decoding is maximum a posteriori probability decoding, which basically maximizes the conditional probability, which I wouldn't go into the detail. So if you do map decoding, what one gets is this, right? So given an alpha and beta, if alpha is 0.6 and beta is 0.1, there would be 30% error. If alpha is 0.1 and beta is 0.9 in a particular sequence, there would be about 5% error, right? So given an alpha and beta of the sequence, what is the best algorithm, or what is a very good algorithm? What can it do? This is what it can do. Even the best algorithms can still make some mistakes. In some cases, some sequences, the mistake would be as bad as 30% error. In some sequences, the mistake can be very small, about 5% error. This is the very good algorithm. This is what it can do. So as you asked, there can still be some errors. Now, map decoding algorithm is probably too complex for a simple enzyme to do. Can there be a simpler algorithm that enzymes which doesn't have a brain, or it has to do some relatively simple task? What can it do, right? It wouldn't do that complicated computing. So it turns out that we propose, so we use this and we can show that enzyme, this algorithm, this complicated map algorithm will simplify to something very simple in some regime of alpha and beta for some particular value of alpha and beta, which is relevant biologically. The algorithm simplifies to this. The algorithm says wherever you see zeros, look at those zeros, and if you think of this as an island of zeros, there is some particular number which called k. So particular k value, for example, k equal to 3. So this k, there is a particular critical value of k, which is given by the property of the sequence. And if you know this k value, the enzyme have to just fill every island which is smaller than k. For example, k is 3. This is a 1 0, just fill it. K is 3, there are 2 zeros, just fill it. Here k is 3, there are 3 zeros, you can fill it. But if there are 4 zeros, don't fill it. Right? So enzyme has to just understand, if there are a large number of zeros, don't fill it. If there are few zeros, fill it. This algorithm is exactly, we show that this algorithm is exactly same as this map decoding algorithm, and that gives you least error. So this is the k value at a particular critical value of k. For example, critical value of kt is 3. That is the least error you would get by just filling 3 or 4, depending on the alpha and beta pair values. This is what we did. You have a mother sequence, and error correcting with, for this particular real sequence, the critical k turns out to be 6, and you do this, you still get about 14% error, but that's the best you can get by any fancy algorithm, and you get a sequence which is pretty as good as the mother sequence. So this is the proposal that we have, that enzymes can look around and fill it just by looking how many nucleosomes are without a mark. That's the only thing that enzyme have to look at. Locally look at few nucleosomes and then decide to fill or not, right? There are some physical mechanisms which would make this possible, and I wouldn't go into the details, which I will be happy to take. So to summarize, what can a machine do? Map decoding algorithm is the best so far, right? We can think of. And we show that if you fill an island of smaller or k, that is as good as map decoding algorithm, and it can reconstruct the experimental data reasonably well. So that's the proposal that we have. As I said, the details are there in this paper you can read, and I'll be happy to take questions. So these are some of the people in my lab who did the work. Sangram did the early part of the work that I did. Nitya, who did the other part of the work, the epigenetic part, who's not in this picture. And these are my collaborators in the funding agencies. And I'll be happy to take questions.
Nobody else is asking. I'll ask one more quick one. Do we know like mistakes in so in the DNA sequence world, we say whenever there is mistake in copying genetic information, it can potentially be pathogenic and disease causing, right? So is there a certain association of mistakes in copying epigenetic and chromatin information leading to like causal of pathogenicity or disease causing?
If there is, so now this is a question that we don't know the answer to very well, in the sense that how much mistake can it tolerate, right? That's the question that some amount is small, so some amount of mistakes it can tolerate, presumably. But if there is, of course, large variation, certainly it can lead to difference in gene expression, and that can have consequences.
And is that tolerance threshold different for different tissue types and organ types, right?
I don't think those things are well understood at all. Those are kind of being studied because even these experiments, as you can see, these are all, like, for example, 2018, 2019 papers, where we are just trying to understand what is the pattern and is it inherited at all or not. Those are the things that we are just trying to understand. And now we have to still understand what happens if there is how much mistake or how much tolerance can there be, and so on and so on.
Anyway, I think, Ashish, we can probably call it a day.
Yes, sir. Thank you, Professor Ranjit, and I'll quickly, for a fascinating talk, and I'll quickly introduce Professor Alok Bhattacharya, who is one of the PIs of the center, and to give the vote of thanks for as we conclude the workshop. So Professor Alok Bhattacharya is a professor of biology and head of the biology department at Ashoka University. He's a recipient of the Robert McNamara Fellowship, Rockefeller Biotechnology Career Development Award, Shanti Swarup Bhatnagar Prize, J.C. Bose Fellowship, and Fellow of Indian National Science Academy and Indian Academy of Sciences. He also served as one of the vice presidents of INSA. Following his PhD at JNU, he trained at the National Transgenic.
I think.
Yes, sir.
I just said that's fine.
Okay. So finally, his current research interests are rare diseases, computational genomics, and biology of pathogenic parasites. Yeah. Over to you, Professor Bhattacharya.
Ranjit, was an interesting presentation. I'm listening to you for the first time.
Thank you.
I really enjoyed it. When we started bioinformatics computation biology in 1990s in JNU, we didn't realize that we'll come all this way so far, about 30 years.
Yes.
It has really gone with this thing. This workshop was quite intense. I can see that few of the hardcore ones survived till the end. But we'll have all the talks recorded so that people can actually look at it in over a period of time. And that's something that will be good. Now, this is something that Ashoka University has started. We have a very interdisciplinary program. Our teachings are also very interdisciplinary.
Yes.
And our program allows people to do, for example, double major. You can work on physics as well as biology, etc. So this is really an ideal place where you can go into interdisciplinary area, and computation biology and mathematical biology are one of our top areas to do that. And we of course have Gautam Menon who's spearheading this area into it. So obviously this workshop is a very, very important event for us. And we are making, this is the second one we are organizing. Last year we also did that. And hopefully the next year we will have it a physical one, not an online version that will help to do that. This year we have, like tomorrow we have a hands-on this thing, and next year probably all of you who come give a talk can actually have a demonstration, and people can try out some of your tools, and we'll work a few months ahead so that we have all the data and everything worked out for this. So that this becomes eventually a center we can all work towards in this whole area of mathematical modeling. Interesting. That's why I was talking to Amitabh. Amitabh used to work with Ram Ramaswamy, and we collaborated at that time. I think this is what excited Amitabh, so he got into computation biology. I think a lot of people came that way. I think the community is expanding, and this is the time that we should get it and try to get into problems which are much more complicated. I mean, already it's complicated enough to actually look at three-dimensional geometry of chromatids from a two-dimensional data. So that has certain issues. So we would really like to go into this and eventually able to model a whole cell. And this is sort of dream that someday I'll be able to see that somebody actually models a whole cell, because a lot of things we can do, for example, new kind of drug discovery, which actually are not designed to target a specific molecule or a specific pathway, which actually does other things. For example, it creates noise or creates perturbation in the system and uses that way to cure problems. Many of the physiological problems or other problems are because of a change in perturbation or change in the network, etc. So that kind of really big problem, we should be able to actually do it sometime. And I hope that this workshop will actually motivate a lot of young people to get into that. And hope that they will start thinking big, and they can actually communicate with all the speakers and develop this area in a big way. I hope that they have been motivated enough, excited enough, and they've listened to some of the top people of the country in this area. Let's hope that we will continue to forward this. And I think, Ashish, we should have a group, people all who have registered. And so we can
do that,
make a society or something, but at least have a discussion group or something we can create in computation biology, where developments, if people have questions, people have queries, there can be a common place where it can be posted related to this area. And one of the experts who are part of this can actually answer these questions. We can initiate that this can become a movement at some time. And I remember the way Ram Ramaswamy and I started bioinformatics in JN in 1990s. Eventually has become a school in JN, where it spread all over the country. So at that time we were one or two people doing it. So I'd like to see mathematical modeling also become something Okay, so anyway, thank you, Janjit, and I want to thank all the other speakers who actually gave their time and really try to explain to all the participants the kind of work that they are doing. And okay, so I'll stop here and wish you all good luck. With that,
I'll just add the recordings for day 1 are already available. We'll hopefully process day 2 soon. And a big thank you to all the speakers and finally to all the participants. Hope you all enjoyed the talks. And as Professor Alok said, we look forward to the next one being in person next year, next edition, and hopefully you can join us. Thanks again. Goodbye for now.
goodbye.
Bye.